Good morning, Satakshi. Good morning, madam. Madam.
Hi.
Hello, good morning, madam.
Good morning, good morning. Earlier it wasn't working, so I had to reload. How are you? Yeah, thank you for sending me the message and all, So,
Pardon, madam.
Thank you for sending all the messages and all.
Yeah, madam.
Yeah.
Actually, we were also not certain whether to hold it on Shivratri day, but the date was decided long back. And then all the speakers were also invited. So that was the reason why, and also I was not keeping well. For about 15 days, I was out of action.
Why? COVID or what?
COVID. I was hospitalized also.
Oh, that's too bad. Many people, NK also, and Archana, all of them were hospitalized.
Yeah. I came back on 29 Jan.
I see.
So the time was too short.
Yeah.
And things kept happening. So we sent all the ABCD members the links towards the end.
Yeah, yesterday morning only we got.
Yeah, because that is when the students did all the things required.
Yeah.
For starting the program.
Yeah. I heard Lakshmi Priya also was hospitalized due to COVID.
Yes.
It's really bad, huh? I had not expected in this third wave about people yeah. Okay, everybody.
Hello, good morning.
Hi, good morning.
Welcome to the program.
Yeah.
Thank you. How are you today?
I'm good.
Thank you for getting up so early.
For us, it is around 9 o'clock in the morning.
Okay. What time do you
Yeah.
What time do you usually start your day?
Yeah, right.
Yeah, it's 10:30 here, so I'm probably ready for bed.
My daughter is in Buffalo.
She is at UB?
She's doing chemical engineering, MS in chemical.
Oh, no kidding. How does she like it here?
She's liking the place. Yeah. And also the university.
It's not too cold for her?
Pardon?
It's not too cold for her? It's so cold here.
It is. But at that age, it's a new experience.
Yeah, I'm glad. And did she enjoy the snow?
Yeah.
What's the weather like there?
Here, the temperatures are rising.
Okay.
And today actually marks change of season.
Oh, it does. Okay. So you're going...
The temperatures are going to rise from today.
That sounds nice. We're really cold here. I'm all wrapped in my blankets. Trying not to freeze.
Your summer temperatures may be like our winter temperatures.
I think that's true. It only gets to what 70, 80 Fahrenheit here in the summer, so it's not too bad.
We use centigrade.
I know I can't. I would convert for you, but I couldn't.
A few days ago, I read a story written for children. In that, the child runs fever, and
Okay.
he is used to temperatures in centigrade scale. Suddenly, he hears the temperature in Fahrenheit.
Fahrenheit thinks he's dead.
He thinks that he is about to die. It was just a mild temperature.
Yeah, and that's maybe the same with my children. They think they were like frozen if we did it in centigrade.
Yeah.
I enjoyed all the talks yesterday, or this morning.
You could join.
That's nice. I joined under my son's name. I had to find my Google password for my own.
I see. So, I was not knowing that you had joined, even though you said that you would be joining. I thought probably you have not joined because I didn't see your name.
No, yeah, I was under William. It was me. I was like, I better try to fix this so they know who I am for my talk later.
Once this COVID scare goes off, we would see an occasion for you to visit our university.
Oh, that would be lovely. I can't wait to travel again.
Yeah.
Are things better there? They seem to be coming
We
down
have
here.
a MOU with your university. Our university signed MOUs some time ago, and Professor Patel has been instrumental in that.
Oh, that makes sense.
Professor M.S. Patel, Mulchand Patel.
Yeah, Milton and I teach together. That's how we know each other. We teach dental biochem together.
Yeah. Right.
I'll have to ask him where he is. Why he's not at the talk now? He's probably sleeping.
I think he would attend.
I think he would.
He's very enthusiastic, and
yeah,
I think he would attend.
I enjoy working with him.
Yeah. He actually was our alumnus.
He did undergrad there?
I don't know what you call it. It is equivalent of MS.
Oh, a master's. Okay.
Because we call BS as graduation.
I see.
And then masters.
And do you have to do a master's?
Yes. Our BS program, so far, we call it BSc, Bachelor of Science. It is for 3 years.
I see. And then the master's would be another 1 year or 2 years?
2 years.
Okay.
We don't have 1-year master's programs here.
Okay.
Because we don't go by credits.
Oh, you don't by credits?
2-year program, and the number of credits are fixed for first year and also second year. Unless and until the students complete all the credits, they would not be graduating. And another thing is that they cannot take what is offered in second year while they are in first year. So they have to spend 2 years.
They couldn't. Do they ever stretch it out, or it's always just 2 years? You couldn't go 3 if you wanted to?
Yeah. Actually, my daughter is completing in 1 year. She's going to complete in 1 year.
Good for her. That's impressive.
Another 3 minutes. After that, we can start your talk.
All right. I know everyone had to get up early. I feel bad.
No, not at all. 9 is a reasonably good time for us to get active.
Yeah, I feel 9's a good time here.
Yeah. But for you, it is quite late. Because of time zones, I could not help it, other than doing it time.
This is pretty good, actually. My husband made me set an alarm. He's like, "Don't fall asleep on the couch." I was like, okay. I did. I set my alarm. So.
You have a big research lab?
It's not too big now. I have 2 postdocs, 2 PhD students, a master's student, and an undergrad. So 6 people now.
Okay.
I just got a new grant, so I'll probably start hiring hopefully soon.
You are planning to hire PhDs now, students for PhD program?
Yes, I think that's what I'll do. It's hard to get postdocs here, so mostly my one postdoc is my husband. So I say I can't marry everyone. So mostly PhD students.
Sanjeev, Dr. Sanjeev Upadhyay.
Yes, ma'am.
There is a request from Professor Patel that he can watch us, but he cannot speak. So he is requesting to be unmuted.
Okay, let me see if I can do that.
I think only students who generated link can do it.
I'll call up Sakshi
and pass on the message. Now it is 9 a.m. Good morning, everybody. Thanks for joining the program so early.
Ratraba, you want to wait till MS Patel sir also joins just for opening remarks if he wants to give something?
Okay.
We'll just wait 1 minute more. Sanjeev, you're finding out?
Looks like I don't have Satakshi's phone number.
Okay, I'll just call. I've got it.
Yeah.
Ravi, can you see if we can give him the...
So I don't see him joining in this.
Yeah, he is not here. Maybe
only if he has joined, you will not be able to speak. He has to join using this link, Google Meet. He might be watching on YouTube link.
YouTube, okay.
Yes.
Yeah, that is.
I'll just send him this link.
Can you send the link once again? Maybe before close of the talk.
Yeah, good idea.
Yeah, we can ask him. We start the program now. It gives me pleasure to introduce Professor Christine Scanner-Tooley. She is working as Associate Professor in the Department of Biochemistry, Jacobs School of Medicine and Biomedical Sciences. She did her BS in Biochemistry from University of Rochester, magna cum laude in 2000. She received PhD in Biochemistry, Cell and Developmental Biology from Emory University in 2005. She received a fellowship in biochemistry and worked in University of Virginia in 2011. Later, she worked as assistant professor, biochemistry and molecular genetics in the University of Louisville from 2011 to 16. Then she joined University at Buffalo as assistant professor of biochemistry in Jacobs School of Medicine and Biomedical Sciences. And she continued as assistant professor between 16 and 19. In 2019, she joined as she became associate professor in the Department of Biochemistry, University at Buffalo, Jacobs School of Medicine and Biomedical Sciences. Her research focus is aging, cell growth, differentiation and development, DNA replication, recombinant and repair, gene expression, molecular and cellular biology, signal transduction, stem cells, transcription factors. Her main goal is to understand the role of N-terminal methylation in human development and disease. Now I welcome Professor Christine Tooley to present her talk.
Okay, can everyone hear and see me?
Yes.
Okay, thank you.
Yes.
I can't see you, so if you have any questions or you can't hear me, just feel free to jump in. So thank you for that introduction. I really enjoyed, and thank you for this opportunity to speak. I really enjoyed the talks that I was able to get to this morning for me, but yesterday for you. And I'm happy to start it off today. Again, I'm Christine Tooley. I'm Associate Professor from the Department of Biochemistry at University of Buffalo. And I'm going to talk today about the work that we do in my lab on N-terminal methylation. So I'm one of the few people in the world that work on N-terminal methylation. So I feel like my job is now to spread the word that it's a very useful and important post-translational modification. So we work on N-terminal methylation, and it's different from the...
Excuse me. Can you please go to presentation mode? We can see those all of the slides together as small stats.
Oh, you can't. Okay, I can't see that. Mine looks like presentation mode. Is that better? No.
No.
No, we can see only slide sorter mode. So maybe you can switch to presentation mode.
I was in presentation mode here.
The window which you have selected is not that, maybe.
Oh, okay. I know what you mean. Okay, hold on one second. I don't know how to. No, we don't want that. So it won't share. So you just see my slides. Is that what you're saying?
It was showing the sorter mode, not the presentation mode. We can see all the slides, not the one slide you highlighted.
So what you can do, I suggest, is you first put your slides on full screen mode and then go back to Google Meet. There you click on present now and select only the window which.
How do I? Because when I do that, it's full screen. When I do the
there's one more option.
There is an arrow.
Yeah, you can go for a lateral bar and select one of the slides and reduce this width of the lateral bar. That way it will be full screen. Many times this problem happens.
Okay, I see. I could try that. Okay. Is it sharing now?
Yes, yes. Now you can reduce the width of lateral bar.
Do what?
Or what you can do at the bottom right, you see, you can slide the bar towards more, more, 100 and you can make it like 130% or so.
Oh, to zoom in, you mean?
Bottom right, you see that there is a plus sign. So
yeah, okay.
Yes.
Yeah,
you make it around a slightly less. Fine. This is fine.
Is that good?
Yes.
Yes.
Okay. Okay, so you can see that now?
Yes. Yes.
Okay. Okay, I'm sorry. All right. Okay, let's see, how do I... Okay. So, so we work on internal methylation, and it's a little different than the methylation of proteins that you usually think of, which happens on arginines and lysine side chains. Our methylation only happens on the alpha amino group of the protein, so the very end terminus of the protein. And we can have mono, di, or trimethylation, and then also if it happens to be a proline, then you can only go up to dimethylation. But what's interesting about the trimethylation and the proline dimethylation is that it does put a pH insensitive positive charge on the end terminus. So I got interested in internal methylation when I discovered the first known eukaryotic N-terminal methyltransferase during my postdoctoral work. And I named it N-terminal RCC1 methyltransferase, or NIRMT1, after RCC1, which was its first known substrate. So it was interesting in that it was a nuclear member of the metal family of 7 beta strand methyltransferases. It wasn't one of the set domain methyltransferases that you find for the histone methyltransferases. What was nice about it is that it did have there were a few proteins known to be N-terminally methylated before I discovered the methyltransferase, and they had a very set XPK consensus sequence where X could be any amino acid followed by a proline, and then it was either lysine or argine on the N-terminal. So using this consensus sequence, we were able to predict over a couple hundred different substrates. And then I went on to actually show that there was an expanded non-canonical census that added another about 150 targets. So overall, between the canonical and non-canonical targets, we have about 300 predicted targets of NIRMT1. And I just list a few here that I'm going to talk about today in the talk. The first, RCC1, which again was its first known substrate. Also retinoblastoma protein, which is a very famous tumor suppressor, DNA damage binding protein 2, fingers and protein 2, and then also myosin late chain 9. But these are just a few of the different substrates that NIRMT can methylate. So one of the first things we did after we had the modification and also the enzyme is to try to figure out what the biochemical role of the modification it was itself. And so we took advantage of that we had a known consensus sequence, and we made mutations in this consensus sequence to look for ones that disrupted methylation. And we found that if we could mutate the lysine to a glutamine or a K4Q mutation in the consensus, that we could prohibit methylation of the substrates. So then we would take different substrates, express the wild type and then the mutant form, and see if we could look for different phenotypes. And the first protein we did this for was RCC1. So if you take RCC1 wild type as a GFP fusion expression cells, what you see is that it localizes nicely with DNA. So we have the DNA here in blue, we have the mitotic spindle in red, and wild type RCC1 co-localizes to the mitotic spindle with the DNA, and it's a very nice tight localization. So if we make a mutant now that can't be N-terminally methylated, this K4Q mutant of RCC1, what we see now is that this RCC1 is very diffuse and no longer co-localizes with the DNA. And we went on to show that it's this N-terminal methylation is actually regulating protein DNA interactions and not actually protein histone interactions. So we were able to show that methylation regulates protein DNA interactions, and then many other groups have gone on to show this for other substrates. And I just show an example down here from Yincheng Wang's lab, where they use DNA damage binding protein 2, and they show the same thing. They make a K4Q mutant, and they show that here wild type co-localizes with DNA damage, where if you have this mutant that can't be methylated, it's diffuse and can't localize to the damage as well, shown here in green. So we had now an enzyme, we had a modification, and we have a biochemical function that it's regulating protein DNA interactions. And so if you look at the list of different targets, what you see is that many of them are DNA damage proteins or transcription factors. And so that makes sense, being that this modification regulates protein DNA interactions. And so the next thing we wanted to do was to see if they actually regulate these processes. So if you lose N-terminal methylation, can you disrupt DNA damage repair and transcriptional regulation? So just showing a few examples of things that we've done over the years. This again is from Yuncheng Wang's lab. He looked if you lose methylation of this DDB2, do you disrupt nucleotide excision repair? And they showed here if you have a mutant that can't be methylated, they're not able to fix these pyrimidine dimers that appear. So there's impairment in nucleotide excision repair. My lab went on to look at double-strand break repair. So we treated cells that were knocked down for near-empty-1 with things that make double-strand breaks, synthesis gamma radiation and etoposide. And what we were able to see is that cells without near-empty-1, without N-terminal methylation, are more sensitive to double-strand breaks. We then looked at different transcriptional targets. So myosin light chain 9 has a nuclear role as a transcription factor. And what we saw is now if you lose its methylation, you make a mutant that can't be methylated. It's unable to promote transcription. And the same for another target, ZHX2. You have this K4 mutant in light gray. And when you express it, it's not able to promote transcription as well as wild type in the dark gray. So we were able to collect all these information now. We have the modification, the enzyme, the biochemical function, and we've shown that if you lose this N-terminal methylation, you can disrupt things like DNA damage repair and transcriptional regulation. And so thinking about this, we wanted to go even bigger and look if there were more phenotypes that we could find from loss of this methylation. And when you think about DNA damage repair and transcriptional activation, those things are often misregulated in cancers. So next we wanted to look to see if there was a role for near-empty-1 and N-terminal methylation in oncogenesis. So the first thing we did was look in the OncoMind database for cancers that maybe had misregulation of near-empty-1. And it was interesting in that there were a few cancers that had near-empty-1 lower expression, and then there were also a few that had near-empty-1 that were overexpressed. So we started with the cancer where they had low expression of near-empty-1, and one of the predominant types of cancer there was breast cancer. So we took a breast cancer cell line, MCF7 cells, and we used lentivirus to stably knock down near-empty-1, which would be the black bars, and we looked at different oncogenic phenotypes. And so what you can see is that when we knock down near-empty-1 in these MCF7 cells, we can increase their growth rate over 5 days. We can also increase their ability to migrate over 3 days. And then we took these knockdown cells and we put them in the fat pads of nude mice, and we looked to see if we could have looked at tumor growth over just 7 days. And only after 7 days, you could see that there's a significant larger tumors grown when you have a loss of near-empty-1. And so here they're quantified, and here's a picture too. So on each side of the mouse, we had a control and near-empty knockdown. And when you have near-empty knockdown, you get much bigger tumors in just about a week. So that was interesting. We showed that near-empty-1 could act as a tumor suppressor. And if you lose it, you're going to gain oncogenic phenotypes. And these are in cancers that have traditionally low near-empty expression. So then we took a type of cancer that have high near-empty expression, and we wanted to see if we can see the opposite in these cells. So one of the most high expressing types of cancer for near-empty-1 is colon cancer. So we took HCT116 colon cancer cells and we knocked it down, and we just wanted to see if we could slow growth. And so here in the red is the control, HCT116 cells. And when we knock near-empty down in the purple here, we can see a significant decrease in growth over 4 days. So near-empty is interesting in that it can work as both a tumor suppressor and an oncogene, and dependent on a type of cancer. And we think we haven't got into this pathways yet, but we think it's going to be dependent on exactly which near-empty substrate is predominating in each cancer type. So we found a role for NERMT1 in oncogenesis, but I was also interested in, because nothing was known about it at the time, I want to know if it also perhaps could be playing a developmental role. So while we were doing the oncogenic experiments, my lab decided also to create a NERMT1 knockout mouse. So this is before the CRISPR knockout mice became available as a technique. So this is your traditional NERMT1 knockout mice. It's not conditional in any way. It's constitutive knockout. And so I wasn't sure what was going to happen. I thought perhaps they might be dead, or at worst they might have no phenotype. But they had actually a very interesting phenotype. And the first thing that we were able to find is that the females were sterile. So we weren't able to mate homozygous females with homozygous males. So we had to mate homozygous males with heterozygous females. And so when we saw the litters from these mice, the very striking thing, the first thing we saw is that the homozygous litter shown here, pups, are much smaller than their heterozygous litter mates. And then when we watched them grow, what would happen is that the NERMT1 homozygotes would die a much earlier death. So only about 40% of them lived over a 1 month old, and only then about 30% of them made it past 6 months old. And so when we looked at these guys that made it past 6 months old, we saw some very interesting phenotypes, and a lot of them were considered premature aging phenotypes. So you can look here, this is a 9-month-old mouse. Here's the wild type, and you can see in our mouse, they are much smaller, and they already have this kyphosis of the spine. They're already losing their hair. They also have premature graying. When you look at their skin, you can see these collagen bundles, which are reminiscent of skin fibrosis. We looked at the ovaries of the females because they're infertile, and we found them to be highly cystic, which is consistent with PCOS phenotypes. And lastly, one of the most tricky phenotypes is that we have this liver degeneration. So by 3 months old, you can see here, this is a healthy mouse liver. It has about 4 lobes. And our mice, the liver is mostly degenerated down to 1 lobe, and you can see it's highly necrotic. So this is very interesting and not all that surprising when we thought about the role of NERMT1 in DNA damage repair. We have these mice that seem to be accumulating damage and that are accumulating also these premature aging phenotypes. So this is pretty much a very brief overview of the things that I did in the first half of my lab. And so now we took all this data and we're focusing on 3 more kind of focused questions about more molecular mechanism and how NERMT1 is working. So the first question that we're interested in asking in my lab is, how is NERMT1 regulated? The second one is, does N-terminal methylation interact with other N-terminal post-translational modifications as part of a code? I'm not going to talk at all about this question today, but just to say we have found some very interesting data that there are proteins that can be either N-terminally methylated or N-terminally acetylated. And so we were the first to show that these can happen on the same protein. And it seems to be that when they switch off on a protein, the N-terminal methylation regulates its role in the nucleus, and the N-terminal acetylation regulates its role in the cytoplasm. So again, that's another story that I won't talk about today, but of interest of my lab. And the last question that we're interested in now is, what NERMT targets contribute to the observed knockout phenotypes? So we have these very nice oncogenic phenotypes, and we have these very nice aging phenotypes. And so the question is now, how do you get from loss of N-terminal methylation to these phenotypes, and what exact targets are involved? And so that is going to be a good chunk of the things that my lab will be working on going forward. So I'm going to start with a story first about how NERMT1 is regulated, and these are new papers from my lab. And then while we were answering this question, we ended up coming back and answering a little bit of this question too. So I'm going to start here, and we'll end with this question here. So I have a postdoc in the lab who's very interested in transcription, and I had to laugh at Anne's talk yesterday when she said you can work on anything but transcription. In my lab, the rule is you can work on anything but lipids. So when he came to me with this project, I was okay to go forward with it. He wanted to know how NERMT1 is transcriptionally regulated. There was really nothing known about this at the time. So when he looked at the databases, there were 11 predicted near MT1 splice variants, but only 4 were predicted to be full length. And so he took these 4, about 1,200 base pairs of these promoter region up ahead of the predicted start sites of these 4 variants, put them in the luciferase construct, and looked to which one had the most expression. And what he saw is that splice variant 2 really had the highest expression. It looks to be the main splice variant of NearMT1. And so he wanted to take this 1,200 base pair region, cut it down, and see which one is actually the promoter region. So we can see here, I'll just go through quickly. He was able to cut it down to 12 to 8 to 600 base pairs, still had the same amount of transcription activity, even when you got to 300 and 100. So he can get it down to 100 base pairs and could still have enough, the same promoter activity. And then he wanted to see if you needed anything past the start site. So he extended it, it's 100 base pairs plus 75 past the start site, and that didn't increase transcription either. So from there, we decided that you really only need this 100 base pairs right in front of the splice variant 2 for transcription. And so this is it here. And you just want to see what kind of transcription factors are predicted to bind at this site. And so we found 3 different motifs. There was an SP1 motif, an ETS motif, and a Cre-ATF motif. And so first he just made truncations that removed 1 or 2 of these motifs at a time to see if he could alter transcription. And so here's the one here that removed the SP1 motif, and there was no change. And then here he had this one here where he removed ETS and the Cre, and that significantly reduced transcription. So that indicated that transcription of NearMT1 could be being related by these ETS or Cre transcription factor families. And so we further went and then tried to mutate these motifs to see if we could disrupt transcription. If you mutate the SP1, there's no difference. If you mutate just the ETS by itself, there's no difference. SP1 and ETS together, no difference. It's not till when you get to the Cre site that you now see a significant decrease in transcription from the 100 base pairs shown here. If you add a SP1 mutation to the Cre, there's no difference. However, if you do add an ETS mutation to the Cre, you get a significant more decrease in transcriptional activity. So this indicates that ETS is contributing, but Cre is probably the predominant factor, because if you mutate ETS by itself, you get no difference in expression. And then again, the triple mutation SP1, ETS, and Cre is no different from ETS and Cre alone. So we took this to mean that this 100 base pair region in front of NearMT1 is its transcriptional promoter, and that it's ETS and Cre perhaps that are driving transcription of NearMT1. So we wanted to make sure that we can actually see either part of the Cre or ETS family of transcription factors actually binding this 100 base pair sequence. And so I'll try to go through this quickly. We made a 100 base pair just DNA oligo, and we incubated it in cell lysate, and we looked to use Western blot to see if we could see transcription factor binding. So we had the 100 base pair promoter region, and then because Cre bind so close to the end, we also had one's a 100 base pair, but it's shifted, so 80 before the transcriptional start site and 20 past the transcriptional start site. And so when we use these oligos to look to see which transcription factors bind, we started with the Cre-ATF transcription factor families, and we looked at proteins that were predicted to bind in this region from the ENCODE database. And we found that, yes, CREB1 does bind the NearMT1 transcription start site when you do shift it to the 80-20, but neither ATF1 or ATF2 did. So this looks like CREB1 could be the potential transcription factor binding at the Cre site. We did the same for the ETS transcription factors, and we found that both ALK1 and ALF1 could also bind the promoter region from cell lysate, but ETS1 and GABP alpha could not. So we do have some transcription factor selectivity in each family. We also looked at SRF transcription factors because they're known to bind with the Cre and ETS family, but we found that they were not binding the NearMT promoter here. And then if we made a mutation in the Cre, we could see that this in the Cre binding site, we can get rid of Cre binding. You can also get rid of the ETS binding as well. So that again shows that Cre is our main binder and is some have cooperativity with the ETS family transcription factors. So, so we took this to mean that CREB1 is probably our dominant transcription factor driving NearMT expression. And CREB is a very interesting transcription factor, and then it is evoked in a lot of different times. And that would be So these include DNA damage, they include cell cycle, they include stem cell differentiation. And so we wanted to use this to see what kind of conditions would stimulate NERMT1 expression. When would NERMT expression be important? So we started, because of our background in showing that NERMT1 had a role in DNA damage, we thought maybe CREB would induce NERMT1 during times of DNA damage. So we treated ourselves with doxycycline, camptothecin, etoposide, and hydrogen peroxide, thinking that we would see induced expression of NERMT1, but in fact, we don't see any change in NERMT expression with these DNA damaging agents. So then we moved on to serum starvation, and we looked to see there if we would see NERMT1 expression being induced. And when it was interesting, when we actually did the serum starvation itself, we see NERMT1 expression go down, but then we do see 6 hours after serum reintroduction, we see now this NERMT1 expression being induced. And so we wanted to see if that was CREB dependent, and so we treated with CREB inhibitors, and we looked again at the 6 hour time point, and when you don't have a CREB inhibitor, you do see this increase in NERMT1 expression, but if you treat with the CREB inhibitor, you get rid of this induction. So this is showing us that NERMT1 induction at 6 hours post-serum starvation is CREB dependent. And this is different, this is just an example here, from a lot of the other CREB genes, which are transient quick expression. So CREB's known to induce expression of genes quickly after serum starvation, so within 30 minutes. But now NERMT1 is a different, more delayed response, which we see at 6 hours. So we didn't see NERMT1 expression change in DNA damaging, but we do see it with serum starvation and reintroduction. So the next thing we wanted to look at was stem cell differentiation. So we took C2, C12 mouse myoblasts, so muscle stem cells, and we differentiated them in culture and then looked at NERMT1 expression levels. And what we see is that with 1 day of differentiation, we see a significant increase in NERMT1 expression, which maintains through 2 days and starts to go down after 3 days. And so we want to see again if this is CREB dependent. So we used a CREB inhibitor, and if once you do differentiation with the CREB inhibitor, you see no induction of NERMT1 mRNA levels. So again, this is telling us that this NERMT1 expression in muscle cell differentiation is dependent on CREB as a transcriptional regulator. So that I won't go through this too long, but just this was CHIP to show that in vivo that CREB1 could bind actually to the NERMT promoter. So we just did CHIP and ACT116 cells to see if we could see CREB1 at the NERMT promoter. Now that we know that it comes on at serum starvation, and so you can see here, yes, that NERMT1 does bind, or CREB does bind the NERMT1 promoter during times of serum starvation and recovery. And as does ALK1 to a more limited extent. So we know now that CREB can bind the NERMT1 promoter both in vitro and in vivo. And so now we want to get to the biological relevance of this. So if we see that CREB is inducing NERMT1 expression during muscle cell differentiation, is this biologically relevant? Does NERMT1 actually play a role in muscle cell differentiation? So in order to get to this, we made a CRISPR-Cas9 NERMT1 knockout line in the C2, C12 cells. And then, so you can see here, that was a very nice knockout. We lose all N-terminal methylation, and we lose NERMT1 expression. So then we took this NERMT1 knockout line, and we treated it with muscle differentiation media, and to see what happens if we disrupt muscle differentiation. And the first thing we notice is that PAX7, which is the general regulator of muscle cell differentiation, is not expressed in NERMT knockout cells. It's not even expressed at D0. So these cells not only don't begin muscle differentiation, they seem to lose their muscle identity when they lose NERMT1. So we see no PAX7 expression at all. And so we looked a little further down the differentiation pathway. So we looked at myogenin, and you can see that it comes up at day 2 in normal cells of differentiation. And again, our cells never really express myogenin. So just to be thorough, we went all the way down to myosin heavy chain, which is one of the later markers of muscle differentiation. You can see in these wild type cells, they can go and they form these myotubes, which stain nicely for myosin heavy chain. And when you look at our NERMT1 knockout cells, what you see is, again, they aren't able to form these tubes at all. And you see no myosin heavy chain. So this is very interesting to us. We have these muscle myoblast cells, which seem to have not only cannot differentiate anymore, they seem to have lost their muscle identity. And so we're trying to figure out exactly what these cells now were. And so muscle myocytes or myoblasts come from mesenchymal stem cells, but so do a lot of other types of stem cells. So from these cells, you can get connective cells, you can get chondrocytes, which are the cartilage cells, adipocytes, the fat cells, or osteoblasts, with the bone cells. So we were wondering if we lost NERMT1 and we've lost this muscle cell identity and their capacity to differentiate, perhaps they've taken on another one of these different types of identities. So we took these NERMT1 C2C12 muscle cells and we did quantitative RT-PCR to look to see if they're perhaps going down another lineage. So just as a control, we use another muscle marker, MyoD, and you can see, of course, they do not express this MyoD, again, confirming they're not going down the muscle lineage. We then looked at PPAR gamma, which is a marker of adipocytes, and you can see no difference between our knockout and control. Then we looked at SOX9, again, which is a chondrocyte marker, and there's no difference between the knockout and control. What was really stunning when we look now at the osteoblast markers, we see a huge increase in alkaline phosphatase expression and also in type 1 collagen. And so we thought, oh, so they're not actually differentiating into muscle cells, but now they've started to take on bone characteristics. And so we tried 3 more different osteoblastic markers, RUNX2, Osterix, and osteocalcin, and all of them are significantly increased in these NERMT knockout cells. And so this is without differentiation. So this is just these muscle cells grown normally in culture without adding differentiating media. They are already taking on these bone characteristics. So what seems to be happening is that when you lose NERMT1 in these muscle stem cells, they not only can't differentiate, they start to lose their muscle identity and start to switch to bone cells. So when you have NERMT around, what we think it's normally doing is preventing bone differentiation and promoting muscle cell differentiation. So this turned out to be, it started the project looking at transcriptional control of NERMT1, and now we've gotten a role for NERMT1 in stem cell development. And so when we thought about this in combination with our NERMT knockout mice, we thought, well, maybe NERMT1 is having a more general role in stem cell differentiation, because we see all these premature aging phenotypes, and a lot of these things can result from misregulation of stem cell populations. And so we thought we should probably look at a different type of stem cell and then use these mice to see if we can find defects in stem cell development and differentiation. So the first group of stem cells that we decided to look at were these were the neural stem cell niches. And we basically chose these because I had a new graduate student in the lab who comes from a neuroscience lab, and he was very interested in looking at the brain of our mice. We had never really looked at any of the phenotypes in the brain. And he's, after looking at these aging phenotypes in the other organs, he really thought there would be some differences in the brain. And so to start with, we decided to start with the areas where the postnatal neural stem cell niches are. And so there's 2 areas in the mouse adult brain where we have neural stem cell niches. And the first is the subventricular zone of the lateral ventricles. So when you're looking at mouse sections, these are the lateral ventricles here. We'll see a few of these coming up. And in the line, in the sides of these lateral ventricles is where the stem cells reside. The second place in the adult mice is the hippocampus. And so there'll be the region up here. And again, we'll be able to see that better in the next slides. But up there in the hippocampus, this is the second region where we have these adult stem cell niches. So we decided to focus on these 2 areas when we started looking at the mouse brain to see if we could find first any morphological defects. So first we looked at the lateral ventricles. Again here, these are the lateral ventricles, these white hollowed out holes. And you can see we started at P14 and we looked at aged mice. So P14 is 2-week-old mice, and there was no difference really in the lateral ventricle volume between mice at P14 and wild type or control. But by the time we got to 6 weeks, which is quantified down here, we get this huge increase and significant increase in lateral ventricle volume. And this volume gets even bigger by 3 months and then holds steady at 6 months. So that was very interesting. We now have a very gross morphological defect in these lateral ventricles that house the neural stem cells in adult mice. And so we're interested to see, how this arise. So they could arise from a overall expansion of brain size, or these can enlarge at the cost of surrounding a neuronal tissue. And so we looked at overall brain size to see if the brain was bigger all around. And so when we did the whole brain quantification, we saw this not true, that brain in the empty mice actually shrink, and you can see that here. And so, but then we wanted to look at different tissues. So we looked at the cortex, which is out here, which we wouldn't expect to be affected so much. And then we also looked at the striatum, which is adjacent to lateral ventricles, which you might predict would shrink as the lateral ventricles enlarge. And that's exactly what we saw when we quantified the striatum here and here. We see a significant decrease in near-empty mice. Here we get these lateral ventricles that are expanding, and we're getting this shrinking of the striatum and the adjacent areas. So then we want to look for morphological defects in the hippocampus. So again, here I'm just showing you the hippocampus. It's broken up into two regions, the dentate gyrus here, and then the cornu-ammonis region, which has three subtypes, CA3, CA2, and CA1 here. And this is what it looks like actually in the brain here. So we did our crystal violet staining and just looked at our hippocampic morphogenesis. And this was really interesting. So at P14, here's our wild type and our knockout mice. They look pretty much the same in the hippocampus. At 6 weeks, it's true, very much the same. But by 3 months, what we see is the dentate gyrus, which is here now, and the CA3 are significantly decreased. And we quantified that down here. And then by the time you get to 6 months, you'll see that the dentate gyrus is almost completely gone, as is the CA3. Again, that's quantified down here. So now in these areas where we have these stem cell niches, we see huge morphological differences in our mice. And so that, again, made us think that maybe we're having problems with stem cell regeneration and neuronal repopulation in these areas. So then we just wanted to quickly look at behavioral readouts. So we wanted the hippocampus controls memory and the striatum controls movement. So we wanted to see if there were associated behavioral deficits with these. And I'll just go through this quickly for time. We use the Barnes maze test to look at memory. So basically you put the mouse in a maze and you shine a light on it and it wants to get out. And so you attach a escape hatch to one of the holes. And then you put the mouse on it for a different amount of training period so it can learn where the hole is. And then when you take the escape hatch away, the trial means how much time does it spend near the hole that used to have the escape hatch, meaning it remembered it, and then how much time does it spend at the other hole. So you just basically do a ratio of time at correct hole minus time at incorrect hole over total time. And that gives you something called the spatial memory index. And if it's over 1, that means they remember. And if it's under 1, that means they don't. And so this is a short-term memory. So we gave them 2 training trials to learn where the hole was. And then 15 minutes later, we put them back on to see if they could remember it. And at all 3 age groups, our mice cannot remember where the escape hatch was. And so there was some concern that they weren't able to learn it in just the 2 trials. So we did the same exact thing with 10 trials to see if you give them 10 trials, can they learn where the hole is? And even though our mice were somewhat slower, they at every age could learn where the hole was. So it's not that they can't learn it, it's just that they can't remember. So we tried then a long-term memory experiment where we do the 10 training trials and then leave them for 24 hours, put them back on and see if they can remember. And again, all 3 age groups, our mice are not able to remember where the escape hatch was. So now they have both short and long-term memory impairments. So we saw this degeneration of the striatum too. So we looked at motor control. And if you do a rotor rod for motor coordination, we really didn't see much difference. There was a slight significant difference at 6 months. But what was really striking is that when we do an open field test, which looks at speed and distance traveled, what we see is that our mice are hyperactive. So even though they're having striatal neurodegeneration, they're not slower, they're actually more hyperactive. So we have some reasons for this. There are neurons specific in the striatum that inhibit movement. And so we're thinking perhaps that these neurons are specifically dying in our mice. Okay, so I'm sorry that's a lot to take in, but I will get to the stem cell stuff. So we have these morphological differences in the areas where the stem cell niches are, and we have these behavioral deficits. So then we want to actually look more molecularly at the stem cell niches to see if there's a problem with stem cell development. And so I'm just going to show you briefly. So this is the SVZ. So this is the zone in the lateral ventricles. and they have the quiescent stem cells, or NSCs, and these stand for the marker GFAP, which you can see here. These will differentiate into the amplifying progenitors, or the intermediate progenitor cells, and these are going to be marked by SOX2, which will then differentiate into neuroblasts, which are marked by double cortin, or DCX, and these neuroblasts will then ultimately differentiate into neurons. It's pretty similar in the SGZ and the hippocampus. We have the GFAP positive type 1 radioglial cells. These are the quiescent stem cells. They will differentiate into your immediate progenitor cells, which stand for SOX2, which then differentiate into neuroblasts, which stand for double cortin, and then ultimately into neurons, which stand for nu-N. So we just wanted to use all these different markers to look at the different populations through differentiation to see what was different in our mice. So first we looked at GFAP. Again, this is a marker for the quiescent neural stem cells. And they're going to be, we're looking here at the SVZ, so they'll be in the walls of the lateral ventricles, and these are P14 animals. And we can see here we get very nice GFAP staining in our wild type mice, but in our NAMT mice, there's like a void. There's no GFAP staining. So there's a pretty significant loss of GFAP staining in our SVZ. So this means that we have a depleted quiescent neural stem cell population. So then we thought we would look to see, well, have all these, were these cells never here, or have they prematurely begun differentiation? So we looked at the next marker for the intermediate progenitor cells, which is SOX2. And what we see in our near-empty mice in green here is that we get this large expansion of these intermediate progenitor cells, these SOX2 positive cells, and that's quantified here. And then we were wondering, are these, so we have all these intermediate progenitor cells, are they themselves dividing, and that's why we have so many more, or did they come from somewhere else? So we co-stain these SOX2 cells with Ki67, which is a marker for proliferation. And when you do the ratio of SOX2 Ki67 double positive to total SOX2, we actually see no difference. So this told us is that we have this expanded population of SOX2 intermediate progenitor cells, but that they themselves are not dividing, so they're coming from somewhere else. So we follow them on. We want to see if they differentiate into neuroblasts. So we stain with double cortin. And you can see here we do have an expanded double cortin population compared to wild type, and that's quantified here. And then we also wanted to see if they were proliferating. So we took the ratio of Ki67 to double cortin. And when you do that, you see no difference. So we have higher proliferating, but we also have higher double cortin. So these cells are proliferating, but no more at a rate no more than wild type. So that tells us we're missing our quiescent stem cells, and we have an expanded batch of IPCs and neuroblasts. So we wanted to know where these cells are coming from. And this speaks to an earlier expansion of quiescent stem cells that have begun premature differentiation, haven't already gone through differentiation by P14. So we wanted to go earlier into just at birth. So we looked at P0 to see if we can see an expanded population of the quiescent stem cells at this time. And that's exactly what we see. So this is GFAP staining, again, at P0. So these animals were just born. We quantified the GFAP intensity, and at P0, it's much higher. And you can even look at the width of the stem cell zone here. It's much bigger in the wild type mice, and that's also, or in our mice, it's quantitated here. So what's happening, it looks like, is that we get a really expanded population of quiescent stem cells at birth, but they prematurely enter differentiation, and by P14, they've already gone through the quiescent phase, and then now they're turning into IPCs in neuroblasts. And we also looked to see if these cells here at P0 were proliferating still. Well, what we found is that they're actually proliferating less than the wild type mice, which tells us that somewhere in embryogenesis, this new expanded quiescent stem cell batch was made, and already by P0, they themselves have stopped proliferating. So we have this expanded quiescent stem cell pool at 0. We know by P14 that it is depleted, and so we were wondering, does it stay depleted or does it return? So we looked out at 3 months, and what we found is that at 3 months, you can see here there's a nice GFAP positive zone in the wild type, but that is gone in our mice. And again, we looked in these cells. Obviously, they're not there, so they're also not proliferating. So that was very interesting. We have these stem cell populations now. We see we have an expanded quiescent stem cell population right at birth that does not stay quiescent and then begins to prematurely differentiate. And so when we looked in the literature to see if there were any other mice reminiscent of this, what we found is that mice that lose retinoblastoma protein in these neural stem cells have very similar phenotypes. And I spoke very earlier ago that retinoblastoma protein is actually one of the near-empty targets. And so we were wondering if loss of near-empty one leads to RB loss of a function in these mice. And so just really briefly show what they have in these mice is we get an expanded population of SOX2 cells, and we get an expanded population of double cortin cells, just like we see in our mice. What they also show is that these cells go on to differentiate, but ultimately die. So if you look at new-N staining of mature neurons, you can see you get a much decreased density of these neurons, and that's because they're dying. And they found that they're dying is because they're not able to exit cell cycle. So neurons, once they differentiate, are post-mitotic, so they have to leave cell cycle to survive. But when you lose RB, which of course is a cell cycle repressor, you can't exit cell cycle and you end up staying in cell cycle, and this causes death. So we were interested, we see this neurodegeneration in our mice. We're interested if maybe that's what's happening in our mice, that these expanded neuroblast populations are differentiating, but then they're ultimately dying. So we did our new-N staining, and this will be your subventricular zone here, and this is the striatum where we see all this neurodegeneration. And what you see is you can see the staining with new-N, and a lot of these neurons also co-stain with Ki67, and that's the yellow here. And so we've quantitated over here the new-N Ki67 double positives, and you can see there's much increase in our near-empty mice. So this tells us that we have these expanded population of differentiating cells, and they do differentiate, but they're not able to leave the cell cycle. And so we wanted to see, does this cause them to die? So we did cleaved caspase staining. It's hard to see in this picture, but by 6, we looked at 2, 3, 4, 5, 6 weeks, and it takes it to 6 weeks old, but we see now a significant loss of new-N density, and we do see this cleaved caspase staining. So this is very reminiscent of the RB mice, which is very cool. We have these stem cells that can't stay quiescent. They go on and differentiate, but ultimately they can't leave cell cycle, become post-mitotic, which causes them to die, and then this results in the neurodegeneration. And so I said very early that we're interested in trying to connect phenotypes to near-empty targets. And so we wanted to see here, well, is RB misregulated in our mice? So we looked at RB targets, cyclin A2, cyclin E2, and E2F1, and we do see RB usually represses these genes when it's functional, and in our mice, we now see a huge de-repression of these genes. So then we want to look at RB protein levels. When RB becomes deactivated, it becomes phosphorylated and then degraded. So we looked at RB phosphorylation first in our mice, and that significantly increased, which is quantified here. And then we looked at total RB levels, and they go down. So this tells us losing near-empty 1 results in RB1 being not N-terminally methylated, which means it becomes phosphorylated and degraded, and it's not able to repress its target genes. Something interesting else we found, they show that RB is promoting apoptosis in these neurons that can't exit cell cycle, but they weren't ever able to show which target it was. So RB is known to misregulate PUMA and APAF1, and that leads to apoptosis in these dying neurons. But neither of those things were misregulated in the other studies. And so we looked at them in ours too, so there was no change in PUMA. APAF1 we couldn't pick up, but we thought of another RB target, NOXA, that could be driving apoptosis in these cells, and that is in fact significantly increased here. So we have now a beginning of mechanism where we have near-empty 1, loss of that, you get no methylation of RB, which results in its phosphorylation, de-repression of its target genes, and increase in target genes that promote apoptosis. Okay, so overall, this is our model now that we've come up with of how near-empty is working for at least in the neurostem cell niches. So we believe near-empty 1 is important to keep stem cells in the quiescent state. And so when you need stem cells to go on and start dividing, self-renewing, and also differentiating, then you're going to turn near empty off. So when they're in the active state, in the active part of the niche, you're going to want near empty off. And so that allows it to both replicate and begin differentiation. And so when we don't have near empty one, that's why differentiation can go as planned. And so then it's once these neuroblasts migrate and differentiate into fully mature neurons and want to exit the cell cycle, that's when we want to turn near empty back on. And so when we don't have near empty, they're not able to exit cell cycle and they end up dying. Okay, so just to wrap everything up then, what we want to do now in the future with these projects is look what's happening downstream of RB. So we really need to start working out the whole molecular pathway, looking at, so we've shown near empty loss leads to RB misregulation, and so we need to know which RB target genes are misregulated. We found a couple candidate genes, but we'd also want to look comprehensively at RB targets. So we want to use cut and run analysis for that. A really interesting thing about RB misregulation in the brain is that if you have misregulated RB, you also have activated CDK5, which then phosphorylates tau. And so what we see in RB mice, when you have loss of function, is that you get these tau tangle formation. And so what we're really interested in looking at now in our mice is, do our mice also form tau tangles? And so I think that'll be very interesting. And then RB is also really heavily involved in muscle stem cell differentiation. So we need to go back to our muscle model and look to see if RB is involved there mechanistically. We see, there's our role for RB in our mice, but in the neural stem cells, but there are many other neuroempty targets that can be involved. And so we want to do some single cell sequencing of our wild type neuroempty knockout neural stem cells, look to see what other signaling pathways are dysregulated, to see if we can find other neuroempty targets that are contributing. And then we also want to look short versus long-term knockouts. We're making a conditional mouse to see if different signaling pathways are affected in the short and versus long-term. We also we're tossing around the idea of using a NERMD1 inhibitor to stimulate stem cell growth in the short term as a therapeutic treatment. You could see if you have something like stroke and you were to quickly give an inhibitor that would boost your stem cell, and then you could remove it and then they could differentiate and live. So we're tossing around using that for that. And then lastly, are there other types of stem cells affected? I'm really interested in hair follicle cells because mice have these early graying, this hair loss, and this deafness. And RB is essential for mammalian hair cell development and hearing. And so I think probably there's some problems with the hair follicle cells, and that would be really cool if we could use a NERMD inhibitor to stimulate hair stem cell growth. So that's another interesting project. Okay, so sorry, I think I'm right out of time. But thank you to my lab. Here's James, who did all the neural stem cell work, and John, my husband, who did the muscle stem cell work. And I'll take any questions. Thank you.
Thank you for a very interesting talk. This enzyme, NRMT1, opposes the action of N-recognin. So
I'm sorry, go ahead.
N-recognin has two sites for binding, arginine and lysine N-terminals.
Okay.
Proteins with arginine and lysine as the first residue, and proteins with phenylalanine, leucine, isoleucine, etc. bind to the other site.
Okay.
This enzyme, does it have two binding sites likewise?
It does not. It's really small. I mean, so it has one active site as far as we could tell, but it does have a variety of binding sites for activators. It binds different things that activate it and inhibit it. So it does have a variety of binding sites, but only one active site.
And binds both the proteins in the same site, irrespective of lysine is present or say.
It does. It binds. Yeah. It binds all proteins in the exact same spot, regardless of structure or sequence of the N-terminus.
That's very interesting.
Yeah, that was surprising.
And is this modification post-translational or co-translationally also? Because the N-terminal N emerges as the translation progresses.
Yeah, that's a very good. That's a very good question. We speak of it as post-translational, but the enzyme itself interacts with a lot of the ribosomal proteins, and there are targets of it. So I would not be surprised if it's also co-translational. I just have never looked at that.
I see. Thank you very much. Are there any questions from the audience?
Yeah, can I ask? Hello?
Yeah, go ahead.
It was an excellent talk, Dr. Tule.
Oh, thank you.
Yeah, I'm Dr. Lakshmi from the Department of Biochemistry again. So basically, I just was interested when hippocampal developers not happen, whether these mice are suffering from seizures anyway, because it is a size for controlling your balance, fine? And
yeah, I mean, right, you would think their hippocampus is mostly gone by 3 months old. I always am amazed that they're, those that survive they don't have seizures, which is shocking. And they can, the ones that make it past 6 months do live, long enough, but, without a hippocampus pretty much. So it's amazing to me that they're alive.
And the second question I had was, as astrocytes are changing, fine, the GFAP expression is changing. And it is known that astrocytes are controlled for a lot of neuronal synaptic connectivity modulations per se, especially in learning and memory. So anything is known in that concept, what is happening to your neuron in terms of gliotransmission and the receptor expression?
Yeah, I don't know. I mean, mechanistically, I don't know, but I can tell you that so the GFAP staining of the neurostem cells goes down, but the GFAP staining in the so that and the surrounding tissue goes up. So I take that to mean there's a lot of there's an increase in astrocytes in the surrounding tissue, and I'm unfortunately I'm not a neuroscientist, so I don't really know what to make of that yet, but GFAP staining elsewhere does increase. So we'll have to figure out what that means.
Thank you so much, ma'am. So good talk.
Oh, thank you.
Yeah.
All right. Well, thank you very much for having me. I really enjoyed.
Thank you very much for a very interesting talk, and we would like to know more about it because many of us are working in related areas.
I know it touches a lot of areas, so I usually have something for everyone.
Okay, thank you.
Thank you
for accepting our invitation.
Thank you for having me.
I now invite Professor K.N. Ganesh. Welcome, sir. Before we listen to Professor Krishna and Ganesh's talk, I would like to introduce him to the audience. Many of you know that he is the director of Indian Institute of Science, Education and Research, Tirupati. He was previously the director of Indian Institute of Science, Education and Research, Pune. He did his MSc from Bangalore University and then obtained his PhD from Delhi University. After completing PhD from Delhi in 1977, Professor Ganesh moved to Cambridge University to obtain one more PhD. He completed his second PhD within 3 years and then came back to India in 1981, joined CCMB, Hyderabad. Here I would like to add that, sir, your student Rajendra and myself were lab mates.
Oh, I see.
In 1988.
Mm-hmm.
He worked as a scientist in the National Chemical Laboratory, Pune, from 1987 to 2006, after which he held directorship at IISER Pune. He received number of scientific recognitions and awards. Shanti Swarup Bhatnagar Award for Science and Technology in Chemical Sciences in 1998, Millennium Medal of Indian Science Congress in 2000, Silver Medal of Chemical Research Society India in 2004, TWAS Prize in Chemical Sciences in 2006, and H.K. Baroda Award. These are only a few. He is a Fellow of Indian Academy of Sciences in Bangalore, the Indian National Science Academy in New Delhi, the National Academy of Sciences in Allahabad, the World Academy of Sciences, and recipient of J.C. Bose National Fellowship at the Department of Science and Technology. This is only a few of the awards he received in a long list, which I could get from the internet. I'm sure there are many more which I might not have mentioned. With this, I welcome you, sir, to give the talk. And this is L.J. Parekh Memorial Lecture. We are holding these lectures since 2019. Adayonath, a Nobel laureate, spoke in the first lecture. So I welcome you, sir, to give the lecture.
Okay. Thank you very much, Professor Ratnaprabha, for your kind words of introduction. Let me also thank MS University of Baroda for giving me this opportunity to give Professor L.J. Parekh Memorial Award. And it is really a great honor to be with the kind of speakers, what you mentioned, who have earlier given this award. Thank you very much. Let me now share my presentation. Are you able to see the screen?
It has opened now, but it is not in presentation mode.
Is it in presentation mode now?
No.
No, I don't know. You see, it's always a problem with Gmeet. You know, when we have it, I don't know what other settings are there. So what I'll do is that, since you cannot do that, you know, I have some animations which may.
Sir, you must, in case you have not shared the entire screen, you may try that, sir.
Entire screen is better, is it? Okay. Okay. Is it any better now?
Yes.
Okay.
Yes. Now.
Okay. Thank you. So I'm going to speak to something which I have a manufactured title called Spine Surgery of Peptide Nucleic Acids. Let me in the beginning confess that this is not a medical lecture, not even much of biology. I'm using surgery, what you call as a structure editing, surgery of structures. In that context, I have used the word surgery. Now, I'm going to talk to you about peptide nucleic acids, which are analogs of DNA. I don't think these introductory slides are necessary for this audience, but let me quickly get through that to give you some background about my work. All of us know about the DNA structure, which has got a sugar-phosphate backbone, and that is very, very important for today's talk. And then the two strands of nucleic acids are held through complementary base pairing between G and C and A and T. This is high school stuff, all of you know about that. And a consequence of this chemical structure of sugar-phosphate backbone, to which the nucleobases are attached at the glycosidic position, is that it forms a DNA forms a duplex. This is the lowest energy conformation when the two strands are complementary. As a consequence, you have this three-dimensional DNA double helix structure, which is very, very famous, first hypothesized by Watson and Crick. And when you see this double helical structure, you see that the two strands are wound around each other and with the base pairs in a perpendicular axis. This results two grooves, as you see, like a twisted rope, what is called as a major groove and a minor groove. Now, these are important because they are the places where proteins and small molecule drugs, they recognize the DNA. Now, we are all familiar with DNA forming duplex, but the structural complementarity, that is through hydrogen bonding between T and A and G and C, gives us to a lot more interesting opportunities. For example, if you have a AT base pairing and you have this purine adenine binding to thymidine from one side, what can also do that if there is a third strand having thymine, which is shown in the red color here, it also has complementarity, which is called the Hookstein base pairing, with a 5-membered ring of purine and the other lone hydrogen atom on the NH2. So this gives rise to a triple helix. So thymine can recognize the AT base pairing from the major groove side to form a triple helix. Similarly, if you have a GC base pairing, that can be recognized by another cytidine through the major groove to form a CGC. So DNA can form a triple helix. In the formation of triple helix, you have what's called the Hookstein hydrogen bond, that is the one between the 5-membered ring and the cytidine. You also see that for the cytidine to recognize the GC base pair, it requires protonation. Without this proton, it cannot recognize the nitrogen here. So it requires protonation. What it means is that the CGC triplex can be formed only at acidic pH. This requires protonation at 6.0, but it is present even at physiological pH. There is partial protonation possible. So these are the DNA triple helix. The third strand, what is shown in the red one here, it comes and sits in the major groove of DNA. What you see is in the black strand forming a triple helix. So DNA can form a triplex, again a consequence of complementary hydrogen bonding pairs. Now, not only that, and all of you are familiar with this slide too, DNA can form a tetraplex structure. If we have 4 guanines coming together, they can form interbase pairing like this with a combination of Hookstein and Watson-Crick bonding, and all the 4 Gs come into one plane. So if you have a stretch of poly Gs, running like this, then it can wind around itself to form these kind of tetraplex structures. And these are very, very important because in the telomere action, a lot of G tetraplexes are involved. And when the cell divides, and at the end of the chromosomes, the DNA, you require these tetraplex structures to extend so that the complete replication happens. Now, in order for this tetraplex to form, there is a requirement. You see that in the structure, there are 4 oxygen atoms inside. So all of them have negative charges, and so they repel each other. To neutralize that repulsion, you require a proton, you require a potassium K+ to neutralize that. In organic chemistry, they call it as a crown ether kind of complex. So tetraplex structures are formed in the presence of sodium plus or potassium plus, depending upon the kind of sequences. So this is the G tetraplex formation, which is now a therapeutic target also of cancer. Now, if Gs can form tetraplexes, opposite have to be a C chain, poly C. What do Cs do that? Cs can also form a tetraplex structure. It is called as the I motif. Now, earlier I told you that a protonation of cytidine is required. And here, with this protonation, you can form a C+ duplex. But the tetraplex here is not like the one which is like G4. You have 2 strands, like a duplex, CC duplex, and you have other 2 strands. And the CC base pair is intercalated or interdigitated between the 2 base pairs. So this is called as the intercalating motif or I motif. So DNA can form duplex, it can form triplex, it can form tetraplexes, and again 2 kinds of tetraplexes. One is the G4, other is the I motif. So what it means that if you put the right kind of sequences inside the cell or a test tube, they automatically assemble, is a programmed assembly to form these kinds of secondary structures. Now, very recently, not recently, almost about 20 years ago, it was recognized that one could utilize short pieces of DNA or oligonucleotides themselves as drugs. This area, which all of you have heard these terminologies, antisense technology ribozyme, DNAzyme, siRNA, microRNA, including CRISPR-Cas. In all these kind of applications, a small stretch of DNA or RNA, a synthetic DNA or RNA, can itself be a drug molecule. The way in which it acts is it goes and binds to the complementary stretch in the cell on the DNA or a target RNA. And the moment it forms these kind of duplexes, either they sterically or degradatively, they stop the gene expression. So when you have a defective protein causing a disease, you can kill the corresponding RNA, or you can stop the expression of that by these kinds of oligonucleotides. A very, very simple way. In the normal sense, if you want to design a drug for a protein, you should know the three-dimensional structure of the protein. Even then, it is not very easy to design a molecule which can bind specifically to the cavity inside a protein, because we still do not understand the three-dimensional folding of protein or the protein folding. On the other hand, taking DNA is very, very easy as a target, because all you require is complementary sequence information. Now with the human genome sequencing, knowing the full genome sequence, it's very easy to target the genes of relevant to the disease by using any of these applications. For these, a small stretch of oligonucleotide itself is a drug. Now we have to think it not like a gene, not like a genetic material, but a small drug molecule which goes and recognizes the complementary stretch of DNA. I'm sure many of you are familiar with that. Some of you might be even using in your research. Now, for this thing to happen, if you take a short stretch of DNA and put it inside a cell, there are several problems. One is the DNA, it has negative charge, right? So the lipids, the membrane lipids also have a negative charge. So DNA negatively charged molecule cannot go inside the cells. So it gets sterically repelled. The second thing is about the stability of DNA itself. There are several nucleases which immediately chop off the DNA or RNA and prevent them from reaching the target. The third, probably the cell permeability I've already talked about. The other is pharmacodynamics. If DNA is a drug molecule, it has to follow all the pharmacokinetics and pharmacodynamics principle of any other drug. So these are the challenges of using DNA or RNA as a drug. Nevertheless, this technology has already matured, and more than about 6 or 7 drugs in this series have been approved. And these have been approved to diseases which are not amenable for small molecule drug development. Now, in order to address these kinds of problems of DNA, what chemists have done is they have chemically modified DNA to overcome many of these problems. Now, if you look at the DNA, sugar, phosphate, sugar, I've just shown you a partial structure. If you look at from the chemist's eyes, what it says is that there are several places where we can modify the structure. Just like you have the structure-activity relationship in drug development. For example, you have all the nucleases, they hydrolyze the phosphate bridge. In the phosphate, you have POO minus, the natural phosphate of the one. The nucleases cut that phosphate. If you want to make them resistant to nucleases, one of the things is to convert phosphate oxygen, one of them, let us say, into sulfur. This is called as a phosphorothioate, and the nucleases cannot hydrolyze this linkage. So you can make them DNA stable to the nucleases. Or one can attach, we know that in RNA at the 2' position, there is a hydroxyl group here. You can attach other kinds of groups here, R, so that the presence of R also retards the nuclease cleavage. You can also do several other chemical modifications. C5' is one source for modification. The oxygen of sugar can be replaced by sulfur, carbon, or NH, etc. to get modified sugars. So many of these chemical modifications give some solutions to the problem of nucleus resistance, cell permeability, etc. But the drug development is far more complex. So for the nucleic acid therapeutics, by any of these approaches, antisense, siRNA, aptamers, okay, you can prepare all these modified oligonucleotides, but there are several other problems which you have to worry. For example, the sequence specificity. If they have a secondary structure, how do they fold them? Ionic, non-ionic effects, nucleotic stability, and many of them can go and bind to proteins purely because of non-specific interactions and thereby reduce the specificity. So all these things become problem, and one has to tune the structure and chemical modifications to address these things. In addition, they may also go and bind to several of these receptors. So the antisense activity is a complex one. Only in the last decade or so, one is understanding about what happens when you put a polyanionic DNA or a chemically modified DNA. What are the other kind of side effects that can trigger? In spite of that, today there are about 6, 7, 8 drugs which are available. Although they're expensive, they cannot compete with small molecule drugs. So the chemical modifications of oligonucleotides continues, the interest continues in that. Now this tells you about the kind of chemical modifications which are possible. In the very first generation of chemical modifications, phosphate oxygen was put into sulfur. So these are called the phosphorothioate. As I mentioned, they become stable to nucleases. But this still has a charge which can retard the cell entry. So one can put a methyl group. It is called the methylphosphonate. So these were the first generation antisense oligonucleotides, and the very first drug which was for an eye infection was based on these kinds of chemical structures. But these phosphorothioates, being little hydrophobic, they have a lot of non-specific effects going in binding to untargeted regions. So in the second class of modification, second generation, the phosphate modification was done away. It was kept as phosphate. But at the 2' position, several chemical groups were put, like fluorine, ethoxyethyl, methoxyethyl, methyl group, etc. Now these ones, they made them stable to the nucleases. And we also know that several drug molecules have fluorine, and fluorine has this capability to improve the cell penetration. So these were the second generation antisense oligonucleotides. Now in the third generation, people started playing around with the backbone. You know, the sugar conformation, it can be endo or exo. I will not go into the details. For binding, sugar has to be a proper conformation. So that was fixed by making this kind of a link, which is called as lock nucleose acids. In one of the cases, the most recent one, for a drug for Duchenne muscular dystrophy, it's been approved, that has the whole sugar, ribose, replaced by what is called the morpholinone group and phosphoroamides. So all these modifications involve modification of sugar, modification of backbone, etc. But sometime back, about 25 years ago, a new kind of modification came, which is much simpler than all these things. So you can modify it, but the chemical synthesis, you had to be able to do that, and you had to maintain stereochemistry. All those are problems. Then came this third generation oligonucleotide, one of them, which is called as peptide nucleic acids. This is the topic of my research. As you know, this is the simplest structure compared to all the above modifications. So I think it is good to start with a simple structure. And I can now tell you today, I will tell you how this simple structure can be modified to tune its properties for various functions. So this is called as peptide nucleic acids. And you look at the structure, the phosphate backbone, the anionic phosphate backbone is now replaced by an amine in this molecule. This backbone has ethylene diamine, NH-CH2-CH3 and ethylene diamine, to which a glycine is attached, nitrogen, carbon, oxygen, say glycine. So it is not a peptide. As most of you know, peptides are molecules which have 2 alpha amino acids linked to them. But here you have only 1 alpha amino acid in ethylene diamine linkage. So it's a polyamide linkage, aminoethyl glycine attached to that. But to this nitrogen, the base is attached. So if you want to go from here to here, this oxygen has been removed. This has been made open. This carbon here has been replaced by a nitrogen atom here. Phosphate is replaced by amine. This is not a naturally occurring molecule. It was designed by Peter Nielsen. And what is most important was that this molecule can bind to the complementary DNA exactly. Now, this molecule, as I said, it is a peptide-like structure. So peptide means it has got an N-terminus and it has got a carboxy terminus. When 2 DNA molecules bind, we know that the DNA has a 3' end and a 5' end. So when you try to recognize a nucleic acid with a polyamide, you can have this situation in which, here, the main important thing is the distance between the 2 bases in PNA, because of this backbone structure, exactly matches the distance between 2 bases in nucleic acids. So as a result, you take a DNA and make a complementary PNA, add to that, it goes and binds to DNA. Not only it binds to DNA, it binds better than the DNA itself. So this is a PNA-DNA duplex. Now, so if you have, and it can form duplex in two different ways, because it has got an N-terminus and a carboxy terminus. Carboxy terminus can bind to 5' or it can bind towards 3'. One is called as an anti-parallel duplex, other is a parallel. Do not worry about these things, but it can bind DNA in two ways, provided the sequence matches. In addition, PNA also forms a triple helix structure. So you have this peptide nucleic acids, just like DNA. It has PNA to DNA triplex formation happens. In this one, two PNA strands can bind to a DNA molecule. So it is called as a PNA to DNA triple helix. I'll be talking about a little bit of these things today. So the important thing about peptide nucleic acids, why it is important, is that it binds to a complementary DNA and RNA in a sequence-specific manner to form the duplex. And the duplex stability of PNA is higher than the natural duplex stability. And an important thing is, it is neither a peptide nor a nucleic acid. So it is stable to both proteases and nucleases. So it can use very nicely for antisense properties. It is stable to the enzymes. Only problem, a major problem, is that this does not enter into cells easily. DNA is difficult to enter because of charge repulsion. In case of peptide nucleic acid, there is a lot of carbons here. They have hydrophobic interactions, so they get stuck in the membrane. They do not enter into cells easily. So how do we improve the properties of PNA? So one gets into the kind of chemical modifications, again, of the peptide nucleic acid backbone, the backbone of the spine. That's what I'm going to talk about. This was the PNA, which bound to DNA extremely well, but it was not going inside the cells. And the way in which the chemists modified was that you see this red oval, an additional bond. Instead of ethylene diamine, they made a propylene diamine, an additional bond on this side, or an additional bond on the glycine side, or an additional bond on the tertiary amide side here was put. And in fact, the amide was reduced to amine, so that this amine is cationic. It can go inside the cells better. Or one could introduce substitutions. The basic PNA is non-chiral. It is non-chiral. But the moment you put substitutions, it becomes chiral. So various chemical modifications were tried by Nielsen. And in fact, we were the first one to introduce these kind of cationic groups without modifying the backbone, because these can go inside the cells much better. Amino groups, or you can attach a guanidinium groups, which can even take them cells inside the better. And we did show that these PNA molecules go inside the cells much better. They are taken up by the cells much better of this cationic without sacrificing the sequence specificity. But those molecules were quite difficult. So what we did was, we took the achiral PNA, what is shown here, ethylene, diamine, glycine, a stretch of DNA. And then what we did was, we tried to modify the structure. We did surgery on the spine. The various modifications we did was cyclic modifications. And I'm just showing you in a single molecule various kinds of modifications. In fact, each of this one modification is a one PhD thesis. I can tell you that. So one can put cyclic structure to make PNA look like DNA. Now we have a 5-membered ring. Pyrrolidinyl, and the way in which you conformationally constrain, you can attach a bridge between the glycine carbon and the ethylene carbon, or you can attach between the side chain and the backbone. So various kinds of modifications were done. And we also introduced the 6-membered modifications, pipecoleic acid, cyclohexanine. On one carbon, you can make all these things. What they do is that they change the conformation of PNA, and they tune their properties to bind to DNA or RNA specifically. That is also very important. So in about 10 years, all these modifications were done, and about 12 PhD students, you make the modifications, you build a sequence, you test the hybridization ability, you test the cell permeation ability. All those things were done with this chiral. But these structures were really difficult to synthesize. It takes a long effort. So we switched to molecules in which you do not have chirality and also no rings. These are very simple molecules. You can put 2 methyl groups here, jump dimethyl groups. And those who are familiar with Professor Balram's work, he has used this called as alpha isoaminobutyric acid or these things form very nice helical structure. You can put jump dimethyl group at the alpha position, or you can put jump dimethyl group at the gamma position. What these do is that they sterically bring conformational constraint without introducing chirality. So we made alpha, beta, gamma, and we also started introducing fluorine. So from 2010 onwards, my main work has been to make these kinds of peptide nucleic acids for antisense applications. Now, I already told you that we can also decorate it with cationic molecules. So these are much, much simpler to make. You can take a natural amino acid and build a PNA chain around that. You can make guanidinium. So these are not cationic, these are cationic. So we have made a number of these kinds of molecules in the last 10 years. Now today, I'm going to take you in entirely into a different regime beyond what we had done in the modifications. So this is PNA molecule. We have been looking at it. So this PNA molecule, like DNA, it has got all the bases on only one side. Now we thought of making peptide nucleic acids. Can it recognize two DNA strands simultaneously? Can I put one molecule which can go and bind to two different DNA sequences simultaneously? It is a jump from the antisense concept. How do we do that? Each PNA molecule or DNA can bind to only one sequence. Suppose we make a molecule of PNA like this, and where at the alpha position, we've already been doing a lot of modifications. Now, suppose I decorate it with another sequence of bases on each of these units. Now, if the two sequences are different, this will bind to one gene, and this should bind to another gene. So we can make PNA bifacial, two faces. So this really is spine, is like implanting something on the spine to make it look like a single molecule which can bind to two simultaneously, two DNA simultaneously. So perhaps you can address two different diseases, simultaneously. And what is important is that you can attach this additional base, the second chain of complementary bases, through different kinds of linkages. Here we have used triazole link. I think those who are familiar with what's called the click chemistry, these are very, very easy to do that. You can take an azide, you can take a base, click it. So you can even do this within cells also, or you can attach through an amide linkage. So my talk today, in the next half an hour or so, is going to talk about how to make these molecules and what wonderful properties these peptide nucleic acids, which can recognize two DNA. How do you show that it can recognize two DNA strands? So this is the important one. So this I'm calling as bimodal PNA, and this is a cartoon diagram. So on one side, which I call as the tertiary amide, this is the one natural one. The blue one is the artificial one, what we have introduced. So if you take the bimodal PNA, this GC, AC, or the triazole side, if you put a complementary DNA here, you get one DNA duplex, PNA DNA duplex. And if you put the complementary on the other side, you will get another one. So here I have two duplexes which are fused to each other. They are not two isolated duplexes. Backbone is the same. It is forming duplex from one side, is forming another duplex from the other side. Entirely new kinds of structures, both for biophysical and biochemical properties. So these things can form not duplexes, but double duplexes. Remember, I told you DNA can form duplex, triplex, tetraplexes, but now we have a double duplex formation is possible. If we can also make PNA to DNA triplex, you can make it. And also I will show you by using something else, we can have three duplexes, DNA, PNA duplex, PNA, PNA duplex, and PNA all hanging around in one molecule. Or if you can make a tetraplex, then again we can decorate with duplex. We can go to even higher order assembly, tetraduplexes of tetraplex structures. So the gist is that you take a DNA with different modifications. Again, I have shown you phosphorothioate, alkoxy, methylphosphate, these are various things. But all these modifications, they can bind to DNA only on one strand. They cannot bind to DNA strands. Someone attempted to make a DNA which can bind to two different sequences. They attached a base at this point so that this can bind to one sequence. The base on the left-hand side can bind to another sequence. This could be attached to C5, C4 prime, or even C2 prime. This is done by Paul Nielsen, but he found that the two bases, they fail to bind to the two DNA strands because you cannot maintain the distance, the conformation is different, there is a steric mismatch. As a result, this did not bind to make two DNA duplexes like the PNA what I showed. On the other hand, in fact, even the normal duplex got destabilized. Now, in contrast to that, the PNA molecules that what we are making, the bimodal PNA, if we take a PNA, what we've done is we have attached a base on this side, onto the alpha sites. We can build on the sequence, or we can attach. Now, the beauty of this thing is that the distance between two bases on one side is exactly the distance between the two bases, because they're separated by the same backbone. Like a scale, the distance is the same. So this can also bind to a DNA. This can also bind to a DNA molecule. One can put it at this position alpha, or one can even attach it to another position on the backbone gamma. So you can have alpha, you can have gamma, and these are the bimodal PNAs, because in all the cases, the distance between adjacent bases is the same. This is possible only in the case of our analogs, peptide nucleic acid analogs. That's our strength. So these are the unique attributes of PNA, which you cannot design such molecules by any other kinds of molecules. So now I've given you the rational hypothesis for that. How do we make it? This is a complex process. All of you know about the so-called solid phase synthesis of peptides. You take a polymer, and in this polymer you attach, keep on attaching at the monomer, and you can keep on building. The same principles, we can do that, but it's a very complex chemistry. We require several different kinds of monomers. One you require with azide, and the one you require with acetylene. So you can make this molecule attach, for example, cytidine. You do a coupling, and the side chain azide is available. So here you can click it with guanine. You get the first base on this side, and then again add another one, azide. You can pick a second base. So by series of peptide-like coupling and click reaction, by alternating these things, one can prepare this peptide nucleic acids having two bases. This itself is a new synthetic strategy, very involved. It requires a lot of skill to get this thing, because you don't purify it. You have to keep on building. So this is how it is made. On the other hand, so this is after making this peptide nucleic acids, have two bases, very high molecular weight. We should show that it is pure by HPLC. You do a mass spectral. You have to make sure that all the base sequence is right. So we have characterized these kinds of molecules. You see that this is a peptide nucleic acid. It has two different sequences on the two sides. So one is called the tertiary amide site. The other one, I call it as a triazole site. These terminologies I use. Triazole site or the tertiary amide site. So the molecular weights, you characterize them, etc. So instead of the triazole, we also did the reaction in which you can attach through amide linkages. So we want to show that this is a universal principle. It should not depend upon the linkage. So, but when you do this thing, you generate a chiral center here. So this chiral center can be S or R configuration. So we also prepared, because depending on the stereochemistry, the properties could be different. So we prepared one with a S configuration here and one with an R configuration to see how they change. So we prepared all these bifacial or bimodal nucleic acids. So this is a very complex one to show, how complex the synthesis. This is for making the gamma amide sequences. I will skip that. Now, after making the sequences, how do you test their hybridization ability? This is a thing which all the undergraduate DNA melting, you do that. What it says is that you have a DNA duplex. You do temperature-dependent absorbance experiment, temperature on one axis. You keep on heating. As most of you know, the UV absorbance at 260 nanometer changes. And when the DNA melts, all the hydrogen bonds are broken here, the Watson-Crick hydrogen bonding, the absorbance suddenly increases because of hyperchromicity effects. The midpoint of this transition, you call it as the melting. The melting point depends upon the sequence. DNA from different origins show different kinds of melting points, and also depends upon amount of AT. It has only 2 hydrogen bonds. GC has 3 hydrogen bonds. All you have to remember is that the higher the melting, the TM is, the higher the stability. Now, this is for a DNA duplex. Now, if you take a DNA triple helix, we showed that the third strand binds to Hooke's team. And if you do a melting experiment, what happens is that in the first step, this is a single step melting. When you have a DNA triplex, the Hooke's team, it comes off, the third strand comes off. And then in the second one, as you heat further, the Watson-Crick strand comes off. So as a result, you will see 2 melting. The first TM is for the Hooke's team strand melting, and the second one is for the Watson-Crick. The moment this third strand comes off, this duplex and this duplex are the same. And so you see the same melting here, the T2 melting. So if you see a double transition, you say it is a triplex, or if you see a single transition, it is a duplex. This is what happens in the case of DNA. But so many times these transitions will be very broad. So many results, what I show you, I will only show you this derivative spectra. It is very easy to determine Tm from the height of this, the peak, the amplitude peak, the maxima tells you what is the Tm. So the results, what I show you, I will only show you these derivative curves. Now, when you go to peptide nucleic acids, this is with the DNA, which is very well understood. If you have a PNA to DNA triplex, when you heat it, unlike two-step process, here both the PNA strands come off and you will see a single transition. So when we melt a PNA DNA triplex, you see only one single transition. You don't see two transitions. So there is a difference between PNA triplexes and DNA triplexes. Okay. So now let us get on to the, how we could study the assembly of these molecules. So I have given you a cartoon diagram of the so-called bimodal PNA. This side on the left-hand side is the one which is the tertiary amide side, and this side is the triazole side. So we're not interested in the backbone now. We are interested in the assembly is on the basis. So what we can do is that you add a complementary DNA to the tertiary amide side, you will get one duplex, and then you can add a complementary strand to the other side, you will get the second, triazole duplex. You see that. So this is a triazole duplex, this is a tertiary amide duplex. Then you can determine the melting point by UV-Tm. So you find that the tertiary amide duplex is melting at 30.2 degrees centigrade, whereas on the triazole side, it has a much higher melting. So the two duplexes are different in terms of their stability. So one is at 30, the other is 48.7. Now what you can do is you take the bimodal PNA and add both the DNA strands together. So both of them, they go and bind, you will get two duplexes. And when you do a melting, in this case, we found that there is only one single transition, 56.6. One would have expected two transitions, but we got 56.6, only one transition. That means both of them are coming off, or accidentally the TMs are same. But then this 56.6, what I'm getting is higher than the individual duplexes. So having two duplexes, they synergistically stabilize each other. That is a very important biophysical. There are not very many model systems to study that. So here you find that presence of one duplex stabilizes the other duplex, and both of them, they are melting simultaneously. So this is the first result, that bimodal PNAs form higher duplex stabilities. Then we thought that this is a termolecular reaction. We have one PNA and two DNA strands. Can we make it bimolecular? Because it's very easy in terms of stoichiometry, studying. So what we did was, we connected the two DNA strands through a loop here, TTT loop. So we have one hairpin DNA and then one BMPNA. So when you put this one, in this case, when you had this hairpin, we found that it shows two transitions, one at 34.6 and one at 65.4. And 65.4 is even higher than 56.6. So again, both of them are higher than the original one. So if you have two duplexes, both of them are getting stabilized. So the first thing is that how two duplexes attached to a single backbone, they exert a synergistic stabilizing effect. This was the first, very, very interesting, unexpected result that we got. And you can clearly see the beautiful two transition melting of that. We assume that since the tertiary amide is low melting, it corresponds to 34.6, and the triazole is high melting, it is 65.4. So this is the first result. Then what we did was, we said that let us make a triplex, real triplex. There were two duplexes. So for triplexes, as I said, you require a polypyrimidine stretch. So we took a PNA molecule in which you have a polypyrimidine on this side and a polypyrimidine on that side. When you have all this CCC at physiological pH, at neutral pH, if you add DG6, it only forms a duplex. It does not form a triplex, it only forms a duplex. And you see the duplex of this at physiological pH is 50.7 degrees centigrade. How do we know it is a duplex? You can do what is called the Jobs plot, and you get a 1-to-1 stoichiometry, 50 1-to-1 stoichiometry. To prove that it is not forming a triplex. So we know that that duplex forms. Now when you add adenine here, now adenine TAT can form triplex at physiological pH, I showed you. So if you have one DA8 DNA molecule, then two of these things are coming in together, they are forming a triplex. So we are now seeing a triplex, and the melting of this triplex was 30.3 degrees centigrade. Triplex is always a low melting. This is a Hooke's base pairing. It's low melting 30.6. But you look at this triplex. Here again, we have two Cs available on both the sides. So what you can now do is you can attach DG, and then the DG goes and recognizes the stretch. So what you are now getting is a triplex, which is now binding to duplexes. So these are extremely interesting programmed. You just take and put it, I am now getting a triplex of duplexes. How do we know that? Again, when you do this TM melting, I see two transitions. One is for the triplex, other is for the duplex. Again, this is terribly stabilizing. 30.3 has now gone to 45.8, and the duplex 73.5. It is again from 50, it has gone to 80. So when you have this kind of multiple duplexes linked by a common backbone, each one stabilizes the other one. You can think of DNA origami, where this principle will be very, very useful for that. So we could show that not only we can form double duplexes, we can form triplex of duplexes. We also wanted to see whether it is only triazole or the amide ones also. So we took the bimodal PNA with the amide linkage. We did similar kind of experiment. One duplex is 45.5 one side, another side it is at 51.7. And now when you have both of them, here we got beautifully, double transitions. You see there are very well defined transitions, clearly saying there is a duplex here, there is a duplex here. So this is the S configuration, stereochemistry, and double duplexes, 52 and 73.9. This was a great thing when we got this result. A lot of optimization had to be done. We were extremely happy. We did with the other stereochemistry, with the R configuration. Again, that also showed two duplexes, 38.7, 69.8, but the S1 is more stable than the R1, but they can form double duplexes. So we have shown the linkage is not important. You can have tertiary amide, we can have tertiary amide, you can have triazole, you can have amide linkage, they form double duplexes. So then we, again, by taking the tertiary amide one, we even made this triplex of duplexes. We could make it even in the tertiary amide ones. So again, it shows. So what we are seeing, this is a general property. It is not dependent upon the sequence or the linkage one. So that is very, very important to establish the generality of the phenomena of having this multiple assembly, multi-stranded assemblies. This is programmable. You just design the sequence, put them, you suddenly get this thing. You can look at the nano. There are lots of applications which I will tell you later on. Now, if you want to form a complex like this, you can form the complex in two ways. First, you take and add G, you can get a duplex. For example, here, first you take DA8, first you can get a triplex. And then in the second step, you add DG6, you get this complex. But you can do the reverse also. You can first add the DG, form a duplex, and then add DA, then it forms a triplex. So is it the same? Do we get by different paths? Because if we're adding all of them, some may assemble by this way, something may assemble. So here what we did was we followed each of them by circular dichroism, at each step. Here on the left-hand side for the lower path, on the right-hand side for the upper path. What happens. Now, the final, the D, CD in the D green one here and the B here, what we did was the final one, we superimposed on each of them. When you superimpose, you find that you get the same product. It does not form which pathway you use, it forms the same. So the pathway for multi-stranded assemblies does not matter, the final product is the same. Okay, now we also know that I told you that this can form I motif. Suppose you get an I motif and each of them can form a duplex from the other side, so you may get a much complex, the tetra duplexes of a tetraplex. Is it possible to do that? So we established that these molecules, the bimodal PNAs, form tetraplexes. Now here I had shown you in the cartoon diagram. For comparison, we did only with the DNA. Now this formation of this N protonated, it gives a band at 295 nanometer, and that shows a negative transition. This is called as the hallmark of I motif. It always shows a formation of structure shows a negative transition at 295 nanometer. I will not discuss now. So if you see a negative transition at 295, it is forming a I motif tetraplex. So we did with PNA, we did with this bimodal PNA that also showed a negative transition here. So our bimodal PNAs are forming, tetraplexus. And whereas at 260, they show this is an additional evidence for I-motif tetraplex formation. So if you take this bimodal PNA, form tetraplexus, which we have shown, and then if you can add a complementary strand, 1, 2, 3, 4, you get tetraduplexes of I-motif. But the problem is, this is stable only at acidic pH. Duplexes are not stable at acidic pH, so we could not realize this thing. On the other hand, what we did was, we went to, so we did not see that. Now, one can make a G tetraplex and do that and assemble these kind of things. That is under progress. Now, what happens at neutral pH? When there is no protonation, the two cytidines, they can form simple duplexes. So then we did, as a function of pH, as a function of pH, you do at the acidic pH, you get a tetraplex, and at the neutral pH, you get a duplex. So one can compare from these ones, which is more stable, tetraplex or duplexes. So we are characterized by different kinds of spectroscopy, and we find that the duplexes are slightly more stable than tetraplexes, whereas in the case of DNA, tetraplexes are more stable than duplexes. So we have a structure, property correlation. Now, since these things, cannot add proton, that requires acidic pH, what we thought was, if you add silver ion, silver can come and bind. Silver nitrate, if you do at physiological pH, add silver nitrate, Ag+, it is known that the cytidines can bind silver, this nitrogen. So you can take, two DNA molecule and put silver nitrate. It forms not through hydrogen bonding, but through silver-mediated base pairing. So the normal, TNA, this is 64.1, but the moment you put silver, because the duplex formation is going to, 80 degrees. Same thing with AgPNA also. That means the normal PNA without anything, it is also showing stabilization because it can form a silver-mediated duplexes. So what we then did was, we constructed bimodal PNAs in which one side is all C, the other side is a mixed sequence. So this side should bind silver. So we did that, and we can also do the other way. We can have silver and the cytidines on the other side. So either cytidines on the tertiary amide side or cytidines on the triazole side. Is there any difference between two duplexes? We did that. So you find that, when you add silver, they form duplexes. This bimodal PNA is forming a duplex. 68.3 is the TM. Now, each of these strands, they have additional complementary strand, complementation possible with DNA. So we take this triplex, silver-mediated duplex rather, and add a DNA, and now you get a double transition here. So one is for this duplex, 49.3. The other one is for 81.5. Now, this one is an excellent one. In the same molecule, I have a silver-mediated base pairing, and I have a hydrogen bond-mediated base pairing. So how we can change the molecular recognition mechanisms by choosing different ones. This is specific for silver. If you put mercury, it does not bind. And the binding is so strong that one can use them also at some point for other purposes. If you have a silver, then you can use this as a molecular conducting wire. A chain of silver paste one, it can conduct wire. So you can draw molecular wires using these kinds of one. So we could convincingly establish that we can form these kinds of multi-stranded complexes mediated by different kinds of hydrogen bond pairing. So this is the opposite way. You can take the tertiary amide cytidines, put base, and again, you see it also forms this kind of silver-mediated, complexes. So we established. Then what we did was, we took a much more complex one. We took a PNA, which is a chiral molecule having cytidine, etc. We put a proline substitution. We had branch, tri-branch, and these things can form, silver complexes, one complex here, another complex, they actually look something like this. And when you keep them for some time, they assemble into beautiful nanostructures, what are called the nanostars. So one can go to higher order assembly by using these kinds of, molecular recognition features. So in summary, what I have tried to show you is that we have created a new kind of peptide nucleic acid molecules which can bind DNA from both the sides, complementary sides. So it can form two kinds of duplexes, what we call as double duplexes. And this I have not spoken about that. On one side, you can have an I motif type, and the other side, you have a duplex. But this I have spoken. You can have a triplex. You can have a triplex of duplexes arranged, and we have characterized them by, in fact, I have not given you, we have done the isothermal titratory calorimetry, get the thermodynamics of this, etc. And then we can assemble complexes in such a way that, the central part is silver complex, the other side is Watson-Crick hydrogen bonding. It is known that the thymidines bind mercury. So we are now preparing a complex in which the central one is silver-mediated complex, the other ones are mercury-mediated complex. So one can change the metal affinity also. So this is just the starting of that, but all the work has been done in the last 3-4 years, and since last year we have started publishing because entirely a new concept, not very easy to get accepted, but now we have successively, just yesterday this ChemCom paper got accepted on the silver-mediated complex. And what I have not spoken to you is about the melting behavior, about the other kind of biophysical things, circular dichroism, etc. I do not have, but I hope in the short talk I've been able to demonstrate to you a new principle of organizing multi-stranded assembly using these peptide nucleic acids, which have two faces, on the same backbone, one spine, but two faces, which is medically not possible. But so now our next thing is to make this kind of G4 tetraplexes, and when you make it, you can actually start linking up number of tetraplexes together. Now these things will be useful even in biological sense because you can use them for promoter inhibition which are triggered by tetraplexes. So one can go to even higher order structures using these ones. This is the tetraplex of duplexes. And if you have, these kind of Y-shaped molecules, I already showed you, one of the 3-arm ones. So one can start assembling, all these kinds of molecules, and it will be very useful. We are now also working on a DNA origami. In a DNA origami, you take a plasmid DNA and you add a lot of staple DNAs. Each of these DNA can go and bind to only one strand, and then finally you can organize them. But instead of a staple DNA, if you add a a single PNA can bind to one strand here, the other one can recognize somewhere else, so you will get an H-shaped plasmid DNA. Again, you can systematically, in a programmed way, you can fold plasmid by using this bimodal PNA, whatever I have shown. So this is a excellent application. So you can use bimodal PNA as a Lego block for organizing higher order structure. I'll give you one potential biological application which I've been discussing with some biologists. We know that in the eukaryotic transcription regulation, you have this starter box promoter region, and there is a DNA beyond the enhancer element, about 1,000 base pairs sometimes apart. So what happens is that several enzymes, when they bind to the starter box, they bring in the bend DNA, they bring them so that the promoter region comes close to the, then RNA polymerase gets triggered on. So in a sense, all these enzyme complexes are necessary to bring the two DNAs together. But you have to get a complex like this. Or it's a complex, machinery, you require. On the other hand, we are now working on a genus PNA, which can actually bind. One sequence can bind to one side, and another sequence can bind to the other side. In this way, can we bring the whole DNA into the stage, minimizing the interference from the polymerase? DNA I've shown. So this is something, a little distant, difficult biological experiment, but that is possible. So this we have already shown below. So other potential applications say supramolecular organization in DNA origami, and you can make these fused duplexes, triplexes, and tetraplexes. Applications for in vivo inhibition, we are now attaching some elements for them to take them inside the cells, and the stabilization of hairpin RNA structures for, for example, microRNA. They're all hairpin structures. By using bimodal PNA, you can target, their very highly stabilization is very high. You can target specific microRNAs inside the cells, and we can have metal-mediated complexes which can form very defined nanostructures. I've shown you some micrographic pictures. A silver complex, the second one, without any silver, is showing a circular nanoparticle. But the moment you add silver, it forms a silver duplex. And this is, by changing the sequence, DNA, PNA1, it shows long wires. And when you have a multi-stranded structures, they show these kinds of nanostructures with some kind of a disk with a hollow shape. So one can design different nanoparticles also for this. So although it's a medical impossibility to do spine surgery and get two faces, at a molecular level, we have been able to do that by doing a spine surgery and an implant. By attaching different chemical moieties, we can actually give PNA two faces on which it can recognize DNAs from either face, leading to very, very interesting structures. So that is the end of my talk. And much of it came from, these are all the work we started after 2017, I must say. And Dhruv was the one who was in my lab as a postdoc who initiated this work. Now he is with the Alnylam, which is a company which is very well known for putting drugs in the antisense. Irena has just done on silver complexes. He has just submitted his thesis. I think I must give great credit to Manoj, who is now doing a postdoc at Carnegie Mellon. He's the one who initiated the synthesis and established all the biophysical conditions, etc., for formation and studying of these duplexes, followed by Pramod, who did the amide. He did the triazole one. Pramod did all the tertiary amide complexes, and Madan did a lot of biophysical work. And these are the group at IIT Tirupati. They were all at IISER Pune. I'm still associated with IISER Pune, and now we are continuing the work in Tirupati as well. So I had to thank them. And finally, thank you for your attention. Thank you.
Now the talk.
I'll be happy to, if there are any questions, I'll be happy to answer.
Yeah. Thank you very much, sir, for a very interesting lecture. I have one question.
Yes.
Have you checked the geometry using intercalating dyes?
No, that we cannot do that because intercalators, they're only specific for DNA duplex. DNA duplexes don't intercalate, unfortunately. Intercalators don't go to a DNA duplex for whatever reason. Hydrophobicity, distance, geometry is slightly different, DNA duplex. So we cannot use the intercalation. On the other hand, I can take a DNA duplex, intercalate it, and charge it with the PM, peptide nucleic acid. When it binds, the intercalator comes out. These kind of things are already known, so we did not do those kind of experiments.
Okay. And one more thing is this is somewhat similar to, although not identical, with peptidomimetics.
Yeah.
And they observed that the ratio of hydrophobic amino acids to charged amino acid, positively charged amino acids, plays a role in selecting bacterial cells versus human cells or mammalian cells.
Yeah.
So, is there a possibility like that with your PNA?
Yes. Yeah, we have not looked at the selectivity, cell selectivity, but we have used, lysine, the cationic we have put as the lysine side chain. Instead of glycine, we can make lysine, or we can make aspargine, etc. Those molecules, they show wonderful properties. They show, we have only done with NAH, 3T3, these kind of cells. But one work, what we have done is we have attached the lysine to GalNAc, galactosamine is specific for hepatocytes. The acyloglycoprotein they bind. So we have published that paper recently. So we took the PNA molecule and attached GalNAc, and they go specifically to hepatocytes. Say to make directing hepatocytes to is always difficult for HCV-C. Hepatocyte C, this works. So it specifically goes to hepatocyte cells. That we have demonstrated by putting GalNAc. And the PNA does much better job than DNA. So that is one medical thing that is done with the company Alnylam, who are now trying to develop that into the next step.
Thank you, sir. Are there any?
I had a little question, ma'am. Good morning, sir. I was just wondering if we have any success stories so far in clinics using PNAs.
PNA's clinic, there are no success stories, mainly because you see, PNA, one of the things is toxicity. In most of the cell systems, they don't show any toxicity. But when it goes to animal systems, it shows some toxicity. So that has, to some extent, these are done with the normal PNAs. But we are not done. So there are very, very few groups who are working on modified PNAs. So there is some interest in that to test toxicity in animal studies with the modified PNAs. Otherwise, in the cells, even tons of PNA has no toxicity at all. So because it is showing toxicity in the animal cells, the field has not progressed much. But that is with a normal PNA. As you know, toxicity is also, depends upon the structure. So when we modify it with few other groups, the toxicity may go up, come down. That has not been tested because there are not very many groups who can make enough amounts of PNA. See, DNA you can order from a company, modified DNA, everything, but not very easy for you to get PNA. So there are very few biologic groups who are working on the biological experiments of PNA. So that's the deterrent for not reaching the higher clinical. But effectively, it can be just like peptides. It can be synthesized. There is no synthesis problem, etc. I think there are some groups now that we have published few papers on the hepatocytes and new kinds of modifications. There is an interest coming in companies to look at these modified PNAs. But no success stories in the clinics yet.
Thank you.
I have a question, Professor Ratnaprabha, if time permits. Balaji here.
Yeah, go ahead.
Yeah, Professor Ganesh, I found it very, very fascinating. I have a couple of questions.
Sure.
So initially, you showed about the PNAs which are like dimethyl, means single bonds, and one with ring structures.
Yeah.
There will be some entropic differences because of conformational freedom. Has there been some correlation in terms of because you said that they are more stable than the ones with ring structure. So how is this entropic factor accounted for in the stability, sir?
Yeah, it's a good question. Now, when you look at the peptide nucleic acids, if you still remember the structure, they're linear chains, all single bonds. Unlike DNA, which has got a sugar moiety. So that sugar fixes the conformation of DNA. Whereas peptide nucleic acid is like a chameleon. It has a number of single bonds, it has multiple conformations. But when it binds to DNA, it adopts itself. It pre-organizes itself to bind to DNA. Again, if it bind to RNA, it must pre-organize itself to bind to RNA. So there is an entropic cost it has to, give. It has to bind to DNA or RNA. So what we did by conformationally constraining them into, putting a cyclohexyl thing or jump dimethyl, we pre-organize the PNA so that the modified structure is more, favorable for binding to DNA or more favorable for binding to RNA. So there is a entropic thing we already built into the molecule so that the binding becomes easy. That's the reason this pre-organized structure bind very well. In fact, the jump dimethyl, we have done the theoretical studies, etc. It is conformationally, it is more like the DNA because of the conformational constraint without chirality. So the entropic cost, the entropy required to bind to DNA and RNA, one can build it through modifications. It pre-organizes itself.
Okay. Yeah. So are there any crystal structures of this bimodal PNA with DNA duplex? Because that will be very interesting to look at and may give a lot more insights.
Yeah. No, we have been trying hard to derive. In fact, the PNA itself, there are only 3 or 4 crystal structures are available in the PNA. PNA, DNA duplex, PNA, RNA triplex. Much of the structure is derived from NMR. So we have been trying, but so far we have not been successful. One is, our amounts of PNA, what we make is just enough for spectroscopic studies, CD, UV, ITC, etc. We don't have much molecule, and I think now I have to put, students to scale up. We have just procured an automated, synthesizer with which we can scale it up. So when we have enough, that's a very interesting thing, because the structure of a fused double duplex would be extremely interesting, how a single backbone can get involved in 2 duplex structures. Yeah.
Professor Ratnaprabha, I have a couple of questions. Is there time or shall I?
So you are going to be the next speaker.
Okay.
So you can go ahead. Yeah.
Oh, okay. So, Professor Ganesh, I found that the melting in a DNA double helix and PNA, the PNA is the one that dissociates first. Is there because of the energetic barrier, or is it just that they are weaker binding than the DNA double helices in terms of stacking interaction or something?
Yeah, in terms of stacking interactions, if we compare DNA duplex, and also PNA-DNA duplex. DNA duplex, it can form A form, B form, it is there, or the left-handed Z form. But the PNA-DNA duplexes, they form what is called the P form, in which the base stacking a little bit is more, is more than 3.4 angstroms. And the helical dimensions are also slightly different. As a result, the PNA breaks much faster than the DNA.
Okay. One last question, sir. In I-motif, there was a requirement for protonation of the nitrogen.
Yeah.
Has there been some kind of a surgical studies where the ring structure is modified so that there's a permanent hydrogen bond donor there, which eliminates the need for a acidic pH?
Oh yes, see, the moment you modify chemically there, then it will not bind to, complementation will be compromised. So, see, what kind of, you cannot, the only thing you can replace H plus is by Ag plus, what we did. You cannot add any chemical group there, because any chemical group could not be hydrogen bonding apart from hydrogen. NF is unstable. So, and the geometry should match. So only we can use metal ions like silver plus, we did that.
Okay, sir. Thank you. I don't have any other question, Professor Ratna.
Thank you very much. Thank you for good question.
Thank you, sir, for very interesting and also thought-provoking lecture. There are so many questions. Thank you very much.
Good morning, all of you. Yeah, I'm here to introduce Professor P.V. Balaji here. I'm Dr. Lakshmi from the Department of Biochemistry here again. It gives me very immense pleasure to welcome to this particular symposium, sir. Dr. P.V. Balaji is a professor in Biosciences and Bioengineering Department at IIT Mumbai. He has obtained his master's degree from University of Mysore, followed by the PhD from Indian Institute of Sciences, Bangalore. His main interests include the computational biology, glycobiology, and bioinformatics. He has shown his work with several publications in high impact factor journals like Glycobiology, BBRC, and so on. So with this brief introduction, I personally and in behalf of the department welcome you, sir, to the symposium and request you to deliver your lecture on gaining insights from annotating the large number of uncharacterized proteins. Welcome, sir. Please.
Thank you very much, Dr. Lakshmi Priya. I also thank Professor Ratnaprabha for giving me an opportunity to share some of the work that we have done with this community. I will share my screen and then present my talk. Is my screen visible now?
Yes, sir. Yes.
Okay. So once again, thank you, Dr. Lakshmi Priya, as well as Dr. Ratnaprabha. This is a very interesting symposium because of the two key works that are proteins and biology. I will be presenting a part of the work that was carried out by my former PhD student Jaya Srivastava as part of her PhD thesis. So I will begin by giving background to the work and what our hypothesis was, and then I will present the results that we have. About 30 years ago, Professor Ajit Varki wrote a special invited review in glycobiology as to what is the function of carbohydrates or oligosaccharides, and he reviewed the existing literature, and he found that the kind of processes, biological processes that oligosaccharides take part are very wide and immense. Very many functions that they are part of, and it is actually a 34-page article, and he sort of documented so many studies. It was quite evident that oligosaccharides are involved in very many types of biological process, very diverse also. However, it was quite normal those days for people who work with glycoproteins to remove the N-glycan part and work only with proteins. That is because typically it was thought that if there is a glycoprotein, this is a nuisance. It is something like a stigma attached to a beautiful protein and it was considered as a ugly duckling. So people used to remove this and work only with proteins. However, this is from Shannon in Weizmann Institute. He solved this crystal structure of glycoprotein and it became a science publication simply because the structure had the glycan moiety intact. But we now know that we cannot just get rid of glycans, or we cannot pretend that they are not important, because if you look at the number of human disorders with a genetic defect in glycosylation pathway, it is increasing by the year. As of 2016, middle of 2016, there were at least about 120 different genetic disorders, each of which had at least some defect in a glycosylation pathway. Not just in humans, even in bacteria, whether it is a prokaryotic world, the glycans play an extremely important role. There is vast amount of literature is there. I have just presented one data where they contribute to variations in the serotype. So when we look at the major biological macromolecules, we find some contrast. If you look at the DNA, that is the genome, the sequence, the way the sequence in an organism, the genome of organism remains same. There can be some small changes in multicellular organism between different cells, but otherwise we say that this is a human genome, this is a E. coli genome, the sequence remains the same. And we have actually enormous information about the DNA in a relative sense. Even if you look at the transcriptome or the proteome or the metabolome like carbon cycle, nitrogen cycle, ATP cycle or signaling pathways, there is adequate amount of information about these spatiotemporal variations. Only in the last couple of years, we have been getting these papers on lipidomics where they are looking at structural diversity in the lipid component. It's important. People have known this, but there were not techniques that were available. But in the last couple of years, I have seen papers which characterize the structural diversity. But when it comes to glycome or the glycans, the spatiotemporal variations are largely unknown. I'm not telling it is not known, it is largely unknown. So on this side, which I have sort of copied from Ajit Varki's 2017 glycobiology article, we see that the amount of information we know about DNA, RNA, and protein is relatively much more than what we know about lipids and glycans. But if you look at the chemical complexity, these glycans and lipids are far more complex than DNA and RNA. And evolutionarily, we see that the conservation is much higher at protein and DNA level than compared to lipids and glycome level. Lipids, we don't have, at least I am not aware of any data on the evolutionary conservation part, but definitely glycome, we do have some data. But all of them are actually quite diverse in terms of the information they carry, the way we look at the primary structure. Now, why is it that glycans have actually fallen behind? What is the reason? The reason is that they're quite complex. The complexity arises from two reasons. One is the structural diversity that is intrinsic. The other is the absence of a template for biosynthesis. What I mean by this is that when we have a replication of DNA, we have a DNA-dependent DNA polymerase. Similarly, for RNA or protein synthesis, there is a template, and using the template, the new molecule is synthesized. But for carbohydrates, we are not aware of any such template. Although it exists, it is more complex than what is simple, as in the case of DNA, RNA, and protein, and we don't know what it is. Because of the absence of template, it is actually limiting our ability to generate large amounts for pure sample, either for structural elucidation or to derive structure-function relationship. It is somewhat similar to what Professor Ganesh was telling in terms of the very small amounts are there, so we are not able to do crystal structure work is what Professor Ganesh said. In some sense, the absence of pure sample actually limits a lot of work. In case of DNA, one can do PCR. In case of protein, one can do genetic engineering to express proteins, but here it is not possible. Okay, so if I were to sort of focus on the structural diversity, we see that the building blocks, at least the simple ones, are primarily configurational isomers, where the difference is in the configuration either at C4 or at C1 or C2, or it can be even in terms of the 6-member ring or the 5-member ring. Okay, this is one source of the structural diversity. The other source is the different ways in which two monosaccharides can be linked. There is only one way that I can link two alanines, but two galactoses I can link in 11 different ways, and whether that makes a big difference or not can be seen simply by comparing the beta-linked and alpha-linked galactose disaccharides. In fact, this is a major cause of xenotransplant rejection from pigs. About a month ago, there was a news report where it was said that a pig heart has been transplanted into a human, and this had 3 knockouts and 6 knock-ins. I have not been able to identify which genes were knocked out or knocked in, but I am sure it will be the gal alpha 1,3 gal transferase. Okay, so essentially these simple differences like alpha or beta linkage can bring about significant differences in terms of their biology. The third one is in terms of the modifications that monosaccharides have, like what is shown here is N-acetylation. There can be many other, but importantly, there is branching possible. If I look at a protein, it has only one amino terminus and one carboxy terminus, whereas a nucleic acid has a 5' end and a 3' end. But in carbohydrates, you can have a reducing end and more than one non-reducing end. So this can be even as many as 5 can be there. This branching also gives rise to complexity. The consequence of this structural diversity is that it is not easy to sequence glycans, and we need a battalion of techniques to even sequence one glycan, which means that you need actually large amounts of pure sample, and this is the reason why it is actually a low throughput technique as opposed to a high throughput techniques of DNA sequencing. But despite of that, we do have some structures, and in 2010, about 13 years ago, an analysis of the bacterial carbohydrate structures that were known at that time was made, and they simply looked at what are all the different monosaccharides that are present in the bacterial carbohydrates that were known. And we found some very interesting observations. If you look at the entire structure that are there, all the monosaccharides except this one, all are present in bacteria, not necessarily the same bacteria, but different bacteria. And we find a lot of differences. For example, enantiomers, L-xylose and D-xylose. Bacteria have L-xylose, but mammals so far, there is no evidence of having a D-xylose. Okay, this is one. There are other examples. This is one of them. If you look at the ring form, bacteria has a furanous form, but mammals don't have a furanous. They have only pyranous form. Bacteria also has a pyranous form. Anomeric configuration, for example, there is a glucuronic acid alpha in bacteria, beta in mammals. Similarly, there are other modifications are also there. So it seems that bacteria can make some monosaccharides that are not in mammals. The question is that, is this evidence of absence or absence of evidence? What I mean is that, is it that bacteria can make other monosaccharides, or even mammals can make other monosaccharides? We don't have any evidence of that, or is that they are not there? This question was not answered. This is an unanswered question. Okay. So here I have compared comprehensively the alphabets, and if you look at the English alphabet, it has 26 letters A, B, C, D, E, F, G, H. And similarly, if you look at the DNA alphabet, there are 4 letters A, C, G, T. RNA alphabet also has 4 letters A, C, G, U. Protein alphabet has 20 letters. Lipids, we cannot directly have an alphabet. I don't know how to define, but glycans, we don't know if they have a alphabet which is sort of fixed, as in the case of DNA, RNA, or protein, and what it means. Of course, there are exceptions in some cases, and in case of glycans, it is premature. Okay, so in this background, we came up with a hypothesis that the glycan alphabet is not universal, or it cannot be universal. The reasoning behind this is that DNA and RNA are information carriers, and variations is antithetical. You have to have some kind of a similarity so that across the board there is a uniformity, and because they are information carriers. Proteins are workhorses of cells, and they are conserved across this during the evolution, because there has to be a specific fold and active site architecture that is essential for function. But glycans, at least I'm restricting myself to prokaryotes, they end to distinction. I gave the example of serotype, variations, and by remaining the same, it is not going to, to serve this purpose. So it looks as if the glycan alphabet cannot be universal, at least in prokaryotes, and we wanted to find out if we can find some kind of a evidence to prove this hypothesis. Okay, because experimentally there are limitations, and we wanted to sort of exploit the large amount of completely sequenced bacterial genome to provide an answer, even if it is say bioinformatics based prediction answer, we wanted to provide an answer to that. So essentially what we did was that we searched completely sequenced bacterial genomes for the presence of enzymes that are required for the biosynthesis of various monosaccharides. I will illustrate what we did using this slide. So suppose that we consider rhamnose. I always consider a nucleated sugar because these are the donor substrate for the biosynthesis of glycan. It's always a nucleated sugar. That's why it's always a nucleated sugar. So the biosynthesis of L-rhamnose starts from glucose 1-phosphate and it's a 3-step pathway, step 1, step 2 and step 3. So we looked at the monosaccharide biosynthesis phosphates that were reported in literature. We collected all the enzyme sequences that were experimentally characterized. That's what we did. And we constructed profiles, what are called as hidden Markov model profiles, HMM profiles. Essentially, they capture the evolutionary history of each of these enzyme families. Once we have a HMM profile, we can actually scan the completely sequenced bacterial genome and then find out if these enzymes are present in a genome. So essentially, if I find a prokaryotic genome which contains homologs of all these enzymes, I infer that it is actually making a rhamnose and it uses this. This is the premise on which this work is built. Once again, I repeat that this is a bioinformatics prediction work and we avoid experimental validation, which is not going to be straightforward as I'm going to convey to you in the next few minutes. Okay. So the number of building blocks we considered were 55 and total number of pathways are 47 because some of the pathways give more than one building block. Okay. This is the total number of building blocks that we searched in the genomes of bacteria, prokaryotes. And when we look at the diversity of the building blocks or monosaccharides, we have pentoses, hexoses, heptoses, and nanoses, that is 9 carbon sugars. These are the numbers. Some of them prefer 4C1, some of them prefer 1C4. The D enantiomers as well as L enantiomers are there. Some are known to be in pyranose form, some are known to be in furanous form, and the nucleotide can be ADP, CDP, GDP. It's a variety of pathways. So overall, there is a lot of variations in the nucleated sugars that we have considered. And also, if you look at the modification, some are simple monosaccharides, but if you look at the modification, there is a amino modification, formyl, acetyl, diacetyl, and then acetyl at C3, C4, C5 position. There's a variety of acetylation are there. There is a 6-deoxy, 3,6-dideoxy is there, trimethyl is there. So a variety of modifications are also there in the set that we have considered to search the prokaryotic genome. At the time this work was carried out, there were 13,000 prokaryotic genomes that are completely sequenced. Now it is about 30,000, but at the time Jaya did this was about 13,000. And this sort of shows the number of strains for each of these organisms. There were most of them only one strain was there, but some of them there were many strains that were sequenced, and I will tell you what is the importance of this strain-specific data. Okay. So I will briefly present the results that we got. Okay, so one thing we found was that in each of these, at the species level, how many building blocks can they biosynthesize? That means that the genome has pathways for the biosynthesis of how many building blocks, and the species, they are ordered in some arbitrary or random manner. Sort of we simply arrange them in the alphabetical order of genus species, and there are a total of 3,368 species are there. We put them here, and for each of the species we simply put how many building blocks that they can synthesize, the maximum number we took. Very obviously you see that there is so much of variation, and most importantly there are some species which do not make even a single monosaccharide. So there are 163 species which do not make any carbohydrate or any monosaccharide. They don't have a biosynthetic pathway. 78 of them can make only one building block, and so on and so forth. It's a very interesting observation that these genomes do not have the pathway to synthesize the building blocks. Now, does it make sense, or is there something wrong in the data that we have? So when we analyze the organisms which don't make even a single building block, we found some interesting things. So see, mycoplasma, spiroplasma, anaplasma are some of them, and they're all human pathogens or plant pathogens, and we see that these organisms have arisen as a consequence of reductive evolution. Reductive here refers to the size of the genome, and they have considerably, reduced their genetic material. They are able to do that because they are, obligate, parasites. They depend upon the host for all the nutrients. So they don't biosynthesize what they want, but they simply, take these necessary metabolites from the host, and carbohydrates are one such that they take. In fact, the molecules don't even have a cell wall because they are always inside a cell. They don't need a cell wall to protect themselves. There's another example that we found from Buchnera afidicola. We had the sequence, complete genome sequence for 25 strains of them, and it can actually, the 13 strains of this Buchnera afidicola can biosynthesize the glucanac, N-acetyl-glucosamine, starting from fructofuranose 6-phosphate. This is a 4-step pathway, and we found that all the 4 enzymes, the homologs, are present in 13 strains, and in 5 strains, none of the 4 enzymes are there. In 7 strains, either one or more of the enzymes are missing. It appears that we are looking at a process of the reductive evolution. There's a gradual loss of genetic material where some of them have completely lost, some of them have actually retained, and some of them are in the process of, sort of living out. And once again, without surprise, it is a endosymbiont. So it doesn't necessarily, live in a harsh environment like some of the prokaryotes, live, because of which it can afford to lose the biosynthetic pathways for monosaccharide. What we have done here is that we have compared the strain-specific variation. This also is a very, interesting observation that we found. What we did was that for a given species, if we have the genome sequence for more than one strain, we asked how much difference do we see in the number of building blocks they synthesize. Okay, so the blue line is for the minimum number, and the red line or the brown line is for the maximum number. Obviously, we see that there is so much of strain-specific variation. This is something that we did not expect, because as I mentioned earlier, when we look at protein and nucleic acid, the building blocks or the alphabet remains same irrespective of the strain specificity. But in carbohydrates, we see that there is an enormous difference in the number of monosaccharide that the strains use. Okay, and there are so many such species are there where they show a strain-specific variation. This is something that we did not expect at all. Specifically, if you look at the strain-specific differences, what I have shown here is for some of the species, the names I have shown here, and this number here tells me how many genomes, strains of, how many genomes are sequenced. For example, if I look at Acinetobacter baumannii, 123 strains have been completely sequenced. If I look at Campylobacter jejuni, 157 strains have been sequenced. E. coli at that time, 714 strains were sequenced. Now, if I look at the different strains of the same species, what is the minimum number of building blocks that is given here? 3, 12, 5, 8, and 3. So what is the difference in the minimum and maximum number? That is what I have shown here. So if you look at the Aeromonas hydrophila, some strains have only 5 building blocks, whereas some of them have 25 building blocks, 20 plus 5. So that's a huge variation. If I come to left side, Rickettsia, Brucella abortus, Neisseria, I hardly see any difference. Even though there are 60 strains of Rickettsia are there, 19 strains of Brucella are there, all of them have the same number of building blocks, 12 or 3. So one can actually relate these variations to the habitat of the organisms. See, if you look at E. coli, they are both free living and host associated. So accordingly, they have actually various number of building blocks that synthesize. Okay, so once again, unlike proteins and nucleic acid, we see that the alphabet is not conserved. The size of the alphabet is not conserved in organism. Even at a species level, we see that there are a lot of variations in the strain. Okay, so we actually checked if it is in any way related to the proteome size, the number of proteins that the organism encode, and obviously as the data you can see here, there is no correlation. This is the proteome size and the number of glycan building blocks that are there, 23, 22, 21. Smallest proteome and largest proteome, we don't see any difference. So the reason why some organisms have more building blocks or less building blocks is related to their functional requirement rather than the proteome size. So we looked at all the 55 building blocks and we asked how prevalent they are. Based on that prevalence, we found that there are about these 3, 6, and then 7 plus 12 of them are very common. That means they're present in more than 50% of the genomes that we sequenced, that we analyzed, and this is like a pictorial depiction of the ones which are more common and the ones which are less common. So one can see that the N-acetyl glucosamine and galactosamine are the more common, rhamnose, and then the galacturonic acid, that is one, and N-acetyl galacturonic acid relative to each other. This is relative to each other among these 12 are present in fewer genomes. Overall, we looked that there are some which are very prevalent, more than 50%, but a very large number of monosaccharides are found in less than 10% of the genomes that we scanned. So we see that there are so many building blocks there, but they are not very prevalent. They are found in only some organisms, but not all of them. But there are fewer there that are neither common nor rare. They are less common. So this is also something that we were not expecting. We see that a very large number of sugars are there, but they are present in a restricted number of organisms. Once again, I'm talking about only the prokaryotes. I have not looked at the data for eukaryotes. And there is a reason for that also. Okay, so this is a pictorial depiction of the phylum-specific differences. The different phyla are along this axis. Along the x-axis from left to right are the different building blocks are there, and this y-axis tells in how many organisms belong to each of these phyla. One can find the biosynthetic pathway, and you can see actually a significant differences. This is something that once again quite interesting. In this particular data, we have not included the common group because they are very prevalent. If we include this data, all this data becomes sort of invisible because of the issues related to this scale. So we have sort of left them out. And it's a very, very interesting observation that we find that there are so much of phylum-specific difference in the number of monosaccharides and type of monosaccharides that organisms use. We also did some a comparison on the types of enzymes that are required for biosynthesis. And if we look at the prevalence, okay, so this triangle is pictorially depicting the prevalence of monosaccharide. These are the 12 monosaccharides which are very common, present in more than 50% of the genomes. This represents the rare sugars which are present in less than 10% of the genomes that we sequenced. If you look at how many such are there, the rares we have close to about 45 or so. The common ones are small in number, about only 12 are there. What type of enzymes are required? Is only few types of enzymes are required, but for this we actually need many more types are there. We have actually analyzed this enzymes, and there in some of the cases we have also looked at the evolution of this, but in today's presentation I will not discuss this data because I don't have that much time. Okay, but we have done that. I'll just give you one or two examples of the observations that we found. Rickettsia and Orientia, these are two types of pathogenic organism. This causes Krebs typhus, this causes Rickettsia, and we see that they have actually taken different evolutionary paths. Both of them actually the starting material here is UDP glucanac. From UDP glucanac either they make N-acetyl manosamine or N-acetyl quinosamine. This is L sugar, these two are D sugar. Quinose is nothing but 6-deoxyglucose. Okay, and we find that in this Orientia, the extracellular polysaccharides are absent, whereas in Rickettsia, the extracellular polysaccharides are present. And this is a experimental data. We find that Orientia does not biosynthesize any glycan building block. It simply uses the host machinery to derive whatever it wants. When we look at the pathway for the biosynthesis of manosamine and quinosamine, Rickettsia has the pathway from UDP glucosamine to this part of the pathway it has, but it has lost this pathway. Why is that so? Because the host can make this, but the host doesn't use this monosaccharide, so this biosynthesis pathway is not in host. So Rickettsia very cleverly has kept only those enzymes which are required to make manosamine and quinosamine, but these enzymes it has conveniently lost it. But you see that they have similar kind of host preference, pathogenicity, and the symptoms they cause, but the vectors they use are quite different. So it is too early for us, or we don't have data to sort of further, investigate the reasons for this, but we find that as a starting point, this is very interesting. One last observation that we wanted to, that I'm going to share with you is that, okay, so some of these organisms which are also have pathogenic strain, we ask the question, is there the difference in the glycan alphabet between the pathogenic strain and the non-pathogenic strain? And we find that there is no such difference in the alphabet or the type of sugars they use. We are showing the data for Staphylococcus aureus here, and what percentage of the 378 genomes have this. We see that same type of monosaccharide is present in all the strains, whether they are pathogenic or not pathogenic. A similar observation we see even for Mycobacterium tuberculosis and even in the case of Brucella and Neisseria. So clearly we see that the pathogenicity is not because of the alphabet size, but obviously it has to be due to the structural diversity starting from same alphabets, but the linkage type branching pattern repeatedly that requires further investigation. So the key observation that we have, what I have shared with you, is that there is no universal glycan alphabet, and even among strains there is significant variation in the glycan alphabet. That means the letters of the glycan that the organism uses, and strain specific differences are minimal in organisms with localized habitat. Size of the glycan alphabet is independent of proteome size. A few building blocks are used extensively, what we call as the common group. And most of the building blocks are used by very few organisms, a rare group. And we have actually, sort of speculated on the advantages of using rare groups, but I'm not sort of sharing that data with you. So what next? I will just look at only a couple of possibilities. There are others also. One is that what we have done is that we have started with experimental data and we have tried to make a prediction about the molecular function, but especially for the proteins for which we don't have function. Now, if we have more experimental data, the prediction accuracy can be improved. But whatever predictions are there, these are the predictions that facilitate experimental characterization. So there is some kind of a synergy that we see, and we have to wait for more experimental data to come so that our prediction accuracy can actually improve. This is one of the things that we foresee. But we are, as I mentioned, we have also looked at the evolution of some of the enzyme types, but I'm not presenting that here because of the limited time that I have. And this is all that I have to share with you. And I thank you all for sort of listening to my presentation. And once again, thanks to Professor Ratnaprabha for giving me an opportunity to share our data.
Thank you, sir.
Thank you.
If the audience have any questions. I want to know one thing, sir. You told about the blocks, building blocks.
Yeah.
But how the blocks are connected, building blocks are connected to each other is also done by enzymes.
Correct.
And that is where the diversity arises. If I have.
That is one of the reasons, sources. Yeah. Yes. Yeah.
So, do you see any similarity in sequence between those enzymes which are working at a later stage from different organisms? Because how do they recognize whether after the first building block, first glucose, they have to come and act and add N-acetyl glucosamine or something like that, or there is something else more than that, first or second or third? How would they decide?
Okay, so that is basically the question you're asking is about the what determines the sequence of the glycan. As I mentioned, since there is no template, we don't know, we don't have answer that is say, nature's secret as of now, but we do know that such a code does exist because every time you grow the organism, it gives the same sequence. So it is not a random process. There indeed is a template that it is completely unknown to us as of today.
And have you seen any homology between these enzymes of different organisms?
Yeah, so the enzymes that you're talking about are the glycosyl transferases. they are very tough to work on. We have actually made several attempts. The reason why they are tough is that they have highly conserved fold, about 4 or 5 different folds are there. But in terms of the diversity of linkage they generate is enormous. We don't have very much data for the glycosyl transferases, so we're actually constrained by the absence of or paucity of experimental data in that direction.
Thank you very much.
Thank you.
One question. Every time on holiday like this, today is a holiday here, I think.
Yeah, here also, and I think Professor Naresh Kumar has a question.
Yeah.
I'll just continue with this. Are these enzymes are independent enzymes or part of a multi-enzyme complex or something like that?
There is no evidence of this being a multi-enzyme complex, but it depends on where the glycan is synthesized. If this is extracellular glycans, there is some kind of assembly. So part of the synthesis takes place inside the cell. Then there is a flippase which flips the partially synthesized glycan to the outside of the cell, and then the others are added later. But if it's an intracellular glycoprotein, even few such examples are there, even in prokaryotes, then they are not membrane associated, they are independent.
No, I was trying to see whether the sequence is related to the organization of the multi-enzyme complex or the arrangement of the enzymes.
We don't have much data to answer that. It can be anybody's guess, Dr. Naresh Kumar.
Oh, thank you. Thank you.
Yeah.
I would like to know if there are any more questions. Yeah, now I thank you,
Thanks,
Dr. Balaji, for accepting our invitation and giving an interesting talk on a holiday like Mahashivratri.
Thank you very much.
Thank you very much. I request Devesh to introduce the next speaker.
Yeah, it gives me pleasure to. Am I audible?
You have to speak a bit louder.
Okay. It gives me pleasure to introduce Dr. Rajan Sankarnarayan. Dr. Rajan is an eminent scientist and group leader, Structural Biology Laboratory, CCMB, Hyderabad. He did his bachelor's and master's in physics from Madurai Kamaraj University, and his PhD was from Molecular Biophysics Unit, IISc Bangalore. He later moved to France, where he did his postdoctoral work at Institute of Genetics Molecular Biology. And after returning to India, he has contributed a lot in the area of proofreading during protein synthesis. He has received several awards and honors to his credit. To name a few, prestigious Wellcome Trust International Senior Research Fellowship, Swan Jayanti Fellowship, National Bioscience Award, Shanti Swarup Bhatnagar Award, J.C. Bose National Fellowship, G.N. Ramchandran Gold Medal, and recently he has been awarded Infosys Science Prize in 2020. He has been actively involved in editing in journals, so he is a editorial board member of several journals, associate editor as well as co-editor in many journals. Few journal of repute includes the Journal of Structural Biology as well as eLife, and he is also a fellow of all the 3 major National Science Academy of India. With this brief introduction, I welcome you, sir. Over to you, sir. Dr. Rajan.
Thank you for the kind invitation and the introduction. Can you hear me well? Anyone can confirm? That will be great.
Yes, sir.
Okay, great. So, first of all, let me share the screen.
You can see the buttons at the bottom.
Yeah,
yeah, mic and then camera, then CC, that is for captions, and then you have
No, I see that. I see that. I see that. No, I see that. I have done that. And then share the entire screen is what I should do, is it?
No, you can share only one window also. That is also possible. If you have already put your slides on full screen mode, you can just select that particular window.
Okay. Are you able to see my screen now?
Yes, yes. Now we can see you, sir.
Okay, great. Thank you so much. Yeah. First, let me start with a small disclaimer. We are working today, so we have no holiday. So, what I will be talking to you today is about protein biosynthesis, as the title suggests. And also, when you are actually making proteins at a phenomenal speed and number of proteins, correct, in the cell, nature makes quite a few mistakes. So originally, people thought that these mistakes are kind of errors which have to be minimized, but now people have understanding that these errors are actually useful under different evolutionary scenarios, and also are having certain implications during stress conditions. So this is the direction that we are going in this field. So I will highlight to you some of the basic aspects of the problem that we are working at, and what are their evolutionary implications as we are exploring it more and more as of today. So this is something which all of you are familiar with, which is nothing but the central dogma in biology, which is talking about the flow of genetic information, which starts from DNA, which is transcribed to RNA, and then translated to proteins. So this is something which is well known. What has come to limelight over a period of time, particularly over the last 10, 15 years, is that each of these errors are tightly regulated. Of course, we knew that each of these processes are error prone, and we knew the errors associated with each one of these processes. However, the errors are also controlled, which means the systems have the ability to relax the error as and when it is required, as well as to tighten the errors depending on the environmental scenarios. So this kind of a regulation is what is making life quite interesting, and that is an aspect which people are looking at more and more. So of course, the importance of proofreading is something all of us know in real life, as well as people who work in the area of press, for example, printing press or anything wherein informations are transferred. It's so essential to make sure that we make limited mistakes, otherwise it can lead to awkward situations, as can be seen from this slide. So minimizing errors is something which has to be one of the hallmarks of biological systems when they replicate genetic information or transfer genetic information. So now if you look at these 20 amino acids which are normally used in protein synthesis, as all of you know, there are 22 genetically encoded amino acids, but the 20 are the most common ones. Many amino acid systems, like say for example, an acidic amino acid like glutamic acid. Are you able to see my cursor, by the way?
Yes, sir. It is visible.
Okay. So something like glutamic acid, and you look at another amino acid like phenylalanine, for example, they are chemically as well as structurally quite different in terms of their side chains. So the enzymes that choose them to actually charge or activate them on top of transfer RNAs, which eventually take them to the ribosome for protein synthesis, are quite specific, which means they don't make mistakes which are too high, and therefore the enzymes on their own can solve the problem. However, there are amino acids like isoleucine and valine, for example, or threonine and serine as shown here. These are very, very similar, as you can see from their side chain. Their chemistry as well as the size are not very different. Therefore, these enzymes make good amount of mistakes and more than what is required for faithful translation of the genetic code. And this problem was pointed out by none other than Linus Pauling way back in the 50s. And what he told, even without having seen protein structures and how enzymes actually operate in terms of recognition, he said that some of these amino acids which are used for protein synthesis are so similar, therefore they can't be selecting on a chemical basis these amino acids much more than what is required in terms of the accuracy. And there are quite a few amino acids which have these problems. And therefore, what we now know, correct, after several decades of work in this area of looking at amino acid tRNA synthetases, which are enzymes responsible for taking an amino acid and putting on top of tRNA. What we now know is that these enzymes are quite accurate, and they also have what are called proofreading functionalities. Why this proofreading is very required is because if you put a wrong amino acid on top of the tRNA, the amino acid is shown by star. Say, for example, instead of the correct one, if you put what are called non-cognate amino acids on top of the tRNA, now at the ribosomal level, what is checked is only the anticodon of the tRNA and the codon of the mRNA pairing. Okay, nothing is checked at the level of what is the amino acid that is coming on top of the tRNA. Therefore, any mistake which is happening at the level of charging are carried forward. And therefore, this kind of property of the ribosome is used by many synthetic biologists over the last 10 years or more to put artificial amino acids or unnatural amino acids on top of tRNA. And therefore, these can be used for defining a particular property on a protein sequence so that you can use it for fluorescence purpose or tracking in the cell and so on and so forth. So those kind of synthetic biology approaches use the porous nature of the ribosome to allow the amino acid that is sitting on top of the tRNA to go through without any further cross-checking. So in addition to non-cognate amino acid, you also have, for example, the opposite enantiomers, which is what will be the focus of my talk today, which are, when I say amino acids, so far whatever I was referring to, you must have assumed that protein synthesis, when I refer amino acid, we are talking about mainly the L amino acids, which are of one chiral form. However, over the last 10 to 20 years, there's increased realization that you have significant amount of D amino acids present in all biological systems. Starting from bacteria to all the way to humans, you have this D amino acids, and many of these D amino acids are also functionally used by the organisms. Okay, so the problem for proofreading is twofold. One is at the level of non-cognate amino acid, and the other one is to take care of the opposite enantiomer, which will be the focus of my talk. And what happens in terms of proofreading is that once you have the amino acid charge, particularly here I'm talking about non-cognate L amino acid, say for example L valine instead of L isoleucine, say. Now the CCA end of the tRNA after getting activated would flip to what are called these proofreading domains. So several synthetases have additional modules which can take care of these errors, wherein the smaller amino acid or the non-cognate amino acid would be bound, and then they will be removed from the tRNA, thereby increasing the efficiency of the process. And people, by looking at multiple genomes, when they analyze, they realize that it's not only the editing domains are covalently linked as the part of a protein sequence, you also can have freestanding proofreading modules. What I mean by that is that you can have a small domain which functions on its own. So after a tRNA synthetase does its job, these small domains can actually go and repair the mistake that is done by the synthetase. The importance of proofreading came to the fore with this path-breaking work by Professor Paul Schimmel and co-workers way back in 2006. And for the first time, they showed that if you have a tRNA synthetase, which does not completely compromise the editing activity because of a mutation, but even a marginal reduction of its enzymatic activity can result in totally misfolded proteins, leading to cell death and resulting in cerebellar Purkinje cell loss in a mice and leading to ataxia. So basically, that the cellular systems cannot tolerate errors beyond a certain point was shown primarily in this work, which was followed by several other works later on. So, so that was just to tell you the importance of proofreading. Now I come to protein biosynthesis, which is the aspect that I will be talking about. And as all of us know, the protein biosynthesis majorly, not majorly, almost exclusively uses L-amino acid, except for the case of glycine. It's an achiral amino acid. However, the mirror symmetric counterparts, which are the D-amino acids, are removed or not used for protein synthesis, despite their presence in the cellular system, which is a point that I would be emphasizing. Now, if you want to make a protein with purely D-amino acid, you can in principle make it, correct? You can chemically synthesize it. And this is the Ramachandran map. The Ramachandran map would be a centrally inverted Ramachandran map of the L-amino acid ones, which are shown here, which is with the red dots. So one can actually make D-amino acid proteins, which would be a mirror image of the L-amino acid counterparts. And this was beautifully shown way back in the 90s by Kent and co-workers, which appeared at the cover of Science, as if you make a chemically synthesized HIV protease using purely D-amino acid, that HIV protease would would be the mirror image of the L-amino acid protease. And not only that, now this protease would start cleaving the D-amino acid peptides instead of the L-amino acid peptide. So not only the protein becomes the mirror image, but also its substrate becomes the mirror image. So which kind of, this picture suggests that our image shows that you can have a mirror image world in principle by making D-amino acid based proteins. So, however, the problem comes in a given polypeptide where green here is denoted, as L-amino acids, and the red shown here kind of, represents L-amino. So the green shows L-amino acids and the red shows, say, D-amino acids. While you can make complete, green or complete red polypeptides, as I showed you before, but you cannot have substitutions of L-amino acids in a growing polypeptide chain of D-amino acids or vice versa. And that is something which can lead to, problems in protein folding, as shown in the case of the Ramachandran map, and therefore you cannot have folded proteins. So it will lead to misfolding, and therefore D-amino acid should be excluded from protein synthesis. Now, that brings us to what are all the factors which are available in the cell to take care of it. And this aspect of this chirality-based proofreading, or chiral checkpoints as we call them, are kind of missing in terms of the treatment, because people assumed over long periods of time that proteins are made of only L-amino acids and therefore we need not worry about them. So what we kind of showed over several years is a summary of the different chiral checkpoints. The cellular pool will have both L and D amino acids. That has been shown in many, many studies. So the first and foremost point is that of the tRNA synthetase, where majority of the tRNA synthetase are quite specific for the L amino acids. However, there are some which can activate D amino acids as shown here, and these error rates are too high for protein synthesis. So they produce some D amino acid tRNA, and once L and D amino acid tRNAs are produced, the next step is that of elongation factors, which I'm sure all of you know, that they are responsible for capturing these amino acid tRNAs and take them to the ribosome. So what people have shown over the years is that even these elongation factors have higher specificity for L-amino acid tRNA, but they are not exclusive for L-amino acid tRNA. They can also bind to D-amino acid tRNA with a reduced affinity. And then the last step is that of the ribosome. Once these are taken, the amino acid tRNAs are taken to the ribosome also is quite selective, not exclusive, quite selective for L-amino acid tRNA. What people have shown using the crystal structures of ribosomes over a period of last 20 years or so, that the peptidyl transferase center of the ribosome is quite specific for L-amino acid peptide coupling compared to D-amino acid peptide coupling. And therefore, each step of protein synthesis ensures that the D-amino acid tRNAs are rejected, even though they are not exclusive for L-amino acid tRNA. Once in a while, they make mistakes. So therefore, you produce significant amount of D-amino acid tRNAs in the cell, and these need to be recycled. So what happens is that if you leave these D-amino acid tRNA, then they start competing with L-amino acid tRNA for protein synthesis, and therefore you can have global misfolding. The other problem is that you have now significant amount of tRNAs which are amino acylated with D-amino acids, and therefore they deplete the tRNA pool. Therefore, there is no free tRNA for recycling because these are dead-end products. So there has to be an enzyme which should do this job, and this was identified also way back in 1960s by Paul Berg and co-workers, where they showed that there is a protein called the DTD, which would specifically look for D-amino acid. And the importance of this enzyme, which we were interested or excited about, is that there is only one D-amino acid tRNA deacylase, which takes care of all the 19 D-amino acids. And also, it does not have any reference to the tRNA, which means the tRNA sequence can be of any kind, which can be tRNA isoleucine or tRNA asparagine, and so on and so forth. More importantly, the side chain of the amino acid can also be variant. For example, you can have a positively charged amino acid like arginine, or a negatively charged amino acid like aspartic acid. So what we were interested in as a structural biology laboratory was to understand the mechanism by which this enzyme operates in the cellular milieu. The other problem the DTD faces is that it has to now operate in a cellular scenario wherein you have real excess of L-amino acid tRNAs, where the errors are really far and few, which means it should not cross-react with the L-amino acid tRNA pool and deplete the pool that is required for protein synthesis. So the enzyme has to operate with a very high specificity on almost all D-amino acids and almost on all tRNAs. So that was the mechanistic challenge that we wanted to figure out. And to summarize this part, this DTD basically biochemically has been shown to recycle the tRNA and tRNA. So therefore, you have now new tRNA available for protein synthesis. So we wanted to find out how this enzyme operates, and several years back, we solved the structure of this enzyme to understand the mechanism. As you can see, the molecule is a dimer with two different monomers shown in different colors. The active site is shown here with a D-amino acid tRNA alpha. You can see that there is an adenine binding pocket, there is a ribose binding pocket, and now we will zoom it on the active site. As you can see here, the first and foremost the structure gave was that the side chain was kept outside the binding pocket, and this, the reason for keeping this outside the binding pocket is the following: that it can act on any side chain. And we model based on the structure the three theoretical possibilities by which you can model an L-amino acid, and we showed that there is a glycis-pro motif, which is shown in the slide here. I'll just repeat this movie one more time. As you can see here, this is the dimeric molecule, and we are focusing on the active site. So there is a monomer green from which a glycis-pro motif is inserted into the active site. As we zoom, it will become obvious because that is the crux of the mechanism by which this enzyme operates. So this glysis probe is responsible for selecting the chiral center in such a way that no L-amino acid can sit in this pocket in all the three theoretical possibilities. So to summarize a series of stories that we kind of published from the lab over the last several years, this is what we see as an overall picture. If you take L-amino acid tRNAs, the DTD does not act. Even if you increase the enzyme to 10,000-fold or more concentration in the tube compared to acting on D-amino acid tRNA, they do not touch the L-amino acid tRNA. The D-amino acid tRNA, obviously, with or without the elongation factors, are cleaved very efficiently by this enzyme, as the name suggests, as D-amino acid tRNA deacylase. What we found very interestingly was this enzyme can also act on glycine. The fact that it is acting on glycine suggested that this enzyme works on a L-amino acid rejection mode of function rather than a D-amino acid capture, because that is the mechanistic mode of function of this enzyme, that anything but L will bind to the pocket. Now, if this enzyme binds glycine attached to tRNA glycine, what we showed is that the discriminator, the discriminator base of the tRNA as well as the elongation factor prevents the glycine to be depleted. As you know, the glytRNA is a substrate for protein biosynthesis. Therefore, this enzyme should not be removed. So we found certain unique factors in the tRNA of the tRNA glycine, which ensures that this enzyme does not act on glycine attached to tRNA glycine. However, the function of action of glycine on tRNA glycine by DTD is basically a requirement which is for removing glycine attached to tRNA alanine. That is a mistake made by another tRNA synthase, which I will not go into. So basically, the enzyme's mode of action is L rejection mode, and therefore it removes D-amino acid tRNA, and it also has another cellular function of removing glycine on tRNA alanine. Now I will take you through the evolutionary implications of this work that we started looking at nearly 5-6 years back. As you can see from this diagram, that the DTD function is conserved across the board, which means it is universally present. However, the job of this is done by two different variants. So if you look at all bacteria, you have this enzyme called DTD1. This is the enzyme which I was referring to earlier, which is called DTD1. Because people first studied that enzyme, so that's called DTD1. However, in archaea, little later, people noted that there is another enzyme which does the same biochemical function, but evolutionarily it is not related to DTD1 in the sense that it has a totally different origin. Its protein sequence, the amino acid sequence, as well as the fold of the protein is very different than the bacterial, DTD1. Therefore, they named it as DTD2. The eukaryotic scenario becomes interesting because, as all of you know, the eukaryotes arose by an endosymbiotic event of basically a bacteria being engulfed by an archaea. So this is something which is a well-accepted model for eukaryotic evolution. So therefore, if you now look at this combination, it became quite interesting. All the ophisthochons, what ophisthochons are? Nothing but yeast and animals. They have only DTD1, and the plants have DTD1, which is of bacterial origin, as well as DTD2 of archaeal origin. So which make plants very special. I will come to it a little later. But before that, in ophisthochons, particularly in the Animalia branch, you had another DTD, which was originally annotated as DTD2. And of course, we, after studying its function, we changed it to ATD for Animalia-specific tRNA deacylase rather than DTD2, because it has a totally different function, which I will be describing in the next few minutes. So, to understand this role of ATD, we solved its structure at a very high resolution. And we found that the glycispro motif, which is shown here. So, mind you, this cis is shown here. It is gly attached to a proline through a cis-peptide bond. Normally, in proteins, you always have a transpeptide, right? Most of the time. However, this is a cis-peptide bond formation, which is very unique. And that glycispro is shifted to a glytranspro in ATDs. So that was the most interesting thing. While the rest of the protein, as you can see here in this superimposition, where they are almost garments, these are very low, suggesting that except for the glycispros switch to a glytranspro motif, the protein was more or less identical. Now, as I already told you, this glycispro is responsible for capturing the center. The fact that it had moved now from this position to a transpro motif by creating some space, we hypothesized that this protein now can act on some L amino acids because the rigidity with which it was capturing the D amino acid would not be the same, and therefore we assumed that this protein would do some L amino acid deacylation. And to cut a long story short, what we showed is the following. You know, when you go from bacteria or archaea, which are unicellular organisms, to multicellular organisms, like say, for example, yeast and higher up, the genome expands significantly. Because of the genome expansion, the number of tRNAs also expand. So instead of having, say, a few tens of tRNAs in bacteria and even in yeast, for example, when you come to animals, you have few hundreds of tRNAs in certain cases, and some cases few thousands. Now, the problem for synthetases is that people have been looking at for the last 60 years or so mistakes that are made by synthetases of taking a wrong amino acid on top of the correct tRNA. Because the tRNA is a much larger molecule, therefore the enzymes responsible for selecting the tRNAs are much more accurate as can be expected. So people are looking at only this. However, when it comes to the animal systems or higher systems in which the number of tRNAs are what we call them as tRNA isodecoders, when the isodecoders expand in the context of animals, now the synthetases have a different problem. They pick up the amino acid moiety quite accurately, but they actually start making mistakes with the tRNA. What it results in is by putting the correct amino acid on top of the wrong tRNA. And this was the case of an alanine, which is shown here, is charged on a tRNA threonine by a mistake. And we showed that this protein ATD, which appeared in the context of animalia, in the animalia branch of life, is responsible for clearing this error, leading to minimal mistranslation. And this is what we showed a few years back. Now, the proof of the pudding is in eating. So we wanted to show, okay, now the ATD is having a biochemical role, which we showed in the test tube. What is its physiological role? Which means, does it make a difference in the cell? And we obviously anticipated that if you knock out ATD in the cell, that the cell will have problem. And that's what we set out to do in collaboration with Dr. Chandrasekhar of CCMB. And to our utter dismay, what happened when we knocked out this protein ATD from both HEK cells as well as from ES cells, nothing happened, which means the cells were more or less the same. However, when we actually decided that now the knockout of its own, there's nothing happening. So can we now look at the systems under certain stress conditions? And what we found was that under oxidative stress, the cells started behaving or showing toxicity, as you can see here. Not only the HEK cells, even the ES cells showed significant toxicity upon addition of oxidative stress. So therefore, to check this is because of protein unfolding kind of issue, we took to a methodology which was basically developed by another colleague of ours, Dr. Swastika Chaudhary from CCMB. So what he has generated is a construct of luciferase. By making a double mutant of this enzyme, he made it quite fragile, which means any proteotoxicity in the cell would result in luciferase aggregation. So if you have a GFP tagged protein luciferase, if the cell were to be under any proteotoxicity stress, now the GFP will be shown as aggregation, because the luciferase aggregates and therefore the GFP will be seen as pumped up. So when we did this experiment, what we could very clearly see, while the wild type does not suffer because it has ATD, it does not suffer. But the ATD knockout under normal conditions has no known or no obvious toxicity, but under oxidative stress has significant proteotoxicity. And we also showed by mass spectrometry experiment that you have certain substitutions that are happening, which is of threonine to alanine substitution. Now, when did ATD appear in which branch of life is the next question that we asked. And this took us to organisms called cyanoflagellates. These cyanoflagellates are universal, unicellular organisms. However, they actually behave or exhibit certain behaviors of multicellularity in terms of their coming together, as you can see here, under different environmental conditions. So there's a lady by name Nicole King, who is working in University of Berkeley. What she has been working on these systems for a very, very long time by sequencing different cyanoflagellates, including this, the classical one, which is Sulbanboyaka, etc. What she had shown is that even though these organisms are not multicellular, however, they exhibit behaviors of multicellularity. And if you look at their genomes, several genes that are required for multicellularity in organisms which are multicellular have started appearing in the context of these unicellular organisms. What it means is the seeds of multicellularity or the relics of multicellularity have already developed in the context of cyanoflagellates, is what she has shown using her beautiful studies. So we now wanted to look at what is the status of ATD in the context of multicellularity. And what we found interestingly is that not only ATD, also the error-creating species of tRNA, which is this G4U69 tRNA. I will not go into mechanistic details of that, but there is a important connotation for this base pair. So both of them have started appearing in the context of cyanoflagellates, and then they were taken forward. So using this, we proposed a kind of a model, what it suggests the model is shown here, that you have significant genome expansion followed by tRNA expansion in the context of animals, which makes these kind of errors, correct, in the tRNA. And therefore, a tRNA synthetase like alanine tRNA synthetase mischarges alanine on top of tRNA threonine, and that mistake has to be corrected. Now, there are two systems which can do this job. One is this enzyme by name threonine tRNA synthetase, which can correct this error. However, under oxidative stress, it has been shown that this enzyme is no more active, and therefore, a protein like ATD, which has to appear to remove this error so that you can minimize this mistranslation, without which it will lead to cell death. And therefore, what we are currently looking at is why nature would create a problem in a particular place and solve it then and there. So the kind of questions that you are asking now is whether these tRNAs are being routed to some other purpose is what we are looking at. The next 10 minutes or so I will spend on another aspect of DTD, which I mentioned earlier. So if you look at the distribution of DTDs across life forms, you see the plants are the only systems which have both DTD1, which is coming from the bacteria, and DTD2, which is the archaeal origin version. So naturally, the question that we asked next is why should plants have both while the rest of the life forms can solve the problem with only one DTD. So we now started to look at what is the role of this additional DTD2 in plants. And when we started looking at the literature, to our surprise, what we found was that a Japanese group, when they did a screening, the transposon-based mutagenesis screening in Arabidopsis plants, what they found was that the GEK1, the DTD2 was originally known as GEK1 in Arabidopsis, then later it was renamed to DTD2. The DTD2 knockout plants were highly sensitive to ethanol, and then eventually they showed that it is not ethanol that causes toxicity, but it's basically the product of ethanol, which is, it is converted to acetaldehyde. The acetaldehyde is the one which creates toxicity. So you can add acetaldehyde directly and then show that this toxicity is mainly because of acetaldehyde rather than ethanol. Now, the question to us, which was surprising, was what is the link between a protein which is removing D amino acids from tRNA and acetaldehyde toxicity? Because this is something which we found quite difficult to link in terms of the phenotype and the biochemical function of the protein. So to test that, of course, the question was what is the link between anaerobic stress metabolite acetaldehyde and the chirality-based proofreading enzyme. So what we hypothesized was that acetaldehyde is known to modify proteins and nucleic acids. So this is known for some time. So, but the question is, where is the acetaldehyde modifying? So what we hypothesized several years back is that possibly the acetaldehyde is modifying some part of the tRNA, and this protein may be responsible for removing that modification from the tRNA is what we hypothesized, but we had no clue. And to cut a very long story short, what we showed was now the acetaldehyde can make very efficiently, ethyl adducts on top of the amino acid component of the tRNA. So if you have the amino group on the amino acid, the D-amino acid tRNAs can get modified with an ethyl adduct. And this we have characterized very thoroughly using mass spectrometry. And only DTD2 protein is responsible for removing this error, and no other protein, either DTD1 or procreal tRNA hydrolase, which also works on similar substrate, none of the protein can actually do this job. So what we showed is that the ethyl adducts that are formed by acetaldehyde on top of D-amino acid tRNAs are specifically removed by DTD, so that now the tRNAs can be recycled. The biochemical activity is shown here, but I will not go into the details. As you can see, even 5 nanomolar to 500 picomolar enzymes are good enough to have a significant activity on these ethyl adducts, which none of the other enzymes can do. So the activity is a very new activity because such a biochemical activity is not noted so far in the literature. So we named it as NEDAT deacylate activity, deacylase activity, or ND activity. And then we took a very similar approach to that of what we did in the case of the multicellular systems as to when did plants acquire DTD2 and what was the reason for DTD2 to be recruited. And when we looked at that, again, these are, of course, beautiful times for people who are looking at the evolution of processes at different time points in evolution. And very recently, there are these genome sequences of intermediates in land plants that have appeared in Nature Communications, Cell, and so on and so forth. So when they sequenced these intermediates in evolution of land plants, what they found is a very similar theme, as I was mentioning in the multicellularity scenario, that several adaptations which are required for land plant or the plants to migrate from water to land have already happened in the context of the water, the land plant ancestors. And these are the important ancestors of land plants with their sequence, like Klebsiella medium and Charophyte. And what they showed is several of these changes have already the genes have already appeared in the context of these streptophytic algae. And therefore, when we looked at the role of DTD2 here in this context, as you can see here, the DTD2 is present in archaea. So obviously it is not there in bacteria because you have the other version called TTD1. And if you look at the last eukaryotic common ancestor, you do not have DTD2 either, in this, or if you look at any of these, early algae, say for example, none of the glaucophyte, rhodophyte, or chlorophyte that are sequenced are known to have DTD2. And even, the mesostigma, which is one of the very ancient charophyte, streptophytic algae, does not have, the DTD2. However, starting from chlorocybus, which is a subaerial charophyte, you do have DTD2 and all the way up to land plants. So what we suggested is that this recruitment of DTD2 has happened at the level of land plant ancestors. Obviously, the next question that you would ask is why? Why, the land plant should have DTD2 recruitment, and that too from archaea. And of course, this is what I mentioned. So this is an archaeal origin gene, which has been shown for the first suggesting that some folds are used very often. So it kind of strengthens the view that if you have an ancient molecule, which is kind of responsible for helping the system in certain key scenarios, now you are recruiting them wherever you have a major decision to make, is what we see. Correct? So we are only trying to connect these two. If at all it is true, is there a code that defines that which molecule to be called when you have a problem or when you are in a critical junction, and where is that code stored and how is it stored is something which we will be looking at over a period of time. So with that, I will conclude. These are all the people who did all the work. I would like to particularly mention here two graduate students, Mazid Mohammed and Santosh Kuncha, whose PhD work I described today. with help from postdocs, Raghvendra Singh and Akshay Bhatnagar and others in the lab, and thank the funding agencies. And I thank you all for your attention. I'll be happy to take any questions.
Thank you for an interesting talk. I have a question regarding D-amino acids containing enzyme when it was designed or when it was made.
Who is this asking the question? I can't see.
I'm Ratnaprabha here.
Oh, okay. Oh, sorry. Hey.
Yeah.
Please go ahead.
I forgot switching on the camera.
No problem.
Yeah. So you gave this example of HIV protease.
Yes.
Yes. It is a textbook matter that whenever you have an alpha helix made with 3 amino acids, the side chains come too close to the backbone and the helix is destabilized. This enzyme does not have any alpha helix, and I was also wondering, is it that the evolution of protein folding ruled out incorporation of D amino acids in longer proteins?
Okay, so let me try to answer one by one. The first question you asked, what I was trying to explain, then you can't have a heterochiral polymer. Okay, so if I have an alpha helix made of L amino acids, which is what all the proteins are having, now if I put a D amino acid inside, correct, that L amino acid peptide, then I will have problems. However, what those guys did was to generate total HIV proteins from the amino acids. So that protein within the alpha helix would become a image of the helix that you see in the L amino acid. So that's all. Everything will be shifted from L to D. Mixing is the problem. So you can make either D or all L. What I was trying to say is the heterochirality will be the problem. So how do you remove D when you have only L? Right? So there are two issues we are talking about here. One is the origin of homochirality. The other one is the perpetuation of homochirality. The origin of homochirality is kind of fixed, so I will not go into that if somebody asks specifically. But once you decide that I want to make proteins only with L, then I have to perpetuate that very effectively, without which I can have serious issues. That is what I was talking about. So what was the second question? The evolution part, something you're asking.
One more thing I would like to know is there are many bacteria which make toxins, and also there are venoms which contain D amino acids. They are generally peptides, so the structure question does not arise. But maintaining both these types of amino acids and selectively incorporating them in the peptides.
That's a good question. That's a really good question. So the ribosomal protein synthesis is exclusively L only. Correct? There are multiple chiral checkpoints, so you cannot make these peptides with ribosomal machinery. Now, the peptide that you are referring to, depending on the cell wall of like a cell wall, are the toxins that we are affected. They all come through what are called the non-ribosomal peptide synthesis systems. So these are large molecules, correct? Huge enzymatic complexes, which have each domain responsible for adding one amino acid. So therefore, the systems use such enzymes for generating those peptides, and none of the ribosomal machinery is used for generating the peptides. I hope I answered.
You have partly answered, but the question actually is, inside the cell then there is a pool of D amino acids and L amino acids.
Yes.
And how these D amino acids escape degradation and how they are added to tRNAs, how they are prevented from adding to tRNAs.
That's what the whole talk of mine was on that, correct? So you have the cellular pool which has both L and D amino acids. Okay. Now, the enzymes that are responsible for putting the amino acid on top of tRNA are amino acid tRNA synthetases. They are normally very specific. Okay, say for example, 99% of the time they take the L amino acid, but some synthetases make once in a while a mistake by adding a D amino acid on top of tRNA. So those mistakes is what I was talking to, and how this enzyme called DTT solves the problem. I was the whole talk was on that. Thank you.
So do you have more errors incorporated in those systems where these peptides are synthesized?
Not necessarily, because the D amino acids are present across the board. Maybe I can add one more example which will make it a little bit clearer to you. Even certain L amino acids are kind of converted by racemases to D amino acid, and you keep them as an inactive amino acid pool. Because if you have a real excess L amino acid, that also can create problem. So the metabolites also are kind of controlled in the system. Right. So the D amino acid removing systems are starting from bacteria all the way to humans. There is not even one life form we know which does not have this enzyme. I hope.
Thank you for answering patiently all my questions. I think Professor Balaji has a question.
Yeah, it was a very nice, interesting work, Dr. Shankar Narayan. I have one question. So initially you showed data for the discrimination of 3 amino acid tRNAs. It was anywhere from 20 to about 12,000 or so. And also this deacylases, they are very, very sort of specific, at the same time non-specific, because they don't care which tRNA and which side chain is there. So I was wondering why nature has evolved such different types of selectivities. What advantage does it get? Because if it wants, it can always make very, very specific protein. And whenever it wants, it sort of relaxes specificity in a certain way. Is there some thought on why some tRNA synthetases are specific and others are not?
Okay, so this would require a very elaborate answer, but I will just give a very short answer to it. You can refer. I know coming from MBU, you will be able to pick it up very quickly. There are 2 models for solving this problem, which is basically an enzyme-based mechanistic problem of how you pick up chiral molecules. So there are 2 models. One which was originally proposed in the 1950s, if I am not wrong, by Ogelson, which is called a 3-point model. Okay, 3-point attachment model. So if I have a chiral system, how enzymes would recognize it by attaching to 3 entities. So that was the original model. Little later, Junior Coastline modified it something like 10 years back, which was a Nature paper in which he published that these 3 points of attachments are not required, are not sufficient, but you need 3 points of attachment and one approach, which is called 4-location model. Okay, that explains most of the tRNA synthetases which are recognizing smaller amino acids would recognize them with very high specificity. However, when you have an amino acid like say tyrosine or say aspartic acid, okay, now their active sites are much larger. So you have 2 different ways of placing the side chain by attaching to the same chiral point. So that generates this error for some tRNA synthetases in today's scenario. So this is of highly evolved tRNA synthetases of today. However, when you go to say pre-Luca, okay, the last universal common ancestor, majority of the synthetases may not be that chiral specific because you are still trying to fine-tune the systems towards chiral selectivity from a very ancient enzymatic module. So there those errors are going to be very high, and therefore you have to have something like a chiral proofreading system called DTD to solve those problems. So that is how the systems are evolved. I hope.
Yeah, I got it. Yeah, thank you so much. Yeah.
Thank you, Balaji. Any other questions?
Thank you very much, sir, for accepting our invitation and giving this talk. There are many questions regarding evolution of living systems and the choice of L amino acids over D, and Also, it's continuation.
Sure.
Thank you very much.
Thank you very much for the invitation. All the best. Yeah.
So we break for lunch now?
Yes, we take a break for lunch and join at
1:30. 1:30 is the schedule.
1:30.
Yes,
Yeah.
yeah. So
Join at 1:30 and we continue with the later half of the symposium. Yeah.
Hello, Dr. Prabha. This is Professor Rizwan. So, yeah, my lecture was scheduled to be around 12. I think.
It's 1 o'clock. Actually, I think there is some delay or there's some mix-up. So
Okay.
can we have your?
Or can we take, Ratnama, can we take, if Professor Khan wants, can we take lecture now and then break for lunch after his talk if the other person is knowing accordingly?
No,
that is possible. That is possible. Then
No. What I am suggest, you should have your lunch. I'll be also going to have something. So can we get assembled around 1 o'clock?
Then it becomes less than 20 minutes. Will it be okay at 1:15?
Yeah, that's okay. That's fine.
Yeah.
That's
Instead
fine.
of 1:30, we meet at 1:15.
Yeah, 1:15 is okay.
Yeah. Let all participants know that.
Okay, 1:15 will be back.
Yeah.
Okay.
Yeah. Thank you. Thank you. So please inform all our students and the faculty members.
And don't close the meeting. Let the meeting be on here.
Yes.
Yeah, I think that is better. Please inform everybody.
So, Sandeep, you can put a slide that we are breaking for lunch, meet at 1:15 and put it on display in this meeting. Sandeep or Satakshi, whosoever is here.
Good idea. Yeah.
Yes, that will be good. Keep sharing that till 1:15.
I'll call up Satakshi. Okay.
I think, yeah, that is the best way to do this.
Good, Ravi sir. It's not showing up yet.
I don't know what is the problem.
Now it's showing up.
Showing up? Okay.
And student groups also, we have to put the same message.
Yes. So let this be on display. Gayab ho gaya, sir.
So there is some problem. I don't know what. Okay. Let me do it again.
I have put it in research scholars and MSc groups also.
हेलो संदीप, ये अनशेड्यूल्ड ब्रेक हो गया है 1:15 तक, तो मीटिंग को क्लोज नहीं करना, उसको ऑन रहने देना। अभी रवि सर ने एक स्लाइड बना के डिस्प्ले किया हुआ है मीटिंग जूम एट 1:15। ठीक है, हाँ।
Hello, Dr. Ratna, how are you?
Hello, I'm good. How are you, sir?
Yeah, fine. So there was slight change in the this your schedule.
Actually, what happened? We made one old schedule, and after that, we found that one of the speakers was not allotted time. Okay. Then we moved them up and down. In that, there is this discrepancy in the schedules.
Okay, it's fine.
Sorry for the inconvenience caused to you.
Yeah, it's all right. There is no issue.
Today is a working day for you.
No, it's holiday. What about your office?
We also have a holiday. But then we have announced long ago, so we thought that we continue with.
Yeah, I was in this mission that you may reschedule this.
We felt that then we will again be inconveniencing the speakers. See, once we have told them that their talk will be on first, they would make all their arrangements accordingly. Even though there is no travel involved here, they would have the other official responsibilities which they might have readjusted. And it should not cause inconvenience. And there are speakers from abroad.
Yeah, I've seen that.
Considering all that, we thought that we will go ahead.
So how much is the faculty strength?
In our department, there are 18 positions, but there are many retirements in the recent past.
I see.
The number has come down to 8 or 9. That's all.
So even it's less than half.
And then we have some temporary positions sanctioned by the university to take care of this additional load. So 4 more people are there in the department. But the temporary positions are only for 1 year duration.
And what are the area in which people they are working?
I have been working on protein degradation and protein structure, also structure-function relationships. And I do a bit of protein engineering, targeted protein degradation. This is the area I have been working on. There are different people who are working on neurochemistry. There are people working on medicinal chemistry. People working with plant systems, hormones, endocrinology.
You have microbiology department also?
We have a separate department. It is not within biochemistry.
So you don't have a life science faculty?
No, it is science faculty.
So how come you don't have a life science faculty? And the science faculty, you have all these chemistry as well as
physics, biology, geography, variety of subjects. Math, statistics. There are 14 departments.
What about Dr. Chittu?
He expired some time ago.
Oh, it's very sad.
Yeah. He was associated with Microbiology and Biotechnology Center.
Yeah, I know him.
Yeah.
So he was very dynamic person.
Yeah, he served as VC also.
Yeah.
Of Vaishnodevi University.
Yeah.
Srinagar.
So you have online classes or offline classes?
Both are going on, and mixed mode is also going on. Because there's a problem with the hostels.
I see.
Hostels have not removed COVID regulations completely. So they are not accommodating to maximum capacity. Only 40% or so our students are allowed now.
I see.
Then more than the remaining students have to find PG accommodation, or they have to rent a flat or something like that. That also has its limitations right now. So many people who are from distant places, we have students coming from Kerala, we have students coming from Northeast, students coming from Lucknow, students coming from Madhya Pradesh, Rajasthan, or Delhi. Where would they all stay?
Yeah, right.
So that's why the classes are going on in mixed mode.
Yeah.
What about you?
We also having online classes. Now, recently we have a notification that from 3rd of the March onward, the final student, they will be allowed.
Okay.
Only the final year, whether it's a BSc or whether it's a MSc, and later on the research scholars, they will be also allowed.
We have final year students similarly. All the final years are that. And also PhD students. PhD students were never asked to leave the hostels. They continue.
I see.
Yeah. So now we can have the talk.
Yeah. Shall I upload my presentation? Let me find out there.
Yeah. Ravi, you can introduce this.
I'll go ahead with that. Okay, it is, I am Ravi, working as assistant professor at Department of Biochemistry. It is my immense pleasure to introduce to you our next speaker, Professor Rizwan Khan, to you all. Professor Khan is working at International Biotechnology Unit of Aligarh Muslim University at Aligarh. His research thrust areas include protein structure, protein function, aggregation, amyloids, and especially in the two disease models, Alzheimer's and Parkinson's disease. He has over 325 research publications to his credit and with over 12,000 citations in prominent peer-reviewed journals. He is serving as editor in several international journals and is also a member of several protein societies. Today he is going to talk about attenuation of amyloid fibrillation in presence of vitamin and clinically approved drug. Over to you, Professor Khan. We look forward to your talk.
Thank you, Ravi, for the nice introduction. I don't know how come I can, there is some issue because I'm just clicking this from the here presentation site. So how come it's not taking up this command?
Okay.
Have you kept your presentation open?
Yeah, my presentation is open.
Yeah, it is visible now.
It is visible. So now you can go into presentation mode, full screen.
Presentation is visible now.
Yes, it is visible. Yes. It is visible. Yes, it is fine. Yeah, now we can go ahead.
It's visible now.
Yes.
Okay,
it is visible. Great. It is full screen.
Yeah, it's full screen. So
yes.
So that means I cannot see the rest of the speaker because
that is
okay, that's immaterial.
Yes.
So today I will be speaking on attenuation of amyloid fibrillation in the presence of vitamins and clinically approved drugs. So myself, Professor Rizwan Khan from Interdisciplinary Biotechnology Unit. I'm working in this university since last 2 decades. So with this brief introduction, let's start a brief introduction about the fate of the protein.
Can you increase your volume?
Again, I'm supposed to, okay. Okay, let me find out.
Sir, you cannot increase that way. You just have to be closer to the mic.
Okay. Is it clear now?
Yes.
Audible?
Yes.
Okay, that's great. So let's talk about the fate of the proteins. So once,
please make it again in the PowerPoint full screen mode.
Okay, full screen mode. Is it okay?
Perfect. Yes, perfect. Please.
Okay, that's great. So once the protein that is going to get synthesized on the ribosome, it's going to fold properly and it's going to adopt a proper three-dimensional structure. And this hello, there is some background noise that is coming up. So
I would request all of you to please mute your mics, please.
So once the protein that is going to get synthesized on the ribosome,
Sanjeev sir, please mute your mic.
It's going to perform a normal function. So that means it requires a particular three-dimensional structure for performing a biological function. But in some cases, there will be the misfolding of the protein, and this may lead to the loss of the function, and this may lead to the inborn error of the metabolism. In some other cases, there will be the overproduction of the protein. There will be the overproduction of the protein, or because of the formation of the hydrophobic patches, they are going to stuck to each other, and they lead to the formation of the aggregates. And these aggregates, they are responsible for the neurodegenerative disorders. In some cases, there will be the mutation in the protein, and these protein or the misfolded protein, they will be recognized by the proteasome and they are being degraded. So with this brief introduction, we are going to ask several questions. How come we can solve the So the first hypothesis that we have given, since we know there are the different classes of the protein, like alpha class of protein, beta class of protein, and the random class of protein. So can we have some standardized method for the induction of the amyloid fibril formation? Because we know a lot of neurodegenerative diseases and many other diseases, it is because of the formation of the amyloids. So here we want to standardize the taking the different classes of protein to get the amyloid fibril formation. So this is a study that has been published in PLOS One in 2012. So here we have proved that SDS can be used as an amyloid inducer for different classes of the protein. So now this slide is very important because here I am going to discuss about the basic techniques that we have used to characterize the amyloid fibril formation. So the first one is the relay scattering. This is a fluorescence based where we are supposed to excite the protein at 350 nanometer and take the emission at 350 nanometer. This is very sensitive technique. Then turbidity measurement, which is performed by the spectrophotometer, where we are supposed to take the absorbance at 350 nanometer. Then the amyloid, they have a unique type of beta structure, and these can be characterized by the two very unique dye. One is known as a TST thiofluorine dye. So this is also fluorescence based, where you are supposed to take excite the protein at 340 nanometer, and you will be getting the emission at 485 nanometer. And the second dye that is being used to characterize the amyloid, that is the Congo red. In this case, you are supposed to take the absorption spectra in the presence and absence of the dye. If you are going to get, and you are supposed to scan the protein spectrum from 400 to 700 nanometer, and if there is a red shift upon the binding of the dye, so then this is going to confirm that there is a amyloid fibril formation that is taking place. CD measurement is being performed just to characterize the beta sheet content. So here you are supposed to take a CD spectrum from 190 to 250 nanometer. And for the beta sheet, you'll be having a negative peak that may confine around 210 to 228 nanometer. Seeing is believing. Until unless you can see the structure, you cannot believe that. So for that purpose, transmission electron microscopy is being performed to study the morphology of the protein aggregates. So in my coming presentation, I'll just talk about all these techniques. So be very careful about these techniques. So now here I'm going to talk about the basic concept about the protein chemistry. So we know that protein, they may be having a different number of positive and negative charge amino acid residue. Positive that will include histidine, arginine, and lysine, and negative that will be aspartic and aspartic acid and glutamic acid. Now, each protein, they will be having a unique isoelectric point. Now, if we are going below the isoelectric point, so in that case, the protein, it will acquire a positive charge. And if we go above the isoelectric point, so in that case, the protein that will be acquire a negative charge. So with this brief introduction, now I'll come to the result part. Now, again, the first hypothesis that we have given that we want to see if we are going to take the alpha class of protein or the beta class of protein or the random class of protein. So do we have some common mechanisms to induce the amylase fibril formation in all these different classes of the protein. So here what we have thought about that if you are going to take the SDS, because SDS it is going to mimic the characteristic of biological plasma membrane. So it is because of that we thought to take a SDS. And the protein that were being taken, which are being listed over here, that include human cerebrolin, porcine, bovine, sheep, rabbit, ovulin, fetuin, acylofetuin, glucose oxidase, invertase, alpha-lactobuline, alpha-amylase, and column. And if you just see the data over here, so these proteins, they are predominantly rich in the alpha helical structure. So that means they will be having the rest of the other structure also, but predominantly they will be having a alpha helical structure, and it is because of that these protein they are called as an alpha class of protein. Then we have taken alpha plus beta class of protein that include lysozyme and hemoglobin. And the third set that we have taken a beta class of protein where we can see they will be rich in the beta sheet content. Along with that, they will be having rest of the other two also. And that include concanavalin A, cobra toxin, beta lactoglucan, bovine liver catalase, camopypin, ratagine, and benalactin. And one protein that we have taken that belongs to a random class protein, that is the gelatin. So as I earlier pointed out that once the protein that is going to get synthesized in the cell, it's going to come into the close contact with the plasma membrane, and the plasma membrane, it is made up of a phospholipid, which will be having a hydrophilic head and hydrophobic tail. And keeping this point in mind, we have selected SDS, which will be having a negatively charged head group, 12 carbon long hydrophobic tail, and CMC is around 8, and this is going to mimic the characteristic of biological plasma membrane. Now coming to the result part. Now, if you just see the table that is being given at the right bottom, so I'll be talking about all these techniques which we have already introduced in the introductory part: Rayleigh scattering, turbidity, GST, CD, and Dept. Now here the Rayleigh scattering data that is being shown for all the 25 protein that were being taken, which belongs to all the 4 different categories like alpha, alpha plus beta, beta, and random. And just to see that whether if we are going to take all these protein at 5 micromolars concentration at neutral buffer, whether they are going to impart in any aggregation or not, or Rayleigh scattering. So you can see there was no Rayleigh scattering that was being observed once we are taking all these protein at neutral buffer. In the other set, we have taken a 5 micromolar protein 2 unit below pi. Even in this case also, we won't get any significant positive Rayleigh scattering. But once we take a 5 micromolar protein 2 unit below pi along with the 500 micromoles SDS, so in that case, irrespective of the classes, all the protein, they show a positive Rayleigh scattering. While on the other side, if we take a 5 micromole protein 2 unit above pi along with the SDS, it is the same condition, but here it's a below pi and it's above pi. So here you can see this is the purple one, and here also you can see there is no positive Rayleigh scattering that was being observed. So with this data, we can conclude that the condition that is required for the Rayleigh scattering, that is 2 unit below pi along with the SDS. Under the similar condition, we have performed the turbidity. Now we are over here, and you can see all the protein which were being taken in the previous set, they also show a positive turbidity measurement. Then we have performed the TST. This is a dye, as mentioned earlier, that this is a very specific dye which is going to bind with the amylase. So here we have taken care of the control also. So we have taken protein 5 micromoles at neutral buffer along with the TST dye. So you can see there is no TST binding that is taking place. Only the case where we are taking a 5 micromolar protein 2 unit below pi along with the SDS, that shows a positive TST band. While on the, if we are going above pi also, in that case also we won't get the any positive TST band. So this further confirms that the condition that is required for the amylite fibril formation, it is a 2 unit below pi along with the SDS that favors the formation of the amylite. Now this is the CD data that is being shown. This is at neutral pH. So if it is alpha class of protein, so it will be getting a 2 negative peak, and if it is a beta class of proteins, and this is the unordered, so these are the proteins which were being CD spectra that were being taken at the neutral pH, and this is 2 unit below pi. So there was no significant change in the structure that was being observed. Interestingly, once we take all these proteins 2 unit below pi along with the SDS, all the protein, they are going to show a negative peak, very prominent negative peak that may be between 215 to 224, which is a characteristic feature of the beta sheet. Now in the table also it is being shown that if you just see these are the proteins, these are being listed over here, and this is the beta sheet content, and if you just see it is over here, this is at pH 7.4 and this is 2 unit below pi. So there was no significant change in the beta content that is being observed if you just compare this table with this one 5, 8, 6, 6. Once we take this protein at 2 unit below pi along with the SDS, so in that case you can see there is a significant increase in the beta sheet content that takes place. So all the protein, they are going to adopt a beta sheet structure. So now here the 10 images that are being shown of all the different classes of protein like alpha, alpha plus beta, beta, and unordered structure protein. And here you can see they show a clear amylite fibril formation. Now this is a summary of the first hypothesis. So if again we can recall what we have started, that we have said we have taken the protein 2 unit below pi, where the protein, it will acquire a overall positive charge, and then we have added a anionic detergent. So how it is going to influence the formulation of amylite? So we have taken alpha class of protein, beta class of protein, alpha plus beta, and randoms. And once we are going below pi, 2 unit below pi, so the protein that will acquire a overall positive charge irrespective of the classes. And then this is the red one, which shows the structure of the SDS, and this is the head group, which is a negatively charged. This is going to neutralize the charge that is present, or positive charge on the protein, and this may facilitate the formation of the amylite. While on the other side, if we are going above pi, so in that case the overall charge on the protein, it will be negative, and there will be the negative charge repulsion that will be there, and this is not going to favor the formation of the amylite. So with this first hypothesis, we have concluded that SDS have a great potential to induce amylite fibril formation when protein are subjected to 2 unit below pi. Right now, what we can say, it's the electrostatic interaction that is playing an important role, because we have seen there is a neutralization of the charge that is taking place. Although we can hypothesize that hydrophobic interaction that is also important, which we are going to prove in the coming slides. So now what is the second hypothesis that we have given that from the first hypothesis, it becomes clear that only thing that is required is we should have a either if we are going below pi, so in that case the protein, it will be having a positive charge, and then if you are going to add the anionic detergent, so this will neutralize the positive charge on this protein, and this may facilitate the formation of the amylite. So now what we want to do in the second hypothesis, we just want to take a same protein and same detergent, but by changing the pH, we can change the positive charge on the protein. So let's see what is going to happen. So here, this is the second hypothesis that we have given. So we have taken a lysozyme protein. Just assume that at one pH it will be having 8 positive charge, and on the other pH, once if we are going below pi, so it will be having more positive charge. Say assume it may be having 14 positive charge. The protein concentration is the same and the detergent concentration is the same. And here you can see we get a 1 bundles of the amylite fibril formation because it's having 8 positive charge, and here it's 14. So what we speculate that here we should get more amylite fibril formation because the charge is more. So this is this was our hypothesis. So let's see. So so here what we have asked that do charge affect SDS induced aggregation of lysozyme? And this is a study that is being published in Soft Matter in 2014. And the protein that is being taken is a hen egg lysozyme. This is alpha beta class of protein, 129 amino acid residue. 17 are positive charge and it's having 2 domain. And the most important point over here is the isoelectric point of this protein is 11. So then coming to the result part, we have calculated the charge on the protein by charge calculator 3.3. So in this case, what is required, if you know the sequence of the protein, you just feed the sequence on the computer and you'll at a different pH, you can hypothetically you can calculate what will the charge on the protein. So this is a near about the isoelectric point. So here you will be having a 0 charge, and as we are going to lower the pH, because this is the isoelectric point, as we are lowering, so you can see the charge, the positive charge on the protein that is going to get increased. So later on, I'll just ask because these 2 pH that will be important while I'm going to summarize this data. So at pH 9, it will be having approximately 5 positive charge, and at pH 1, it will be having approximately 19 positive charge. Now here what we have done, since this is a different set of the experiment, so what we have done, we have taken 15 micromoles of the lysozyme, and we don't know at what concentration we'll be getting the maximum Rayleigh scattering. We'll be getting the maximum Rayleigh scattering. So here you can see with the varying concentration of the SDS, we have monitored the Rayleigh scattering at 350 nanometer, and we got 0.3 millimole concentration that is giving a maximum Rayleigh scattering. And this is the observation that is being taken at pH 7. Now the rest of the observation at different pH, it will be taken with a 15 micromole concentration of the lysozyme and 0.3 millimoles of the SDS at different pH. So now I'll shift to the another slide. Now here you can see 15 micromoles of the lysozyme. 15 micromoles of the lysozyme, and it is being taken with 300 micromoles or 0.3 millimoles of the SDS at the different pH. So here you can see this is near about the isoelectric point. And as we are lowering the pH, so you can see the Rayleigh scattering that is increasing. So indirectly, we can say that the amount of the aggregates or amylite fibril formation is also being increasing as we are lowering the pH. Just excuse me. Hello. हेलो सेमिनार चल रहा है सेमिनार नहीं सेमिनार है so sorry for the interruption. So here you can see as we are lowering the pH, so we get at around pH 1 where you can see it's having a maximum positive charge, so we are getting a maximum Rayleigh scattering. While this is the site where we are going below the pi, so here the protein it will be a negatively charged and SDS is also negatively charged, so there won't be any interaction, and it is because of that we won't be getting the positive Rayleigh scattering. So sometimes we may be getting the artifacts. So just to overrule that, we have performed the kinetics also. So what we have done, we have taken the protein and the SDS at a different pH, and then we have monitored the relay scattering up to 1000 second. And you can see there is a very steep hyperbolic curve that is being noticed for pH 1, followed by pH 2, then pH 3, and then pH 5, and then this is at pH 7, and then you can see it becomes static. So from here we can conclude that it is not an artifact whatsoever, the observation that we are getting previously. So here from the kinetics also it's proved that once we are going to take a low pH preparation, so in that case it is a very steep hyperbolic curve that we have noticed. Then we measure the turbidity, and you can see in the turbidity also we get a maximum turbidity that is being observed at pH 1. And this is a TST binding assay just to characterize whether this is amylite or not. So the data is being summarized in this table. Now, if you just see, this is unexpected. We were supposed to get a maximum TST binding at pH 1, followed by 2, then 3. But here, what we have noticed that this here we are getting a maximum TST binding at pH 3. The reason is that TST micelle, which is being formed at low pH, they are being disrupted. And it is because of that we are not getting a maximum TST binding at pH 1 and 2. So there are most of the dye which are very sensitive, even the Congo red also, they are sensitive towards the pH. They can work under some certain pH range. And now this is a Congo red data that is being shown. If there is a red shift, so this will indicate there is a formation of the amylite that is taking place. So even in the Congo red also, we cannot go below pH 5 because in that case, we may be getting the false result. So this is the spectrum that is being taken at pH 7 and 5. So you can see the spectrum that is being shifted towards the right-hand side in the presence of the Congo red, which is indicating that there is a formation of the amylite taking place. Now, this is the table that is being summarized over here. You can see this is the pH of the protein that is being shown, and this is the lambda shift that is being reported. So as we are nearer to the isoelectric point, so we are not going to get a red shift. But as we are shifting towards going towards the lower pH side, like 9, 8, 7, 5, 6, 5, so you can see the red shift that is being increasing. And as I told you, we cannot go beyond pH 5 because in that case the dye is going to lose the color. Now we are over here, CD data. So this is the CD, some representative spectrum that are being shown. So pH 1, 3, 5, and 7, just for a clarity sake. And since this is the lysozyme, this is alpha beta class of protein. And this is in the absence of the SDS, and this is in the presence of the SDS. So you can see it forms a beta sheet-like structure with a very prominent negative peak that is around 218. Now these are the 10 images that are being shown. This is at pH 9, 6, 3, and 1. And if you just see at pH 1, it is more dense amyloid fibrils that are being formed. So now this is the summary. Now again, if you just recall that we have taken calculated the charge on the lysozyme, and it was having a 19 positive charge at pH 1, and at pH 9 it's having a 5 positive charge. So here you can see this is at pH 9, so it's having a 5 positive charge, 1, 2, 3, 4, and 5. And one point that you should keep in mind, that the stoichiometry of the protein to detergent is same. So here we just want to prove that if the stoichiometry is same, like 1:20, So if somehow there is a charge that is going to get increase or decrease, how it's going to influence the amylite fibril formation. So here you can see that there is a 5 positive charge. And now 1:20 stoichiometry of the protein to SDS that is being taken. So now this detergent is going to neutralize the charge. And just for the sake of the explanation, we have shown 2 bundles of the amylite. Now here at pH 1, it will be having a 19 positive charge. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 16, 17, 18, 19. And the stoichiometry is the same. But here, if you just see the numbers of the bundles that is being increased, although the stoichiometry is same. So that means from here, what we can say that the take-home lesson is, okay, as we are going to increase the charge on the protein, it's going to favor the more amylite fibril formation. While on the other side, if we take the protein at pH above 11, so in that case, the protein, it will be having a negative charge and there will be a charge-charge repulsion. And in that case, we won't get the formation of the amylite. So from here, we can conclude the aggregation is also to be maximum at pH 1 because of the maximum net positive charge on the lysozyme at pH 1. SDS interact electrostatically and lead to the formation of amylite fibril formation. So till now, we have talked about the interaction, and we have seen that SDS is interacting with a positive charge protein, and it's neutralizing the charge, and it favors the formation of the amylite. So what is the role of this tail? We haven't discussed the role of this tail. Somehow it may be also having some role in the formation of the amylite. So in the third hypothesis, what we have done, we have taken a different albumins. Because the reason is all these albumins, they have more than 90% sequence homology, as well as the isoelectric point is also the same. So human seroalbumin, bovine seroalbumin, porcine seroalbumin, sheep and rabbit. And now we have taken a negatively charged detergent, but with a varying chain length, hydrophobic chain length that is being varied, so that we can show the effect of the hydrophobic chain length. So this is our aim. So this is a study that is being published in Physics, Camp Physics in 2014. So here the question that is being asked, does hydrophobicity affect amylite fibril formation? So here we have taken a detergent like SDS, SDVS, and AOT. All of them, they are anionic, but they differ in the chain length. So, and one thing is important, whatever the 5 albumins that we have taken in this study, all of them, they have near about 5.6 isoelectric point. So this is the beauty of the system, that all these albumins, they have a isoelectric point of 5.67 or 5.6. And the reason is, if we are supposed to perform on the one set of the protein like human seroalbumin, so we may be having some doubts. So why didn't we have checked on some other proteins also? So here, whatever the data that we are going to show, it is not on one protein, it is on the 5 protein. So from here we can conclude in a very different way. So this is the structure of the detergent that is being shown, that is SDS, which is 12 carbon, and it is negatively charged. Then SDVS is 18 carbon. It's also having sulfonate group. This is also having an AOT. This is also having a negative charge, and this is 20 carbon. So only thing is, the head group is the same. It's a sulfonate, negatively charged, and the chain length is different. In one case is 12, 18, and 20. Now again, we'll follow this table. That is Rayleigh scattering, turbidity, TSTs, congruent, CD, and TEM. And since we have taken the 5 proteins, so I'll explain one data, I will explain one figure, and then I'll say that for the rest of the protein, there was the same pattern. Say for instance, in the case of HSA, the Rayleigh scattering that is being observed in the presence of varying concentration of these detergent like SDS, SDBS, and FD up to 11 millimole. So here you can see in the presence of the SDS and SDBS, the Rayleigh scattering initially increase and then it falls down around 3.5 millimole, and then it becomes constant. While in the case of AOT, which is 20 carbon chain length, it remains persistent even up to 11 millimolar concentration of the surfactant. A similar pattern is being observed for the BSA, PSA, SSA, and RSA. Then we have performed the kinetics. Again, you can see this is the AOT, and here there is a steep hyperbolic curve that is being observed, followed by SDBS, and you can see there's a significant decrease in the curve that is being observed for the SDS, and similar pattern that is being observed for the BSA, PSA, SSA, and RSA. Then we perform the turbidity measurement, and here you can see this is a turbidity measurement that is being performed on the spectrophotometry. Again, it's the same pattern as we have reported in the Rayleigh scattering, that for the AOT it remained persistent even up to 11 millimolar concentration for all the 5 classes of the albumin. Then we have performed the TST binding assay just to confirm whether they form the amylite or not. So again, here it shows the same pattern for all the 5 albumin, and this is a Congo red. You can see there is a red shift that is being reported in the presence of these detergent for all the 5 classes of the albumin. And this is the CD data. Now we are over here. CD data that is being shown. This is the albumin, the HSA, and this is in the presence of SDS, SDBS, and AOT, 2 millimolar concentration of each. And you can see they are going to adopt a beta sheet-like structure, which is going to favor the formation of the amyloid. Now we are over here, the 10 images that are being shown. The upper panel is for BSA and the lower panel is for human serum albumin. And this is in presence of the SDS, this is SDBS, and this is AOT. And in the case of AOT, you can see there is a more dense fibrils that are being formed. So from here, this figure requires a little bit attention. I will let you know why. See, in the beginning, we have said that we have taken 5 albumin, and each albumin they have a isoelectric point around 5.6 or 5.5, and then here the result is being summarized. So this is the albumin, and isoelectric point is 5.6, and just see my pointer. These are the results that we have discussed so far. I will let you know why we haven't discussed the result, although we have performed the experiment. I will let you know later on once I am going to complete this part. So once we are we know that isoelectric point is 5.67 on all the experiment that we have discussed so far. In the third part, the protein, all the albumins, they were being taken at pH 3.5. That is 2 units below PI. In that case, all the albumin, they will be having a positive charge. Now you might have noticed that once we are adding a low concentration of the SDS, so there is a like from 0.5 to 2.5, it's promote the formation of the amyloid. Similarly, the case for the SDBS also, because it's 12 carbon. So in that way, we can show that in the SDBS, which is 12 carbon, we get 2 bundles. This is a 18 carbon, where we get this 3 bundles, and this is 20 carbon chain length. Here we get a more amount of amylite fibril formation. And one thing that is important, that here we get the amylite fibril formation from 0.5 to 11 millimole. But here, if you just notice, not beyond 2.5. So in all the figures, if you can see, So it becomes constant, while in the case of AOT, it remains persistent. So what is going to get happen if we are going to above 2.5 millimolar concentration of the SDS or SDML? So in that case, what we have hypothesized that it is going to go to the some other form, that is F prime form. And these data, although we have performed the experiment, since there was no change in the spectrum that was being observed, so it is because of that we haven't shown the result over here. So what is going to get happen? So if you are going to add the this SDS, so this is having all the alveolus, they will be having a negative charge because we have gone above the pi, and since the negative charge, they will repel each other, and most likely the hydrophobic tail they would like to orient like this. So this is our hypothesis. So even the hydrophobic tail, they are not responsible for any change in the structure. So only thing is, the first criteria is there should be the neutralization of the charge and then followed by the hydrophobic interaction. So electrostatic interaction play an important role in amylite fibril formation followed by hydrophobic interaction. So I'll skip this part because that is going to consume more time, because I think I'm supposed to finish in 1 hour. So let me come to the, okay, I'll just let you know. So till now, there may be some question like that. So it looks like very biased that I'm just talking about the positive charge and the negative charge, but it's not the case. Even if you take the protein, this which is having a negative charge, and in that case, if you are going to add the cationic detergent, so in that case also we get the formation of the amylase. So this is also studies published in the very reputed journals. So now I'll come to the therapeutic approach. So recently we are working on this aspect. So there are the different approaches that is being used. Inhibition of protein aggregation by small molecules. So our lab is focusing on the small molecules. RNAi interference, that is also being used to inhibit the incorrect foldings, and upregulation of the chaperones for the clearance. So now I'm going to talk about the role of the small molecule, especially with reference to the, so our lab has taken published more than 100 papers on these small molecules. So just because of the limited time, I'm just going to speak on one aspect, that is the how the vitamin K that is going to inhibit the amylite fibril formation, and some anti-tuberculosis drug, they also play an important role in amylite fibril formation. So I'll go very quickly. So here we are being, this is the study that is being published in Scientific Report in 2016, where we have shown the role of the vitamin K3. So this is a synthetic analog. This is having anti-inflammatory and anti-cancer agent. And here we have taken two proteins. One is the hen-acquired lysozyme, and as well as A-beta-42. So these were the conditions that were being used for the induction of the amylite in the presence and absence of the vitamin K3. So now here you can see this is the TST fluorescence that is being shown. So you can see in the presence of the increasing concentration of the vitamin K3, the TST fluorescence that goes down. And here we have shown how the kinetics that is going to get influenced. So you can see 50 micromole, which is the midpoint of this inflection point, that significantly decrease the kinetics. At 100 micromoles, it's almost diminished. And this is the Congo red data that is being shown. This is the free dye Congo red, and this is in the presence of the 100 micromoles. So it is coming approaching towards the free Congo red, and this is in the presence of the 50 micromoles of the vitamin K3. So this shows that vitamin K3 shows a very pronounced inhibitory effect on human lysozyme as well as A-beta-42. Now, this is the NS fluorescence data also that is being shown. Now here you can see the hydrophobic patches that are being decreased. Now these are the TEM images that are being shown. So this is the control where we won't get the formation of the amyloid under the room temperature. This is the condition, like 65 degrees centigrade for 120 hours, where we get the formation of the amyloid. And this is in presence of 50 and 100 micromoles of the vitamin K3. So you can see these amyloid fibers, they are being precipitated. This is in the presence of the, this is the protein A beta 42. Again, it's the same pattern that is being obtained. Now, MDT assay is being performed, and you can see the cell viability is being restored in the presence of the 100 micromoles of the vitamin K3. So we can conclude that vitamin K3 inhibits the aggregation of egg-white lysozyme as well as A beta 42. Vitamin K3 reduces the cell type toxicity. This study may help us to design rational molecules for the treatment of the amyloid-associated disease. Now, we are working on anti-tuberculosis drug, and we have screened almost 10-15 drugs, anti-tuberculosis drugs, and we have shown that these drugs, they can be also used for the treatment or for the Alzheimer's or Parkinson's, like that. So this is the study that is being published in PLOS One in 2015. So the protein that were being taken, human lysozyme, and the drug that is being taken, pyrazinamide and D-cycloserine. These are the structure that is being shown. And you can see this is the turbidity measurement that is being performed just to find out what is the time of incubation and the concentration that is required. So, and then this is being performed in the presence of the varying concentration of the pyrazinamide and D-cycloserine. And from here you can see pyrazinamide is to be more effective as compared to the D-cycloserine. Then this is the kinetics data that is being performed by using the TST fluorescence. Again, you can see the 500 micromoles of the pyrazinamide. This is going to prolong the lag phase. This is the Congo red. Again, it shows that it's this pyrazinamide. It's more potent inhibitor as compared to the D-cycloserine. This is a fluorescence spectroscopy. This is the control, and this is in the presence of the pyrazinamide and D-cycloserine. Here you can see it's a more clear picture in the presence of the 500 micromoles of the pyrazinamide, which indicates the it's inhibit the amyloid fibril formation. Now, MTT assay that is being performed by using two different type of cell line, PC12, SY5Y cell line, and in both the cases, we have seen the cell viability is being restored in the presence, more significantly in the presence of the 500 micromoles of the pyrazinamide as compared to D-cycloserine. Now, this is the overall summary. Now, lysozyme, if it is being incubated at 65 degrees centigrade, it's going to adopt a partial folded structure. And if you are going to add pyrazinamide and D-cycloserine, so it will prevent the formation of the amyloid. So now the last chapter. So here we have taken another drug, anti-tuberculosis drug, that is the capromycin. And this is a study that has been published in PLOS One, and I'll come to this the result part very quickly. And here, what is the interesting part is you can see even the very low concentration of the capromycin that shows a very significant TST fluorescence inhibition. And at 100 micromoles, you can see the lag phase is being prolonged. And here, what we have done, we have added the 100 micromoles of the capromycin at different phases of this growth curve. And you can see it significantly affect the growth curve. So if you are going to add the capromycin after 8 hours, so you can see the kinetics is totally being altered. And if you are going to add after 18 hours, so also you can see it's going to alter the kinetics of this. These are the time images that are being shown, and you can see from here also it shows a clear-cut inhibition. Now this is a very interesting part. So there are the two ways of the therapy. One thing is the person is already having the neurodegenerative disorder. and the amylite, they are already being formed. So in that case, how come we can go for the treatment? So in that case, we are supposed to disaggregate the preformed amylite fibrils. So here we have shown how come the preformed fibrils, they can be disaggregated. So this is the preformed fibrils that are there, and in the presence of the fixed concentration of the capromycin at 12 hours and 24 and 48 hours, you can see there is a decrease in the TST fluorescence that takes place. And here you can also see the hemolytic assay, the hemolysis is being decreased, and this is the image that shows that at 48 hours, capromycin shows a significant disaggregation of the preformed amyloids. So from here we can conclude that capromycin suppresses the insulin fibrillation in a concentration-dependent manner and possesses a destabilizing effect on a mature insulin fibril, effective in blocking fibrillation of insulin when added during elongation phase. This we have seen, we have added at different time intervals of the growth curve. Capromycin and with the extension of the other anti-tuberculosis drug, which can show the possibility of amylite inhibition. So recently we are working on the combination of, because we know that in the tuberculosis, there is a combination of the drug that are being given. So recently we have published one data. So I'll share whenever I'll be getting time in the next meeting. So with this, I'll just summarize. We have published around, this is not an updated one, more than 350 publications, and we have received the fundings from DBT, DST, ICMR, UGC. I have supervised more than 25 PhD theses, some MD, MS thesis. Total citation is not, it's not updated one, it's 3 years back data, so it's around 16,000, I think. And I'm serving as editor for several journals, also visiting fellow to University of Michigan and University College London. I'm fellow of Biotech Research Society also. And I'll be happy to have a collaboration with your university if someone is willing. And this is my research group. Most of the work is being carried out by Dr. Javed and Dr. Sumit Agarwal, who is presently is a faculty at NIH. And last but not the least, I would like to thank the funding agencies like DBT, DST, ICMR, UGC for the financial support. Thank you for your attention.
Thank you, Professor Khan. It was really wonderful talk, and you covered several aspects of different kinds of fibrillar amyloids. And I have one question for you. So I'm also working on one such disease which has these kinds of mutations, Huntington's disease. I am working on Huntington's disease. So it also have Huntington protein which forms aggregates. So I was just curious to know that the lysozyme which you use as model, so whatever effects you have seen by vitamin K and other drugs, can you say that it can be extrapolated for other aggregates also?
Yeah, sure, why not? But the thing is, what is the PI of that protein, the Huntington protein?
It is around 6.5.
How much?
6.5.
6.5. So then it's a wonderful system. You have a choice. You can go below PI as well as above PI. So you'll be very near about the physiological pH also.
Yes.
Because if you are going to take a lysozyme, which is having an isoelectric point is around 11. Yes. So in that case, we are far away from the physiological pH. So in that case, and I'll be happy if you agree for a collaboration, or we can submit some joint project also. So we have a good facility. We have a good facilities. We have all the instrument related to
Anybody else have any question for Professor Khan?
I had just a little query. Very nice talk, Professor Khan. I just wanted to know, how does your concentrations of drugs or vitamins compare to what is usually used in humans.
Nobody has question. So I would like to thank you, Professor
I
Khan,
have a
for
question. I have a
taking
question.
out your valuable time on this
I have a
holiday
question.
and
Ravi.
sharing your research experience, research work of so many years. Thanks a lot.
Ravi, I would like to ask a question.
Yes. Yeah, sure. That was, I think the system was frozen also. Yeah, please go ahead.
I see. Thank you very much for a very interesting talk. I want to ask you one question that when you are working with lysozyme, it is homogeneous. Right? But when you are dealing with aggregates in diseased individual, generally they say ubiquitin and this heat shock proteins, which are chaperones, they are also found to be trapped in the aggregates.
See, this is a commercial preparation that we are taking, and further we are purifying with the column chromatography to ensure that it is a single peak preparation that we are taking for our students.
My question is that will vitamin K work with that condition also? Have you checked? That is what I wanted to ask.
Condition? Which condition?
Where you have other kinds of molecules trapped in the aggregates.
No, we didn't check with under those conditions. It's a homogeneous preparation that we have taken. We have taken homogeneous protein as well as vitamin K, that is the synthetic one, and it's also pure preparation.
Yeah, I was thinking like if that can work even in a diseased individual, then it is much simpler because vitamin K would not cause that much toxicity. If you are using any other drugs, they can give toxic effects. That's the reason why I was asking.
That's a great suggestion. That's a good suggestion.
And Professor Khan, did you compare the concentrations what you have used to what is usually seen in humans?
Yeah, that's an excellent question. That's what I wanted to hear. Because whenever we plan for some experiment, we always take care of what is the drug concentration that is there in the plasma. Otherwise, it will be a futile exercise. So whatever the drug, whatever the compound, whenever you are going to design your experiment, you should always take that parameter into your consideration. What ultimate concentration that will be there in the blood, and then you can go below and above that. So even I can, if I can share it, I don't know whether that data I'm having. Okay, let me find out. It may be there. Can you see the slide?
Yes.
Okay, let me come to the last point. Because deliberately we don't want to discuss all those parameters because sometimes people they don't understand the things. So I don't know whether I'm having those data in this slide or not. Let me find out. So we always take care of these things. What is the concentration in the plasma, and we should be near about that.
Right.
So anyway, I think the slide is not here, but we always take care of that, even because we are working on some anti-Parkinson's drug and others also. So like this L-Dopa and other, this is all data that is being published, and some we are also taking. We have seen the effect of vitamin C also. This is published in archives in 2000, Archives of Biochemistry and Biophysics in 2017. So anyway, but we have taken care of all these parameters. So we are always comparable. Yeah, it's always comparable because otherwise, because river, they always ask how far away you are from the physiological parameters. So otherwise, it will be irrelevant to do this. It's very interesting that this question I was expecting that someone should ask this. Thank you for asking this
Sir,
question.
sir, I was wondering why did you choose specifically that pyrazimide and DCS for that anti-TB drug study and not other drugs?
Now, as I told you in the beginning also, so these were the drugs that initially we were having in our lab. Now we have screened almost 10, 15 drugs. And as you just see on the PubMed, you will find in our lab, they have shown the combination of the drug and individual drug, how they are going to influence the formation of the enzyme. So this is the old story that I am presenting. It's a 2015 and 17. And in between that, we have almost got 30, 40 publication related to this drug role in the amyloid fibril formations.
Okay, sir.
Thank you. So I'll be happy to have a collaboration with you people and your student.
Yeah, sure, sir. We will get back to you.
And your student, they can come to my lab, and we have a very good lab, and we have almost all the spectroscopic taking NMR and CT fluorescence, ITC, DSC, confocal. So
okay.
So everything is there. So I'll be happy, and we can.
Yeah, we can explore the possibility.
Okay.
Yeah, and I see it as a very good opportunity for all of us. Thank you very much, and thank you very much for accepting our invitation and delivering a very good lecture.
Thank you. It's my honor for me. Thank you. Okay, bye.
So I think our next speaker is Puran Singh Sejwali. Dr. Puran has done his master's at GB Pant University, a place where I also wanted to go for my MSc, landed up at some other place. And then he did his PhD at ICGB with the group of Dr. Chauhan and Pawan Malhotra. Later on, he went on to do his postdoc at San Francisco, where he spent almost a decade. And during all this period, during his PhD and postdoc, he has been working on malarial parasites and different kinds of aspects. Of particular importance, or the most publications show that he is very much interested in the proteases. And for them, he has done structural biology, drug discovery, and small molecule inhibitors. And today we will learn more about these proteases from him, and also the involvement of proteasomal degradation system in this regard. Dr. Puran.
Yeah, thank you, Professor Sanjeev. And I'm trying to share my screen with you.
Yeah.
And let's see if it works. So can you see it?
Not yet. Looks like something coming up.
Okay.
I think you need to pull up the slides on the screen.
Yeah.
Yes. You are there.
Yeah. So you can see now.
Yes.
Yes. Very clear.
Great. Thank you very much, Sanjeev, for a nice introduction. And also thanks a lot to organizers for organizing such a wonderful meeting, particularly during these COVID days. It has been a long time. I think people are attending meetings. And so what I'm going to talk about it, so the previous speakers, both yesterday and today, talked about, there are a few lectures on protein degradation. And what I'm going to touch upon is the protein degradation machineries, and in the context of their roles in primitive eukaryotes. And the primitive eukaryote I use in my lab is the Plasmodium, which causes malaria in humans as well as cellular vertebrates. So the title of my talk is The Roles of Ubiquitin Proteasome System and Autophagy Pathways in Malaria Parasites. So this is for the benefit of students. I would like to go through brief history of protein degradation systems, and I apologize for rest of the audience who might be well aware of the history and know how these degradation machines work. So protein turnover, let me use my pointer. Okay, great. So protein turnover is a continuous process of protein degradation into amino acids and resynthesis of proteins from amino acids. So the concept of protein turnover in the body was demonstrated in 1930s, mostly from the work of Sponheimer, who used radio-labeled amino acid in rats and then monitored incorporation of the radio-labeled amino acids with the time. What we found that in the tissues from different parts of the rats, there was increased amount of radio-labeled amino acid in the beginning, which went on decreasing as the time passed, and that became available as a free amino acid food. So he concluded that proteins are not stable. They undergo a process of turnover. And every protein in vivo has a distinct half-life, broadly ranging from a few minutes to more than 100 days. And I'm sure students know that there are proteins like cyclins, which are there for only few minutes. But then there are protein, I think the previous speaker, Professor Khan, was talking about like albumins, which are quite stable in the body. However, most intracellular proteins were believed to be long-lived until 1980s. And the one difference I would like to point out here between protein degradation during food digestion and the intracellular protein degradation is that the protein degradation in cells is very specific and precisely regulated. So the question arises, why degrade proteins? So the first reason is to maintain quality control, because accumulation of non-functional protein can cause cytotoxicity. Like, for example, mutations do happen, misincorporation of amino acids, and non-native folding and post-translational damage. In fact, I would like to point out here that almost 1/3 of the proteins that are synthesized on ER never acquire their native fold, hence are degraded before their synthesis is completed. So that's a lot of degradation in cells. And then there are several proteins which have regulatory functions, and these proteins need to be degraded. Such proteins like transcription activators, repressor, the under category of protein is cyclin, because if these proteins accumulate in the cells after their jobs are done, that can have adverse effects. And then to maintain amino acid pools in cells during stresses such as starvation, dispensable proteins or dispensable cellular contents are degraded as well. That's what I'm going to talk about. By the way, can you hear me?
Yes, very well.
Okay, great. Thank you. So the two major mechanisms of protein degradation in eukaryotes are ubiquitin proteasome system and autophagy. Autophagy pathway is not present in prokaryotes and archaea. However, a functionally equivalent degradation pathway like UPS is present in archaea. And then, of course, the prokaryotes, other prokaryotes like bacteria have functionally similar machineries to degrade proteins. Like, for example, CLIF-AS is the protein degradation machinery in E. coli and other bacteria. So the discovery of ubiquitin proteasome system was done together in parallel by these three scientists, Arvind Roche, Avram Hershko, and Aaron Sisnover. And they were awarded 2004 Nobel Prize in Chemistry for the discovery of ubiquitin proteasome system degradation. And this is what they proposed in 1980s. That what they observed that, so they worked on also used lysozyme and used reticulocytes. What they found that there is a protein which is called ATP-dependent proteolysis factor or APF. So multiple molecules of the APF are conjugated to a protein, so which is shown by APF, and then this APF protein conjugate undergo degradation, that is step number 3, releasing free ATF, but probably some part of the protein which they have shown as X, and then free amino acids. But they also proposed at the time that, so this conjugate, okay, can be recycled into protein and then free ATF. I will briefly tell about that. And then from here, this ATFX is recycled for further degradation process. This is what they proposed when we did not know much about ubiquitin proteome system, but the model holds true even today. Only thing which has changed is the complexity of the model. So this is a schematic I'm using from East. So this is the modern day picture of ubiquitin proteome system in East. So ubiquitin, so the two hallmarks of ubiquitin proteome degradation are, one is ubiquitin, other is the proteasome. So ubiquitin is the one which flags a protein to be degraded, and proteasome is the component that degrades the ubiquitinated protein. In between, there are multiple enzymes which are involved in the pathway. Okay, so in Saccharomyces cerevisiae, ubiquitin is produced either as a multimer of 5 ubiquitins or as a fusion protein with ribosomal protein. And then from these fusion proteins, deubiquitinases cleave these two and release free ubiquitin. The free ubiquitin is activated by ubiquitin activating enzyme E1 in an ATP dependent manner, and from E1 it gets transported to ubiquitin conjugating enzyme E2, from where it gets transported to the substrate by one of the two E3 ligases. So one E3 ligase is a multi-protein complex, which is known as ring finger-like E3 ligase, which I'll be talking about. The other E3 ligase is a single protein, okay, and their mechanism of transfer is different. While the ring finger ubiquitin ligase accepts the E2 as part of the complex and then facilitates transfer of this ubiquitin to the substrate. Whereas the HECT type ubiquitin E3 ligase receives ubiquitin from E2 to itself and then transfer to the substrate. And then the ubiquitin substrate is recruited to the proteasome, where ubiquitin chain is recycled and the substrate is degraded into peptides and then subsequently to amino acids. And this also involves ATP. So the ATF I was talking about in that model, which the Nobel laureates proposed, is this ubiquitin. And of course, it requires energy here for activation as well as for degradation here. And then in between, there are multiple enzymes. What I showed you here, that this ubiquitinated substrate from here can be directly converted free into the substrate and ubiquitin by deubiquitinases, which are called Depts. Okay. So my work is mainly about the ring finger-like E3 ligases, which are also called Cullen ring ubiquitin E3 ligases or CRLs. There are two archetypical CRLs. One is SCF and other is APC. So these two are involved in regulation of cell cycle, which I'll be talking about later on. So we started working on these degradation pathways in malaria parasites, and this is a schematic of malaria parasite, which you can see that the parasite has a complex life cycle. The infection in humans or vertebrates begins when an infected mosquito bites to get blood. So when the mosquito bites, it injects saliva at the site of bite. So along with the saliva, the sporozoite stage of the parasite is injected. The sporozoite stage goes all the way to liver and infects hepatocytes, where it undergoes development through multiple stages, forming thousands of daughters, which are called merozoites. So these merozoites cannot infect hepatocytes again. So what they do is they infect red blood cells. Once they infect the red blood cells, they develop further through stages which look like ring, trophozoites, and the multinuclear stage Saigon. So Saigon ruptures, releasing these merozoites, and which can reinfect a fresh erythrocyte. And this development, which is known as erythrocyte cycle, continues, is responsible for the symptoms of malaria. whereas the development in hepatocytes does not have any symptoms. Okay. So during this development, some merozoites commit to gametocytogenesis, and then they don't undergo this kind of development. So from here, they develop into gametocytes, male and female. And these gametocytes can no longer develop in the human host or vertebrate host, while in the blood, they wait for mosquito for bite so that the blood can be taken. In the mosquito midgut, where there will be change in pH and temperature, these gametocytes differentiate into male and female, which fuse forming the zygote. Zygote undergoes differentiation forming oocytes, which becomes oocyst, which undergoes further multiplication forming thousands of sporozoites, which are then transmitted into a host by mosquito. So now, so the Plasmodium genome is about 22 MB, coding for about 5,500 proteins. And many of these proteins have stage-specific expression. Not all proteins are expressed in all the stages. That's number one. Which would require elimination of unwanted proteins at the prompt of the parasites differentiate in the next stage. Okay. Other thing is the infective stages like sporozoites here and the merozoites have organelles which are only required for invasion, and these invasion organelles are lost as soon as parasites differentiate into the next stage. So that would also require degradation. The third stage, which is prophozoites. Prophozoites, remember I told you this is the exocytic stage in which the parasite develops in a red blood cell. So while it develops in RBCs, it takes up about 80% of the RBC cytosol, which is primarily hemoglobin, transports that to its lysosome and degrades it. So this is the third process which would require degradation. That's why we got interested in degradation pathways like UPS, the ubiquitin proteome system, and autophagy in malaria parasites. Okay, and while these pathways are very well studied in higher organisms like mammalian cells and yeast, so when we started, we did not know whether these pathways existed in Plasmodium, although the genome data was available. That's where we started, and this I'm talking about in about 2008, 2010. So the major research activities of my lab based on the background I gave you in the last slide are functions and regulation of ubiquitin E3 ligases. So here we are focusing on the ubiquitin E3 ligases which likely participate during Plasmodium multiplication. The autophagy pathway and its roles in malaria parasite development. And we are also interested in drug and vaccine target discovery, which mostly we do in collaboration with medicinal chemists and immunologists. So now, so coming to the ubiquitin E3 ligases, so the reason we are interested in it, that when you look at the Plasmodium cell division, so most of the pathogens have enormous capacity to multiply. And this enormous capacity to multiply is responsible for many symptoms of the disease, but also ensures that the pathogen is transmitted from one host to another host. And so is true for Plasmodium, that it undergoes multiplication during 4 stages during its development. So in hepatocytes, when it develops, it can form up to 10,000 daughters. So a single parasite can produce 10,000 daughters. There are some reports which say 30,000. In erythrocyte, one parasite can form up to 30 daughters. One male gametocyte can form up to female gametocytes. A oocyst can form up to 1,000 sporozoites. And particularly in this stage, the ability of parasites to produce multiple daughters which infect fresh RBCs is responsible for severe symptoms of malaria like severe anemia, okay, which causes lysis of infected parasite. That's number one, why we are interested in. Number two, unlike a typical cell division in which chromosomes replicate and the daughter chromatids are assembled and then pulled apart into opposite poles, leading to division of the nucleus, followed by division of the cytoplasm and cell membrane. Okay. So in case of Plasmodium, the cell division is very, very atypical. Like the nucleus divides forming two daughter nuclei, okay, which are pulled apart forming two daughter nuclei. And now, but there is no cytokinesis. Okay, so the division doesn't stop at this time. So what happens now? Any one of these two nuclei or both the nuclei undergo further nuclear division within the same nucleus, forming a syncytium or nuclei. Finally, when the parasite has produced the required number of nuclei, which can be anywhere from 8 to 32 or 30, the cytoplasm and part of the membrane assembles around the daughter nuclei, forming a new parasite. So which makes it very atypical. And from the pathogen point of view, so most of the time we work on pathogens so that we can develop new treatments, new vaccines, all that. So this atypical nature of Plasmodium cell division provides opportunity to identify targets for development of drugs or vaccines. That's the second reason. And third reason is many canonical factors which regulate the chromosome separation. Like, so whatever the daughter chromosomes are held together by a protein complex called cohesion until rest of the formalities are taken care of. Finally, ubiquitin E3 ligases like APC in this case and a protease kind separase, they cleave the cohesion separating the daughter chromatids. Okay, and this process involves many proteins and protein complexes. Some of them are like ubiquitin E3 ligases, protease separase and its inhibitor securin, cyclin B, CDK, and DNA and several DNA-dependent proteases. But however, when we do bioinformatics search of plasmodium genome, we don't find many of these. For example, this APC, which is the key regulator of this chromatid separation, is not there. SCF is there. I will talk about that in our data. Saffrase is there, but securin is not there. Plasmodium doesn't have typical cyclins, and CDK1 is not there. So that makes it interesting from the parasite biology point of view to investigate the pathway. So now I'm going to focus on the Cullen ring ubiquitin E3 ligases, which we think are involved in multiplication of Plasmodium. So Cullen ring ubiquitin E3 ligases form the largest group of E3 ligases. So I told you that in one of my in the schematic of ubiquitin system that E3 ligases are the one which define substrate specificity for degradation. Therefore, the number of E3 ligases in mammalian cells is about 1,000. Okay, and which takes care of and which can take care of over 20,000 proteins. Although that's a kind of discussion for a different day. And most of the E3 ubiquitin ligases in those thousands belong to this group CRLs. All CRLs have a common subunit, which is Cullen. And Cullen protein is evolutionarily conjured in eukaryotes, although the number of Cullens are different. For example, in humans have 7 different Cullens, and Plasmodium has only 2. And when we compare their amino acid sequence, so we get this schematic that all these have a Cullen homology region, which is present in Homo sapiens Cullen 1, also common to Plasmodium Cullen 1 and Cullen 2. And at the C terminus of the Cullen, there is a motif which is called nadylation motif. Here it is indicated as Na. This is a typical composition of a CRL. So Cullen, which form the platform for assembly of the complex. So at the N terminus of the complex, it serves as a platform for adapter protein, which recruits substrate receptor and which now recruits the substrate. At the C terminus, it serves as a platform for ring finger protein, which recruits E2, and then from E2, the ubiquitin is transferred to the substrate. Yeah. So the question we asked, does Plasmodium have CRLs? If yes, what are the roles of these CRLs? So Jay Maharaja has been a PhD student in the lab who has been addressing these questions. So approach is, so using in silico analysis, in which we use the sequences of the Cullen subunits of humans and cerevisiae and search the Plasmodium genome to identify homologs. And we could come up with some homologs. For example, we could predict choline, we could predict ring, and we could also predict adapter. And these we made recombinant parasites expressing these proteins, and these parasites we used for immunoprecipitation followed by mass spectrometry, and we produce the relevant hits as recombinant proteins and assess for direct interaction. And then finally, we tried to assemble the complex for in vitro activities. To identify roles, we generated knockout or knockdown of desired CRL subunits and assess for doublement at various stages to investigate the mechanism. Just a minute. So this slide is about a parasite expressing choline-1. So in this case, we replaced wild-type choline-1 gene with its allele that contains choline-1 sequence, which is fused in frame gene GFP, and this will be the integration locus or recombination locus. So this is a PCR gel. This is an agar gel of PCR products amplified from genomic lineage of wild-type and knock-in parasites. Here you can see that the knock-in parasites have recombinant locus and don't have the wild type, whereas the wild type have only wild type locus, and this is a positive control. This is a Western blot showing expression of this GFP choline-1 in different stages of the erythrocytic parasite. The R is for ring. This is a late trophozoite cyzone, and this is a live cell microscopy showing expression of choline-1 GFP in the parasites. So this is GFP signal. This is nuclear signal Hux. This is bright field. Okay, this is RBC boundary, and inside this we have parasites, and where this region of the parasite is the one which marks the lysosome. I'll talk about it, and this is merged. And similarly, she expressed the RBX protein with a MIG tag, and this is Western blot again showing expression of RBX MIG. This is immunofluorescence assay showing localization of RBX MESP. And she also expressed SKB1 fused to GFP. And this is a Western blot showing expression of SKB1 GFP. And this is live cell images of SKB1 GFP parasite, where you can see SKB1 GFP signal, which is present all over the parasite. Okay, so now she immunoprecipitated the choline GFP, RBX MIG, and SKB1 GFP. Did mass spectrometry of the immunoprecipitate and then analyze the hits. And this experiment is repeated at least 3 times. So here is, I'm showing you the table of some of the hits which are relevant from the CRL complex point of view. So in this case, our bait protein is choline. We find choline, we also get NAD8. Here our bait protein RBX, we get RBX1. We also get our choline1 and NAD8. Here our bait protein SKP1, we get choline1, abbox protein, and calcyclin binding protein. So this, and I'm saying these are relevant, so that is relevant from the known architecture of choline ring ubiquitin E3 ligases in yeast and mammalian cells. So based on the IP followed by mass spec, so we have this kind of model. So we have plasmodium calcium choline1, which interacts with RBX, gets nadilated as well. This is the black bar. On its N terminus, it interacts with SKP1, which interacts with abbox protein, which is a substrate adapter, and under calcyclin biprotein. So this model is based on immunoprecipitation. So remember, immunoprecipitation tells you what is part of the immunoprecipitate. Immunoprecipitation doesn't tell us one-to-one interaction. So to validate this model, one has to do one-to-one interaction, which one can do using yeast-2 hybrid, which I believe most of the students know, or one can also do using recombinant proteins. Okay, and you produce all your proteins of interest in recombinant form in E. coli or mammalian system, and then assess their interaction by various ways. One of the way we used here, and this is what is here is. So JBA went for in vitro validation of the interaction. So she used a procedure which is called dot blot overlay assay. So this is a modified Western blot. So in Western blot, we run our proteins in SDS page, then transfer those proteins to a nitrocellulose PVDM membrane. We block it, and then we incubate with antibodies to our target protein, followed by secondary antibody. But in this case, there is no SDS page required. We spot our protein on a nitrocellulose membrane. We block it. Then on the membrane, we overlay our test protein. Okay. Then we wash it, and then we use antibodies against our test protein to see whether signal is there or not. So this is what is shown here. So these are different strips which contain spots of PF choline 1 N-terminus, this part here, box protein, calcyclin binding protein. So GST, because these three proteins were used as GST, were made as GST fusion proteins and followed by RBX. And now these strips were overlaid with histidine tagged SKP1. And here we have used increasing amount. So what you see, it's nice signal with PF choline 1 N-terminus with box protein and calcyclin binding protein. No signal with GST and RBX. So which means the SKP1 directly interacts with choline 1 with box and calcyclin binding protein. This is our model, which is validated. In this case, similar assay, what we have done, we have spotted PF choline C-terminus here. So PF choline is about 850 amino acid long protein. So we couldn't make the full length protein in recombinant form. So we have used two different domains. Here we have spotted GST1, calcyclin binding protein, box protein, and then these strips were overlaid with histidine, SUMO tagged RBX. So SUMO is a fusion protein like maltose binding protein or thyroxine protein or GST protein, which is used in E. coli to enhance solubility of your protein. So what we have used here as well. So what you see here is the RBX gave nice signal with PF choline C-terminus, but not with GST, not with calcyclin, and not with box. So validating interaction that RBX specifically interacts with the C-terminus of here, which is true for choline ubiquitin E3 ligases. So as a control, we have spotted here SUMO and then overlaid with PF choline 1 C-terminus and GST. Also, we don't see any signal. So this validates our model that Plasmodium choline 1 forms a ubiquitin E3 ligase complex like the SCF ubiquitin E3 ligase of yeast and mammalian cells, in which through its N-terminus, it recruits PF-SKP1, which then binds to calcyclin B and box proteins. The box protein would be recruiting a substrate, which we don't know, we are working on it. The C-terminus PF choline 1 recruits PF-RBX, which then recruits E2 and from where the ubiquitin will be transported to substrate, and we don't know that as well. So this is working. So what we need to do ahead is to identify substrate of SCF, to investigate its role during parasite development. So our hypothesis is, since Plasmodium does not have an anaphase promoting complex, so SCF of Plasmodium likely regulates the multiplication. This is what our hypothesis is. And then how this SCF is regulated, which I will briefly talk about. So the next, similarly, JEMA expressed PF choline 2 in Plasmodium falciparum. So here she replaced PF choline 2 wild type gene with PF choline 2 fused to CDD in HA. I will come back to this CDD later. So, but let's think about that. It is the HA type PF choline 2, and the recombinant locus. Okay, she confirmed these parasite that these are indeed recombinants, which you can see here that the knock-in parasites have the recombinant locus and do not contain wild type. This is a positive control. This is a Western blot showing expression of PF choline 2 CDD-HA fusion protein, which is present in all stages, predominantly around the stages of 20 hours, 30 hours, 40 hours. So, okay. So I must have told you that, or I should have told you that Plasmodium exocytic life cycle is 48 hours. And the metabolically active phase is this T20 to T30, and it is T30 to T40 stage where it undergoes multiplication. So increased amount of a protein may indicate roles of a protein in during multiplication, which we are looking for. This is just the IFA showing localization of PF colline 2. And similarly, see immunoprecipitated PF colline 2 from the parasites using anti-HA proteins, and this is a Western blot showing that the immunoprecipitate has the full length colline here, colline 2, whereas the control immunoprecipitate from wild type parasite does not have any signal. And the immunoprecipitate see also subject to mass spectrometry. And I'm showing you some of the hits which we consistently identified in multiple IPs. So what we identify is colline-like protein, which is now we have renamed colline 2. Of course, it's our bait as well. And the protein CPSF, which is a substrate adapter, is also known as damaged DNA binding protein complex. We got RBX, we got NAD8. And this is the model we have. This is PF colline 2. At N-terminus, most likely it recruits this DDB, which is a substrate adapter, although we haven't confirmed it. And then we added when we got WD repeat containing proteins. So WD repeat containing proteins are involved in protein-protein interaction. So these could be substrate adapters, and there is also one autophagy protein, ATG18, which is a beta propeller protein, which is also involved in protein-protein interaction. We don't know what is a substrate yet. And then through its C-terminal, it recruits RBX1, which would recruit E2, which we don't know. So we have a model for PF colline 2. So the IP followed by MS data of PF colline 1 and colline 2 indicates that Plasmodium has a SCF and colline 4 like E3. So why I'm saying colline 4? Because the human colline 4 ubiquitin E3 CRL has this kind of complex. So that's why I'm saying that it is a CRL4 like ubiquitin E3 ligase. So you might have noticed that in most of my IPs we are also getting NAD8. So NAD8 is a ubiquitin like protein which gets conjugated using the machinery similar to ubiquitin, but it gets conjugated to specific protein, and those specific proteins are colline ring ubiquitin E3 ligases. So in our IPs we found NAD8 consistently, but we wanted to confirm it whether this NAD8 is coming from conjugated colline or it's a part of the complex. So Manish is another PhD student who has been working on this nadylation pathway. What he did, he expressed HA tag NAD8 in Plasmodium falciparum, and this is the Western blot showing that the wild type parasites don't have, whereas the recombinant parasites have, this is free NAD8 and then 2 high molecular weight bands, okay, and which are conjugated to NAD8, because this is a Western blot of SDSP gel. So these proteins, and we wanted to identify. So what Naveen did immunoprecipitating using anti-HA antibodies. This is the elevate or immunoprecipitate. Did not get these 2 bands in a control immunoprecipitate from wild type parasites. Subjected the immunoprecipitate to mass spectrometry and identified that indeed there was NAD8, which is bait. Also there was colline 1 and colline 2. So confirming that the Plasmodium colline 1 and colline 2 undergo nadylation. So this is the model we have now. So for colline 1, that it also undergo nadylation. Okay. So next we wanted to check what are the functions of these 2 CRLs during parasite development. So we haven't completed the CRL work. So I will be showing you some data for the roles of colline 2 in parasite development. So this is the schematic I showed you, that there are 3 different developmental cycles: the liver cycle, the blood stage cycle, and the insect cycle. So in falciparum, you can investigate the erythrocytic stage cycle because this is the one which is cultured in most of the laboratories, and this is you can culture it continuously. But then there are some procedures you can force parasites to differentiate into gametocytes. And then you can see gametocytogenesis, which undergo through 5 different stages, okay, and which based on morphology and if somebody who knows the parasite can differentiate. So we investigated the role of PF colline 2 in blood stage as well as in the gametocytogenesis. So I'll briefly show you our experimental approach and the final data. So to identify the role of Plasmodium colline 2, what Jeva did, she replaced Plasmodium colline 2 with its fusion with CDD and the HA. So CDD is a E. coli DHFR. It's a mutant E. coli DHFR which acquires or which has stable conformation, and it is bound to its ligand that is TMP or trimethoprim. So it is a stable protein. Therefore, the whole fusion protein is stable. However, if TMP is not there, it acquires unstable conformation, which is rapidly degraded by the ubiquitin proteasome system. So by adding TMP or removing TMP, you can induce knockdown of the protein of your interest. Thereby, you can investigate the effect of the decrease in the protein levels on the parasite development or in any cell. So what I'm showing you is the Western blot of parasites grown in the presence of TMP, which is plus T, or in the absence of TMP. And this is a control. What you see is that compared to the parasite grown in the presence of TMP, the levels of colline 2, this is colline 2, are significantly lower. Now, then you set up 2 cultures. In one culture, you add TMP. In other culture, you don't add TMP for the same parasites. And then you monitor parasite growth for several hours, whatever you want. So Plasmodium cycle is, extracellular cycle is 48 hours. So we have followed these parasites for 48, 96, 144, and 192. So what you see is the orange bar here is for the parasites grown in the presence of TMP, and the blue is for the parasite grown in the absence of TMP. So what you see is that when the colline 2 is knocked down, the parasite growth is significantly decreased, indicating that it is important for parasite development. Similarly, we counted the number of gametocytes cultures grown in TMP for sinus TMP. The here for TMP and we see that 90 gametocyte development. So we haven't inspected the university yet, so we might go there and check. So just don't stand here with me. So what happens to each parasite growth here? Like the is that Plasmodium has so unlike ubiquitin proteasome system, which measures in the cytoplasm. Okay, all of these two lysosome-dependent and also hydrolytic organelle because it contains phases or hydrolysis and its pH is around 5 compared to the cytosolic pH of 4. The organ for lytic function was discovered in 1955 by Christian DOA. I'm sure that I remember this. And he also defined the mode of delivery of cytoplasmic materials to lysosomes, autophagy, which is derived from the word meaning for self-eating. So what was also observed that double membrane structures containing a portion of cytoplasm and organelles without heterolytic enzymes known as autophagosomes were also found in lysosomes. Majorly, autophagy is studied as a cell survival mechanism during nutrient deprivation, and unlike the ubiquitin proteome system, which targets mostly soluble proteins, a smaller protein, the autophagy can target and degrades long-lived proteins, large proteins, as well as cellular contents like organelles. Okay. So although it was observed long ago, but the discovery of autophagy machinery was primarily done by the lab of Yoshinori Osumi, who used Saccharomyces cerevisiae as an organism. He observed that when the yeast cells are starved, they accumulate autophagic body in the yeast vacuole, which is equivalent to the mammalian lysosome. And this is his experiment. So he carried out random transposition mutagenesis, okay, and monitored monitored yeast strains for appearance of these autophagic bodies in the vacuole. So this is a yeast, okay, which put into starvation. This is how the structure looks at 0 minute. It has very clear vacuole. After 3 hours, the vacuole is full of autophagic bodies. Okay, and this is a slightly magnified version here. And using this mutagenesis approach, he identified over 10 genes which are involved in the autophagy pathway. And for this work, he was awarded a Nobel Prize in 2016. Okay, so now there are 35 to 40 different autophagy proteins which participate in this pathway. So like ubiquitin proteome system, autophagy is a complex pathway. It begins with formation of a cup-like structure called phagophore, sequestering the cargo, and the cargo can be ribosomes, cargo can be endoplasmic reticulum, mitochondria, protein aggregates, or protein complexes. Okay, this cup-like structure is expanded into a double membrane vesicle, which is called autophagosomes, that fuses with lysosome. Okay, and then finally the cargo is degraded and the contents are released into cytoplasm for reuse. So these 35 to 40 autophagy proteins participate at different stages of this pathway. One of the protein which is widely used to study the pathways, ATG8, which is shown here as red dot. So this ATG8 gets conjugated to this cup-like structures in the beginning and stays with the cargo and when it fuses with the lysosome. And this is, as I told you, is a double membrane structure. The ATG8 present on the outer membrane is recycled, whereas the one present in the inner membrane is degraded to the cargo. Then there are several treatments one can use to study autophagy. For example, this rapamycin. So rapamycin is an inhibitor of mTOR. mTOR is a negative regulator of autophagy. If you grow your cells in rapamycin, autophagy is induced. Similarly, the universal inducer of autophagy is starvation. So during starvation, autophagy is induced, and which transports dispensable cellular contents, including proteins and membrane organelles, into the lysosome for degradation, and the amino acids are used to meet the basic survival demand of the cell. 3-MA is an inhibitor of PI3 kinase. It inhibits autophagy. Similarly, there are some compounds which interfere with lysosome function, and they can also be used to study autophagy because if cells are grown in protease inhibitors like E64 and peptistatin and chloroquine, which raises the pH of the lysosome, the cargo is not degraded. So increase in the level of the cargo would indicate that autophagy is going on. Okay. So what was our reason to study autophagy in malaria parasites? So when we started, autophagy was not known in the parasites, but being a primitive eukaryote, we did think that autophagy pathway is likely to be there. But one of our major reasons to study this pathway was that I told you that while its development in the red blood cell, this is a red blood cell and this is a parasite. And this part, which is white and contains this black pigment in the parasite lysosome, the parasite degrades up to 80% of the RBC cytoplasm in this compartment, which is called food vacuole. So since the majority of the cytoplasm is the hemoglobin, it is packed in vesicles transported to the food vacuole where it gets degraded. So this hemoglobin degradation releases heme, which would be toxic for the parasite. Therefore, it polymerizes heme into this black pigment called hemojoin, which you can also see here, right? And then the peptides are further degraded into amino acids. So chloroquine, the most popular anti-malarial until recently, inhibits the polymerization of heme into hemojoin, which causes toxicity to the parasite. Since this process is specific for the parasite, therefore chloroquine is very effective and safe for treatment of malaria. So we thought, as it is a bulk degradation process, that this might be carried out by a pathway like autophagy. That was the major reason to start working on this pathway. So Rahul was a PhD student in 2009-2010. So he asked this question: does Plasmodium have autophagy pathway? If yes, what are the functions of autophagy during Plasmodium development? How and where do autophagosome form? And what do they contain? So again, so the availability of genome database makes life much easier these days most of the time, but also it complicates life as well if you have large number of data. But our problem was very simple. All we wanted to know whether parasite has an autophagy pathway. So what Rahul did, so Rahul downloaded the sequences of the known autophagy gene proteins of mammalian cells and yeast, and did blast search of a Plasmodium genome database to identify homologs. So he could identify 15 homologs for homologs of ADG proteins, which are here, and the ones in the underlined with this red line are absent, or we could not identify based on your search. But we could also identify ATG8, which is also known as LC3 or light chain protein 3 in mammalian cells. But we could not identify that a typical homolog of TOR, this is the major negative regulator of autophagy in mammalian cells. So Rahul used ATG8 as a marker to study autophagy in malaria parasites. So he generated ATG8 antibodies, did several experiments that is published, and this is what I'm showing a summary of his work, and then work of several other labs which started working on autophagy around the same time and published. And it's nice to see that most of the findings of these reports were consistent and agree that Plasmodium has a limited number of autophagy genes, which is about 15, which I told you. And this is a localization of ATG8 in malaria parasites. This is the blood distributed Plasmodium falciparum, but the green signal is for ATG8 and the blue signal is for DNA. So what you see is that the ATG8 stains punctate structures all over the parasite, and this black area is the parasite food vacuole, and the black pigment is the lysosome. Okay, what you however see here is that none of the green signals present in the food vacuole, which is equal to plasmodium lysosome. So based on that, we concluded that it does not appear to perform the canonical lysosome dependent degradation process. Although this is still not clear whether it has a role in degradation of hemoglobin, which was our first region to embark on studying this pathway in Plasmodium. The other feature of Plasmodium autophagy is starvation downregulates ATG8 levels. So starvation is the universal inducer of autophagy in all the organisms studied so far except Plasmodium. And autophagy in Plasmodium is also not affected by typically used autophagy inhibitors. This is a Western blot of parasites, control parasites, parasites which were starved, parasites treated with protease inhibitors like E6/4 papstadin, 3-MA inhibitor of PI3K, and chloroquine. So normally, if we focus here, what would happen in mammalian cells if I had used yeast, you would see higher level of ATG8 compared to the control. However, we observed much lower level. So which was in contrast to what we know about starvation-induced autophagy in other organisms. So that's why we say that plasmodium autophagy is atypical. So the next question which we asked, how and where do autophagosome form and what do they contain? So this work was done by under PhD student Renu Sudhakar, who happens to be your master's students. I'm happy to have one student from here, although there are many students from MS Baroda and CCME doing PhD, very bright students. So this is Renu's PhD work. She has completed her PhD. So to address this question, she used under autophagy proteins called ATG18. So ATG18 is required for formation of autophagosome in the beginning. So she performed co-localization study of GFP tagged ATG18 with anti-ATG8 antibodies and also use other antibodies. And what she showed that ATG18 co-localizes with the Plasmodium lysosome membrane here. And this co-localization is very specific because when she mutated some of the motifs, okay, the localization became completely cytoplasmic. Okay. And then what she also showed that this co-localization requires interaction of ATG18 with phosphatidyl inositol 3-phosphate as well as MDR1. So MDR1 is a multi-drug resistance transporter in the membrane of the food vacuole, which confer resistance to several antimalarials. Okay. And if we mutate ATG18 that interacts with MDR1, we lose this localization as well. Then using co-localization ATG8, what she showed that most likely autophagosome form in a region in this food vacuole membrane, which we call perivacuole membrane region, although we don't have more additional data to support this, but so far based on the data we have, we think that autophagosomes likely form at the peri-food vacuole region, and this work is going on. So my last slide. However, although autophagy is atypical in Plasmodium, it does not seem to perform on the major degradation work, but it is essential for parasite development. So what we have done here, in this case, we use two autophagy genes, ATG18 and ATG5. Both are core autophagy genes in yeast and mammalian cells. We replace wild type genes with the mutant E. coli DHFR fusion, so that when we grow these parasites with TMP, the fusion protein is stable, it can perform its function. When we remove TMP, the fusion protein would become unstable, causing knockdown of the probe. Degradation would get degraded, thereby causing knockdown. So this experiment was done using Plasmodium burgii, which causes malaria in rodents. So here we have used Balbsi mice and Plasmodium burgii. So the Plasmodium burgii and Plasmodium falciparum have similar genome size, and most of the genes are highly similar. In this case, for example, the ATG18 and the 5 are more than 90% identical between these two species. So what we did, we infected mice with wild type and knockdown parasites. And the mice infected with knockdown parasites were given trimethoprim or left without trimethoprim. And then we monitored their growth for several days. So the plot here shows the growth curve of ATG18 knockdown parasite with or without TMP. So the black line is for wild type, the orange is for knockdown parasite with TMP, and the blue with knockdown parasite without TMP. So what you see is that while the wild type as well as the parasite with TMP grow normally. And finally, at this stage, the parasite high to mouse, so the mice had to be euthanized, whereas the mice infected with knockdown parasite did develop very little parasitemia, and finally this parasitemia became invisible. So the mice got self-cured, indicating that ATG18 is essential for parasite development. Same is true for ATG5 as well here. So the blue is the mice infected with ATG5 knockdown without TMP. So the mice barely developed in parasitemia and eventually the parasite disappeared, whereas these mice had to be euthanized. Okay, so the conclusion here is the knockdown of ATG18 and 5 drastically reduced parasite growth, and therefore autophagy pathway is essential for parasite development, although it is atypical. So in summary, what I shared with you is that Plasmodium has an atypical autophagy pathway. Autophagosomes may form at the peri-food vacuole region. Autophagy is essential for parasite development. Oh, sorry, it has been right. So we are still investigating what are the specific rules of autophagy during parasites development, how autophagosome forms, and what is the cargo. If the autophagosomes don't end up in the lysosome, what is the cargo they contain? Or the punctate structures which we think are autophagosomes, it is possible that they are not autophagosome. They could be some other structures. So more work is going on both the pathways, and hopefully in future I will have opportunity to share these with you. So with this, I would like to thank my group, particularly the people whose work I showed you, and also the people whose work I could not show, and hopefully in future I'll show you. So this is Renu here, who did the ATG18 part. So Rahul has graduated some 4 or 5 years ago, is not in this picture. Jeba is this, who has been working on the Cullen ring ubiquitin E3 ligases. And who else is here? Okay, Manish is the person, PhD student who worked on adolescent pathway here, whose work I showed you. And I would also like to thank the funding agencies, CSR, DBT, and DST. With this, I thank you all. So now I'm waiting for questions.
Yeah, we are open for questions. Yeah. Anybody who wants to ask,
yeah,
can please.
Thank you for your very interesting talk. I want to know if all the cells, means parasites, they develop in a synchronous manner, because when you are analyzing the bands, unless all of them are in synchrony, you would be getting a mixture of bands, right?
A very important question. Yeah. So what we do it? So in culture, if we grow them continuously for several generations, they lose synchrony. Okay. So, and that's why what we do, so there is a treatment. We do a treatment in which the cells we treat with 5% sorbitol. So that 5% sorbitol kills all the stages except ring stages. Okay. So the reason for that is it is just osmotic lysis of zone, whereas ring stays intact. And if we repeat that treatment for 2 cycles, so we can get very synchronous parasites. And we do that every time we plan to experiment. Yeah, that's a very important question. Yeah, that asynchrony can give you false impression that your protein expression levels are different at different time points.
Yes. Yeah.
Yeah.
Thank you.
Sure.
Anybody else has a question? Please come forward and ask the questions.
I was expecting students to ask some question. If I don't know if they are attending, I hope they are attending.
They are attending in YouTube actually.
Oh,
because
okay.
this has a limitation of numbers. That is why you see very small number of people here. It is only faculty members, and early morning lectures and evening lectures are also joined by people from abroad who are either our alumni or people who are interested. So you concentrate only on one of the plasmodium, means you have this plasmodium vivax, falciparum, all these.
So what we do is we focus on Plasmodium falciparum because that is the one can culture in vitro. Vivax is still very challenging to culture. So if somebody wants to work on Vivax, you need to get samples from patients. So and we can't do these kind of experiments with samples taken from patients. So most of our in vitro studies are on Plasmodium falciparum, and for animal studies we use Plasmodium burgei, which infects rodents, mice and rats.
Okay.
And the other advantage of Plasmodium burgei is then we can take our investigation to insect stage, liver stage, so we can complete the cycle. So that we do in collaboration with Dr. Kota Arun Kumar, who is a faculty at University of Hyderabad. So not very far from CCMB. Yeah.
I have one question.
Sure.
Yeah, this mainly for the RBC that may be more problematic. Whatever compounds we are trying to use for anti-malarial thing, they have to cross RBC membrane and then cross the plasmodium membrane. Then they can act on that, no?
Yeah.
So what are the limiting factors for the transport of these molecules?
So okay, so that's kind of interesting question. So most of the compound and they do cross the RBC. In fact, the parasite, there are three membranes. Okay, one is RBC membrane, and then there is, so when the parasite infects RBC, it forms another membrane which is called parasitophorus vacuole membrane, followed by there is the parasite cell membrane. So there are three membranes. So the compound has to cross three membranes to act on the parasite metabolic pathway, right? So, and all these compounds which function as antimalarials do cross, but then there could be a compound which is a great inhibitor of a metabolic pathway in vitro, but may not kill the parasite just because it can't cross, and that might happen. And the other interesting thing about the infected RBCs is, so RBCs, so when the parasite infects RBCs, so the permeability of infected RBC increases about 1,000-fold compared to uninfected RBC. And that is the reason why when we treat parasites with 5% sorbitol, which kills the metabolically active stage like trophozoite and cyzone, but ring stage, which has just begun after Maharaja invaded, is metabolically not so active, so remains intact.
Sir, what is the full form of TMP that you have used to induce nadylation of protein?
Thymine.
No, TMP. Okay. No, TMP is this. That is an antimicrobial trimethoprim.
Trimethoprim.
Yeah, trimethoprim is an inhibitor of DHFR. So it is an antimicrobial. And so what happens is E. coli DHFR, the one we have used here. So that is a mutant. So this mutant, when it is bound to TMP, it has a stable conformation. But when TMP is not there, its conformation is unstable. Therefore, it undergoes degradation by the ubiquitin proteasome system. So when it is fused to a protein, the fusion protein is also stable when TMP is there. But when TMP is not there, the fusion protein is also not stable and undergoes degradation. That's why you can induce knockdown at protein level.
Okay, sir.
So you wanted to have a control.
Exactly, because what happens in plasma, what happens? So it's still, these genetic manipulations are kind of okay. I wouldn't say that they are easy, but they are well worked out in mammalian system yeast. But in Plasmodium, the genetic manipulation is still very challenging, and you don't have straightforward inducible knockout and knockdown. So if a gene, or let's say if a protein is essential, so you simply can't knock out the gene. So, but if you have this inducible knockdown, and if it works, so you can study function of that protein.
True.
Yeah.
So this is an interesting model system, and this strategy can be used where somebody encounters a problem with having controls.
Right, right.
Are there more questions? Thank you very much for an interesting talk. And
thank you, madam, and all the best to MS University Baroda for organizing this wonderful meeting. And I hope your students will benefit from it, as well as some of the faculties. And if you are looking for collaboration in the areas of your interest, please contact me.
Yeah.
Okay.
Yeah.
I will stop sharing my presentation now.
Now I request Pushpa to introduce our alumni.
Yeah, so hello everybody. Now we begin with our own alumni speakers. So the first is by Dr. Shushmita Jha. It gives me great pleasure to introduce Dr. Shushmita Jha. She passed out in the year 2004. She's done a BSc from St. Joseph's College, Bangalore. And after her MSc in Biochemistry, she went on to pursue her PhD from University of North Carolina at Chapel Hill. She completed a PhD in 2009 on the role of NLRs in neuroinflammation and demyelination in the CNS. After her PhD, she had a very short stint as a postdoctoral fellow at the same university, where she worked on molecular mechanisms of demyelination in CNS. After this small training, she returned to India in the end of 2011, and ever since she has been associated with IIT Jodhpur, where she currently serves as associate professor. She's also been the head of department for a small period of time. Her research interests continue to be in the area of inflammation. Her lab is very well funded with funds from all major funding agencies of our country. Being in IIT has its benefits. Her institute has been identified by the DBT for the Genome India initiative. And on this project, she's a co-PI. Another project that the institute has got is in the most happening field of biology and IT, which is artificial intelligence. Dr. Shushmita is also a co-PI on the project for developing an AI platform for human health. She has filed for a patent for a low-cost portable hypoxia chamber. She has trained several bachelor's and master's students for their dissertation and has already guided 8 PhD students in such a short time. She has won several awards like Young Scientist Fast Track Award, Outstanding Molecular Biologist Award, and very recently the Kusum Sharma Award for Young Women Scientist. I remember Shushmita as a very meticulous student, and I'm sure she's still the same. With this brief introduction, I would like to welcome Dr. Shushmita to give a presentation today. Shushmita?
Yeah, thank you so much, ma'am. Thank you so much for the warm introduction.
Welcome to the virtual platform, and then we'll see you soon on campus.
Yeah, I really hope that we can meet in person very soon. Let me share my screen so that you can all see this.
For some reason, the screen is not sharp, the image.
My image?
Yes.
Let me see.
This seems to be okay.
Is it better?
Yeah.
Yeah. I had blurred the background a little bit. Yeah. So I hope I'm audible and my screen is visible.
Yes, please tell me.
Thank you so much. So before I begin, I would like to thank Pushpa Robin ma'am and Dr. Ratnaprabha ma'am for inviting me today and giving me an opportunity to share our research on identifying novel prognostic markers for glioblastoma. It is a matter of honor for me to come back and speak at my alma mater. So before, let's get into the science right away so that we stay on time. And then we'll come back at the end for questions. You can always stop me if you have questions in the between. So I work on glioblastomas. That is one of the projects that is in the lab. And what are glioblastomas? These are a kind of cancer that arises in the brain from glial cells. Now, they're considered the deadliest cancers because even after aggressive surgery, chemotherapy, and radiotherapy, the median survival after diagnosis is less than 14 months. 54% of the global CNS tumor prevalence arises from Asian countries, and India is the second largest contributor. Now, we know that gliomas arise from glial cells, and we also know that they are heavily infiltrated with innate immune cells, specifically microglia. So within the tumor microenvironment in glioblastomas, there is a lot of heterogeneity. So you have stem cells, you have endothelial cells, dendritic cells, astrocytes, and of course, the microglia. In the typical healthy brain, microglia comprise 10 to 15% of all glial cells. But in glioblastoma, they are 35 to 50% of all cells. They stimulate proliferation and invasion of glioblastoma cells in vitro as well as in vivo. So this is extremely interesting for us. Of course, we've known for a while that the glioblastoma pathophysiology is complicated with aberrant metabolism, immune cells playing a role and being switched off or being in an anti-inflammatory role, and also aberrant metabolism and environment playing a role. In the last decade, clinical trials investigating targeted therapies have failed to demonstrate any significant improvements in survival, and innovative new treatments are urgently needed for glioblastomas. Recently, our focus has shifted towards novel strategies that can modulate this immune response towards the tumor and also the surrounding microenvironment. If we can alter it, or if we can better understand how it plays into the immune environment or the tumor immune microenvironment interaction. We know that recognition and eradication of malignant cells via immunosurveillance of tumor-associated antigens is a key function of the immune system, and that tumors manipulate the immune system to avoid detection of the tumor-associated antigens, and this helps them grow and survive. So then how does the body detect these tumor-associated antigens or pathogens or damage or anything which is different? So we know that the body utilizes pattern recognition receptors to detect pathogen-associated molecular patterns, damage-associated molecular patterns, such as molecules that are released when a cell dies or when wound healing needs to occur, and irritants such as silica or dust or diesel smoke. And then after detection, these receptors also help in initiating inflammation. So pattern recognition receptors are of many different kinds. And you might have heard about toll-like receptors, the Rigai-like helicase receptors. What I'm going to be focusing on today are the NLRs. What are NLRs? So NLRs are nucleotide binding domain, leucine-rich repeat containing receptors. Now, these are evolutionarily conserved pattern recognition receptors, so much so that their relatives or similar proteins are found as far back as in plants. So they have a very strong evolutionary conservation. NLRs are expressed by cells of the innate immune system, as well as the innate adaptive immune system. So specifically monocytes, macrophages, dendritic cells, endothelial cells, and neutrophils. And of course, what we have discovered is that they are very widely expressed in the brain, in astrocytes, and in microglia. So when these NLRs detect pathogen or damage or an irritant, they can form multi-protein complexes called as inflammasomes. Now, some NLRs form inflammasomes, some don't. The ones that form inflammasomes are called inflammasome forming NLRs, and the ones that don't are non-inflammasome forming NLRs. So here you can see that this inflammasome protein complex then gives rise to many inflammatory mediators that can go on to regulate adaptive immunity as well as innate immune responses. So in this slightly busy slide, I'll take you through inflammasome activation. Now, what I would like you to focus on in this is that there are multiple different NLRs here. And we have NLRs such as NLRP3, NLRC4, NLRP1, and many of these NLRs, so there are 23 of these in humans, that respond to different PAMPs and DAMPs and irritants, and therefore provide some level of specificity, but they also have redundancy, and we have been able to see that one can sometimes replace and associate with another, and therefore innate immunity doesn't suffer. So what you see here is that there is a priming stimulus, which is stimulus 1, which leads to activation of NF-kappa beta signaling, transcription of the pro-inflammatory cytokines as well as the inflammasome components. And then the second signal causes inflammasome formation with the NLR protein associating with an adapter protein, typically the AST protein, which has a caspase recruitment domain that can recruit pro-caspase 1 to it by homotypic interactions. This then leads to formation of this inflammasome complex. Within this complex, the pro-caspase 1 undergoes autocatalytic cleavage, and once cleaved and activated, caspase 1 can then cleave and activate many different proteins. In fact, one of the papers from Maya Saleh's group talks about how caspase 1 by itself can cleave and activate more than 70 different proteins, from inflammatory mediators to housekeeping genes. Here, we are going to focus on the inflammatory mediators, pro-IL-1 beta and pro-IL-18. These are pro-inflammatory cytokines that, once released out of the cell, can recruit other immune cells, and inflammasome complex activation can also initiate several cell death pathways, such as pyrotosis, which I have listed right here. So again, inflammasome activation would then be a first step towards recognition. Now, just as we know that inflammasome activation is important for triggering a response to PAMPs, DAMPs, and irritants, we also know that dysregulated NLRs or dysregulated inflammasome signaling is central to many diseases. So my own research has shown that NLRP3 and NLRC4 are central to neuroinflammation. And what we know now is that many of these NLRs, if mutated or dysregulated in terms of signaling, can lead to diseases ranging from MS and TBI effects to diabetes, colorectal cancer, vitiligo, and many other hereditary fever syndromes, as well as autoinflammatory diseases. Now, cancers, NLRs are known to play a big role in many different cancers, from oral squamous cell carcinoma to breast cancer and colon cancer. In fact, several different NLR family members have been known to play a role in cancer, in a protective role in cancer. However, despite understanding the critical role of NLRs in cancer and the physiological and functional significance of NLRs in the brain, we still don't know how these play a role in glioblastoma pathophysiology. Then, for us, the key questions to ask were, are NLRs differentially expressed in glioblastoma tissues versus non-cancerous brain tissues? Is there a reason why within the glioblastoma our innate immune cells are not able to sense the tumor-associated antigens. Next, how does the tumor microenvironment alter innate immunity? We know that within the tumor microenvironment, the environment is largely hypoxic, and this makes the metabolic pathways very different from that is found on the periphery. So we need to understand how metabolism changes within the glioblastoma core. And finally, what are the key molecular signaling events that can then be utilized either as therapeutic targets or to alter signaling so that we can hope in the future to either prolong the lifetime or to delay morbidity of these patients. So to answer these questions, we carried out a data-driven approach. So the first thing that we did was looked at the Cancer Genome Atlas, and the Cancer Genome Atlas has data sets which allowed us to compile and look at NLR expression. But of course, what is missing in the Cancer Genome Atlas is Indian patient data. Now, this is very close to my heart. I feel that we need to have more precision oncology, which is tailored for our populations. And therefore, we set out to set a collaboration up between IIT Jodhpur, AIIMS Jodhpur, and Tata Memorial Hospitals and ACTREC. This then allowed us to get patient samples from Indian patients and blood tissue as well as brain tissue from these patients and compile the protein expression profile from our Indian subcontinent and understand glioma pathology. So we next looked at fresh tumor samples, normal and tumor tissues. So by normal, I mean non-diseased, and in some cases during surgery, there might be some normal margins available that can be utilized. We took serum samples, of course, paraffin embedded fixed sections. And we carried out tissue studies as well as functional assays to understand the role of NLRs in low-grade gliomas as well as glioblastomas using a multi-pronged approach of utilizing the TCGA dataset, human brain tumor tissue, as well as functional assays that utilized primary cell cultures as well as cell lines. So we started our analyses first by extracting the data from the Cancer Genome Atlas. This work was largely done by a very talented PhD student in our lab, Nidhi Sharma. Nidhi was the first PhD student of the lab, and she came from a bioinformatics background, but with a lot of hard work, she ended up learning experimental techniques, and she became one of the pioneers in the lab for many different techniques and was very open to collaboration, which is the kind of student, who ends up not just learning a lot, but also teaching a lot to us. So Nidhi is now a postdoctoral fellow at Karolinska Institute, Sweden, and this is her right here. So what she did was, first, she utilized seed genes. So we selected some genes that we knew play a role in cancers. And we also added some genes that we know play a role in case of macrophages, in case of glioblastomas. We put them in, and we looked at the C bioportal. The C bioportal allows us to visualize, analyze, and download large-scale cancer genomics data, and then we can input our seed genes and generate networks that gives us an overview of what are the altered genes in the pathways that are related to glioma, and therefore it helps us highlight the frequency of alteration, mutations, copy number analyses, mRNA upregulation, downregulation. And what we noticed right away was that the network was altered in 38.9% of the low-grade glioma patients and 38.5% in case of the glioblastoma patients. What does that mean? That means that if you look at the percentages right here, you see that there are certain genes such as TP53 were changed by 53%, EGFR, which is very important for signaling in case of gliomas, and CDK11B were altered by 15 to 20%. And our own NLRs, P6, NLRP3, C4, NLRP12, all of these were altered between 5 to 10%. Notably, we could see that P53 was a driver mutation across low-grade gliomas, and in case of the high-grade gliomas or glioblastomas, EGFR was altered by 65% right here, and another NLR, NOD1, was changed by 40%. So these were very interesting because our hypothesis was that these genes may be altered in case of glioblastoma pathophysiology, and this was our first indication that we might be on the right track. So next, we set out to quantify the NLR gene expression patterns in gliomas versus glioblastomas. And we performed differential gene expression profiling across low-grade gliomas and glioblastomas. And what we saw here in the heat maps is that across the low-grade gliomas, grade 2 and grade 3, which are considered low-grade, and grade 4 is the glioblastoma, we could see that in the low-grade gliomas, there was not much of a difference between the gene expression profiles. So neither in G2, G3, nor as much in case of neither in case of G2, G3, nor so much in case of G3, G4. But we could say that when we start looking at low-grade versus high-grade glioblastomas, that there is a significant change in gene expression between the grade 2 and the grade 3, and between the grade, from what we saw grade 2, grade 3, and to grade 2, grade 4. Interestingly, we also found our NLR genes, NLRP12, C4, NOD2, caspase, and PICARD differentially expressed in glioblastoma with respect to their gliomas. So this differential gene expression grouped by the grade allowed us to visualize and understand that NLR gene expression may be in fact changing across the different stages or different grades of the glioma. So with the gene expression profiling, we helped identify the association of this differentially expressed genes with either an early prognosis or a critical outcome in the glioblastoma patients. So next, we used Kaplan-Meier method to assess the prognostic value of the corresponding genes in low-grade gliomas and glioblastoma. This work was done in collaboration with my colleague, Dr. Sushmita Paul, who's a computational biologist here at the Department of Bioscience and Bioengineering at IIT Jodhpur. So Sushmita and Nidhi together carried out the calculation of overall patient survival based on gene expression and stratified patients on death information into grade 2, grade 3, grade 4. And they found that in fact, in case of NLRP12, there was a significant separation of the two risk groups that are characterized by differences in their gene expression. So it is seen that the high expression of NLRP12 leads to poor overall survival of the patients. Now, while TCGA provides a comprehensive genome profiling from whole tissue, but it neglects individual cell population effects. And more evidence now suggests that cell-specific and tissue-specific role of pattern recognition receptors, especially NLRs, makes a big difference in our understanding of cancer pathophysiology. So then we started looking at the expression of these NLRs in different cellular populations within the tumors. So in the interest of time, I'm going to focus only our data on NLRP12. And what we saw is that all of the NLRs, the adaptive protein ASC, AIM2, caspase-1, and many of our NLRs under normal and inflammatory conditions all showed altered expression in case of the innate immune cells in the brain microglia, and the glioblastoma cells. Using immunofluorescence microscopy and Western blotting, we were able to characterize NLRP12 expression in both microglia and in case of glioblastoma cell lines. And as you can see over here, in case of microglia, the control microglia show high expression of NLRP12, but if you treat them with LPS, there is a downregulation of NLRP12. That is because NLRP12 is typically considered an anti-inflammatory NLR, and you typically see that when treated with an inflammatory agent, the cells will downregulate NLRP12. So typical NLRP12 is higher than not. So next, we carried out colony formation assay to look at the role of NLRP12 in cell proliferation. So to understand the role of NLRP12 in microglia as well as glioma cell proliferation, we utilized siRNA silencing of the NLRP12 gene. And then utilizing bright field imaging and quantitative analysis by gene sustain, we could show that the colony formation increased in case of the B2 cells. Now you might be wondering, what is colony formation? It's really quite simple. You take some cells, so typically very low numbers, so 40 odd cells per culture slide, and then we allow them to proliferate over a period of time and we quantify them. So we either have them as controls, so you have the controls with just the media, just with the apopectamine, or with the scramble siRNA, or here the siRNA against NLRP12. And what you might notice right away is that the ones with the siRNA against NLRP12 show an increase in proliferation. So on quantification, we could see that the number of colonies was significantly more in case of the microglia that had been treated with NLRP12. Conversely, what we could see is that if we did the same treatment on glioblastoma cell lines, the glioblastoma cell lines showed a decrease in colony formation. I hope I'm audible.
Yeah, very nice.
Okay. This has happened to me once recently, that I was speaking online and then the network connection got disconnected. So I just wanted to make sure. Okay. So this allowed us to see that NLRP12 plays a reciprocal role in microglia versus glioblastoma cell lines. So while in case of microglia, we see that inhibiting it causes microglial proliferation, the converse happens in case of the LN18 glioma cell lines. These suggest that NLRP12 inhibition could, in fact, be a good strategy in case of glioblastoma cell lines. Okay, so next, we wanted to see that, excuse me. Next, we wanted to see if this data that we saw from cell lines would in fact hold true in human tissues as well. So this work was carried out by Shalini. Shalini was a PhD student in our lab, and she's now a postdoctoral fellow at New York. Actually, she's at University of Chicago, USA now. And what she saw is that in case of glioma tissues, NLRP12 expression is differentially expressed so that we see colocalization of NLRP12 with microglia, and we see a very high expression of NLRP12 within the tumor core. So this was very interesting to us, and of course, I'm not showing you the data today, but what we ended up doing is we ended up carrying out proteomics analysis of tumor brain tissue and serum samples from patients, glioblastoma patients, and we could see that there was a distinct expression profile in the tumor tissue versus what we saw in the serum, indicating that B12 plays a selective role within the brain and not within the periphery. Now, to the second thing that I had hinted at the very beginning is that the tumor microenvironment plays a big role in innate immune signaling. So to test this out, we wanted to look at how hypoxia plays a role in the gene expression profile. And we looked at, again, NLRP12 because we wanted to test out the expression related regulation within the hypoxic environment. And we had seen that NLRP12 is elevated inside the tumor core. So to assess the role of P12 in a hypoxic environment, we created this portable low-cost hypoxia chamber, which was published soon. The patent was published soon. This work was carried out by Shivanjali, who was another PhD student in our lab. Shivanjali joined our lab as a computational biology student, and then she learned a lot of the techniques in our lab that we at that point of time we didn't have. So a lot of the primary human cell culture she standardized, and she carried out work with this. And also she was one of the students who had a lot of entrepreneurial spirit. So she went on and participated in a lot of these medical technology hackathons, and she continues to be interested in creating new technologies. So I think that is the kind of one of the advantages of being in an IIT, that you can collaborate with engineering colleagues and end up creating technologies as well. So Shivanjali created this hypoxia chamber. She also standardized the chamber, characterized expression of different genes in within the cells. So she did all of this using endothelial cells, primary endothelial cells from human tissue, and then she confirmed that within the hypoxic environment, there was a differential expression of NLRP12, which was found only within the hypoxic microglia. And so we could see that NLRP12 expression was increased in the HIF1 alpha positive cells within the tumor regions in the brain. We could also confirm the same in case of our primary human cells. So now that we've kind of looked at our data kind of from different perspectives, we can start thinking about how do we study these recalcitrant tumors. So one way that I talked about is looking at tissues by a multi-omics approach. So you can data mine, you can look at the cancer genome atlas, you can look at primary cell data, imaging, microscopy. We can carry out cell culture studies with primary human cell lines, animal cell lines, functional assays, and also animal studies. So mouse studies, patient-derived xenograft models where you can take patient cells and put them into mouse models. But again, this is something like the elephant and blind men story, right? Where each one of the blind men is looking at the elephant with their own narrow perspective. So the one that's holding the tail is calling it a rope. The one that's looking at the back is calling it a wall. The one that's holding the legs is calling it a tree, so on and so forth. This is exactly what I thought when we started looking at our cells in culture. So when we look at glioblastoma cells in culture, they look well-defined, very distinct in their shape, and on a flat surface, they spread out and they kind of grow happily. Similarly, when I look at innate immune cells, microglial cells in culture, they are distinct, they have a distinct shape, they mind their boundaries, and they grow flat, spread out. But if you look at, when we started looking at primary cell culture from patient-derived glioblastoma tissue, we see that these cells look very different. They don't mind boundaries, they grow on top of each other, and they prefer to be three-dimensional, sometimes forming these three-dimensional structures in the flat dish as well. So then why are we studying tumors in flat petri dishes when in the brain they are in 3 dimension? So we decided then to start looking at our data in spheroids, which provide a 3-dimensional model which is similar to in vivo tumors. These also allow us to look at the native tissue architecture, the tumor microenvironment, and cellular crosstalk. So we have on the left, you can see in vivo tumors, that have a gradient for nutrients, carbon dioxide, and waste. They also have a gradient for pH. And there is a necrotic zone in the middle, there's a quiescent zone, and there's a proliferation zone. Because of this stratified structure, there is an issue with limited therapeutic penetration, which is a big problem in case of solid tumors and also in case of glioblastomas. When we look at tumor spheroids, we see that there are similarities in terms of the gradients and that we can generate these mixed cellular spheroids, which can largely look at, help us look at the three-dimensional crosstalk and the role of microenvironment, as well as the architecture of a tumor. So we are now able to isolate adult human brain tissue, and this is done in collaboration with our colleagues at AIIMS Jodhpur. And this work is being spearheaded by Pritika. Pritika is a PhD student. She's one of the students that joined in 2020. So very difficult, right, to start out your work during the pandemic. But she was able to generate patient-derived cell culture. And then from that, we were able to generate three-dimensional patient-derived spheroids. This is a visualization of the 3D patient-derived spheroids by immunofluorescence microscopy. This was sectioned. These are 5-micron sections. And this is a spheroid showing you, actually this is just 3 days after they start growing. So they can get as big as a few millimeters in size in the lab. So we are now able to then generate tumors, mini tumors from patient tumor tissues, and also from the normal tissue from the tumor, we are able to create these patient-derived spheroids. Why is this important? Because then we can look at multiple aspects of precision medicine, regenerative medicine, toxicology, drug discovery, omics, disease modeling using these glioblastoma patient-derived spheroids. In fact, this is the basis of our collaboration with the computer science department and the AI faculty at IIT Jodhpur, where we have been able to generate these patient-derived tissue samples, patient-derived glioblastoma spheroids, and generate image data, multi-omic data, and clinical data, which can then be integrated into a multimodal data repository. And our colleagues in the computer science department generate AI-based continuous learning models that can then compare and contrast the similarities between these two patient-based tumor spheroids with the MRI data that we get from AIIMS. And we can also utilize an iterative approach to understand how closely we are modeling the tumor. So in conclusion, I would say that we were able to identify novel innate immune pathways in glioblastomas. We were able to report for the first time that P12 plays a role as a prognostic marker in glioblastoma, and that there is a role between P12 and the tumor microenvironment, and that we are able to develop patient-derived glioblastoma spheroids. None of this work would have been possible without the dedicated students in our lab. I would like to correct Pushpa ma'am, I haven't mentored 8 PhD students yet. 4 PhD students have graduated from the lab, and I'm very happy to report that the first one went to Karolinska. Ishan was at Oxford, and Ishan's story I was not able to discuss today. He has been able to discover dopamine-induced extracellular traps in microglia. This story was published in iScience last year. We have Shalini at the University of Chicago and Shivanjali at New York University. We have master's students also who've helped us with our stories, and of course, the summer students who come in and volunteer and intern in the lab as well. I would like to also acknowledge our collaborators at Tata Memorial, at IIT Jodhpur, AIIMS Jodhpur, and IISA Kolkata, and also at UNC Chapel Hill, and of course the funding agencies. Ministry of Electronics and Information Technology, MIT, Department of Science and Technology, DBT, SERB, and IIT Jodhpur, because of whom we are able to carry out the work that we're able to do it. For the students in the audience, I would really like to have you come over to the lab and visit our lab. So you can scan this QR code, it'll take you to our lab website, and you can get in touch with our lab members by email or on Twitter. And if you have any questions, I would really love to have you over. If you would love to visit, our lab is always open to visitors. We love to have visitors over. Today is something of a coming full circle for me. So my journey in science started at the Department of Biochemistry, MS University, Baroda. I can never forget the first day that I walked into the department stairs. The picture right in the middle is where we are sitting. It was as if a dream had come true. And I still have vivid memories of our first rounds, first presentation. I remember going to Ratnaprabha ma'am's house to understand a paper right before my paper presentation, and she was so gracious, and she helped me understand the nitty-gritties of the techniques that were in the paper, which I had so naively decided I'm going to present. I had no idea what I was getting myself into. I think I learned to learn at MSU. I would not know how to ask questions or just how to do science if I hadn't been to MSU. I think the people who made the biggest impression on me were the people who were there in the department at that time. This is Telang ma'am, Tara ma'am. I get teary just talking about them. They made a big difference to my life. And I think everybody, Pushpa ma'am was around there. She mentored my friend Prakash Maru there. Ratnaprabha ma'am, of course, she taught us so much about biophysics. And I still remember getting excited about centrifuges, just with her classes. Naresh sir, whose lab I joined at that point in time, it was a wonderful learning experience for me. Getting to know so much molecular biology at that point of time, I had no idea. And it was a really nice time getting to interact with and openly talk about science. In fact, the presentations, the journal clubs were something that defined the way we thought about science. Jayashree ma'am with her warmth, and Dr. Acharya sir was amazing, because I still remember going back home after my first year at MSU in Diwali, and my friends told me, you talk different. And I think it was all because the way that we had started to discover, learn all of that. I would like to thank all of you. Sarada ma'am for my love of physiology. I think I continue to do physiology, and I continue to appreciate it even today because of what we learned at that point of time. So thank you so much, everyone. I don't want to take so much time. Of course, one more thing that I have to thank MSU for was helping me meet my better half, Siddharth, who's right here having tea, which also happened at MSU. So thank you so much again for listening to me. I'll take any questions. This is a view of Mehrangarh at Jodhpur. And as they say on this end of the world, Padharo Maro Desh. So we would love to have you over sometime, definitely. Questions, please, everyone. Thank you.
Sushmita, excellent talk.
Thank you, Jyoti. Thank you.
So do you know how you isolate or culture? How long you can keep them in the culture growing? Like, can you passage them? And
which cells are you talking about? My human cells?
Primary cells, like you saw the spheroids you are culturing.
Yeah, so our spheroids, we are able to keep them for very long, actually. So we have been able to continue to grow them for months, actually. So they get very large in size, and yeah, they are so much so. And I've actually collaborated. We have some collaborators in the University of Bonn, and they told me that they have some spheroids that have been growing for about a year also.
So how do you passage them? Like, do you trypsinize them or just dismantle?
So okay, so I guess if you're saying how do you analyze per spheroid, is that what you're trying to ask?
Passage them. You will continue to let
you have to change the media in which
so,
you are growing.
So you basically end up changing the media. That's about all you do. Yeah. So they continue to grow in the same wells. So we put them into these 96 well plate format. Initially, we were growing them on agar, so they were able to grow in that also. But now we grow them on low adhesion plates, which have kind of round bottoms, and we end up keep changing the media every few days, and that's about it.
Okay, okay.
For that, do you use suction or what? How do you remove the old media and add new?
We basically cut the tips off the 200 microliter tips, and then we remove the media from the top. What I find very, very interesting is just like every single tumor is different per patient. The same way, when we grow our mini tumors in the lab, we see that some of them will have this lipid-like layer all around them, which is what is reported by our neurosurgeons talk about it, that some of them have these lipid-like layers, some of them have boundaries which prevent therapies from going inside. We have had at least 20 spheroids where they have such a gelatinous layer around them that it is difficult for us to see even by light microscopy. So the only way I can see is basically if I fix them and I cut them and then I stain them. Otherwise, I don't know what is happening inside. So we either do immunohistochemistry, because technically they're a mixture of cells. We break them up and do flow cytometry, or those are the things that we can do.
Thank you, Sushmita, for such a nice talk. You have illustrated one point that you can, by collaborations and having the right attitude, you can diversify and go into so many aspects of the same problem and learn many things in a short while. And also, thank you very much for carrying such sweet memories of the department and MSU, and once again sharing them with us.
Thank you.
I think NK sir wants to say something.
Yeah, Sushmita, it was a nice presentation.
Thank you.
Nice to listen to you after so long.
Yes.
I have a few questions. I'm trying to understand the glioma tumors in the patients. Till what size you have only when you start getting microglial cells population? Second, what stage you have capillary formation?
So typically anything that is more than 1 millimeter cube requires blood vessel formation. Okay.
But sometimes someone told me that RP from JNU, something 100 to 200 microns, after that you require capillaries for the tumor to survive.
So so what we know now is that a tumor size of 1 millimeter cube requires capillaries to form. I'm sure in other solid tumors too, it could vary across tumors, but essentially beyond that you won't have diffusion of oxygen happening. Glioblastomas are also hardy because they can switch their metabolism. So we know that now. But another thing that we are trying to do is we are trying to populate our gliomas with endothelial cells, and we are hoping that we'll be able to grow tubes within the tumor spheroids, mixed tumor spheroids.
So the what stage then you get the microglial cells infiltrated?
So what we end up seeing, and of course there are not good models, I would say, to temporally resolve this, right? But what we are doing now for temporal resolution is we are able to see over a period of time how the spheroid forms, and we are able to see it within 2 days. But how the proliferation is happening within the core, that we have not been able to establish till now. And what I can say from the existing data across other studies is that the proliferation and the support for proliferation of different cells varies if you have a mouse environment versus a human environment.
Correct.
And also it varies if you have iPSC derived cells or not. So there were a lot of people who suggested, why don't you generate microglia from iPSCs? But then we had meetings where we realized that iPSC batch to batch variations is so much. There is no point. Rather, we should just culture from our primary adult microglia. We are able to culture that, and we can reconstitute. But to answer your question, I would try to guess is that the rate of proliferation would vary across models, and there would not be a single time point where it all comes together, just because we see so much heterogeneity patient to patient.
But what I understand is that the spheroids, basically what I'm trying to do is spheroids, how closely we can relate to the natural tumors. That is the main.
Yeah, so I can speak to that. So the percent, so I can tell you when we started growing spheroids, we was trying to standardize tumor is to microglia. And we first, in the beginning, we just could not have them make the spheroid until we were at a 1 to 10% ratio, 10% ratio of microglia, which is what you typically see across the brain, right? So if we put 10% microglia, then they make spheroids. If there are more, they don't form spheroids. Microglia by themselves, they don't make spheroids. So you need to have that critical ratio. I think we are going over time.
Yeah.
Thank you.
Yes, you have to come to department for this.
Yeah.
Thank you so much, ma'am. Thank you for calling me.
And now this
I have one question.
also more or less resolved. You can come anytime.
Megha has a question, I think. Megha.
I think Megha.
Yes,
I
ma'am.
suggest you write to Sushmita.
Okay.
Or
Yes.
I'll to her because the online meeting we have to stick to time.
Yeah.
The next speaker is waiting.
Yeah. Thank you so much, ma'am. Thank you.
Yeah. Okay. Thank you.
Bye-bye, ma'am. Thank you.
It gives me great pleasure to invite our next speaker. He is Dr. Nilesh Agera of the 2006 batch. Dr. Nilesh did his bachelor's from Christ College, Rajkot, and after his master's in biochemistry, he cleared the UGC CSIR exam and went to NCPS Bangalore for his PhD. He completed his PhD in 2013 under the mentorship of Professor Udgaonkar, where he got some wonderful papers and also one cover in the Journal of Biochemistry. After which, from August 2014 to February 2020, Nilesh was an inspired faculty at the Molecular Biophysics Unit at IISc Bangalore. His research interest mainly is in protein biology, and currently he's working at Intas Pharma as a research scientist. With these few words, I now invite Nilesh for his presentation. Nilesh.
Yeah. Hi, ma'am. Thank you very much for the nice introduction. I'm glad to be here, and I thank the organizing committee for inviting me and giving me this opportunity to present my work here.
You have to increase the sound.
Okay, I'll try to be louder. Is it clear now?
Yeah. Yes.
Yeah. Okay. Okay. So it's again, I'm like really glad to be here, and I have now tough challenge to present here because now audience are tired and it's difficult to keep everybody's attention here. So I hope I'll do a wonderful job here and make everybody here understand what I have done. And one more thing, I try to keep my presentation as simple as possible. I try to avoid going into technical detail so that everybody gets the flavor of work rather than technical jargon of work. So is my screen available
Sorry
to everyone?
to interrupt.
Yeah.
Nilesh, can you switch on your camera?
I have switched on, but I think there's some problem. It's not.
I see, because
Yeah,
we
I'll try
see.
again. So it's on, but it's not displaying. I don't know why.
Maybe.
Okay, let us see at the end.
Yeah.
Yeah.
Okay.
You can go ahead with your presentation.
Okay, my presentation is visible, isn't it? Can you see it?
Not yet.
No.
Not yet.
Okay. Try screen. Share. Yeah. Yeah. Is it?
Yes.
Now.
Yeah.
Yes.
Okay. So today I'll be presenting some work which involves the kinetic studies of protein-protein interaction, which regulates microbial physiology. So it's a multidisciplinary work, and it's always difficult to do justice to such a work. Let's see. Let's go ahead one by one. So as you can see in my title, there's a DNA, but I'm not going to talk anything about DNA. It's all about protein. And before going to data, I'll give a background of what I'm doing and how and why it is relevant. So you can. So I'm going to talk about a toxin and detoxin system. So these are present in bacteria, and just to give a brief introduction about this system, how they are as operon. This operon expresses as an antitoxin and toxin. Okay, and this protein, once expressed, they bind together to form a complex. And what this complex does is regulate its own expression without doing any function under normal physiological condition. Then the question arises why this system are even relevant. And at first it appears that what it does. So, but whenever there's a stress, what happens is half-life of this antitoxin is much shorter than the toxin. So antitoxin gets degraded, and then this toxin goes and binds to its cellular target and kills the cell. And then after knowing this system sounds like an parasitic system or selfish system, which ensures its own existence. Because once there is no such a system, no expression from operon, it is going to kill the cell. But further understanding made us realize that they are not just parasitic systems. So these systems are found in many pathogenic bacteria and other bacteria as well. And what has been realized is that they help in persister formation. So what are persisters? So persisters are the bacterial cell. They exist in dormant cell. They are not actively metabolating cell. And this cell can tolerate a high drug pressure. So basically, this system allows bacteria to survive under drug conditions. And that's why this seems to be relevant. And since these systems are found in pathogens, they seems to be even more relevant. Okay, so this is what the system I'm trying to understand. So which system I'm trying to learn is a CCD. It's a controller of cell death system. So this system, as I explained, has a two protein. The one shown here in blue, CCTB, this is toxin. And the one here is a CCDA, it's antitoxin. So under normal condition, they form such a complex which are hypothesized, again, not characterized. And this seems to be a completely non-functional complex under normal condition. And the only thing they do is regulate its own operon. And what this CCDB does, it binds to this DNA gyrase. And this complex is a non-functional complex. So this acts as a toxic to a DNA gyrase. And it has been hypothesized, and there are some indirect studies which suggest that the CCDA can facilitate the dissociation of complex. So basically, CCDA just not bind to CCDB, but it also binds to a CCDB bound to gyrase and facilitate the dissociation. But it is hypothesized. Nobody has seen this complex that exists. But there are some evidence which suggests that it does. And one such evidence was we observed in our lab by a student Pankaj Jain. And what we saw is that this is SPR figure. It's a very simplified version I'm presenting here. So basically, when we have gyres mobilized on a SPR chip and we pass the CCDB, it forms a complex that you can see here in the rising part. And once the complex is formed, when we pass the buffer, this complex stays, which is seen in the black line. And but if we pass the CCDA of different length, which I'll not go into detail right now, so we can see that CCDB won't stay with the complex, it's coming out. So basically CCDA is facilitating active dissociation of this complex. This is something first we know that it is really doing. So then we wanted to understand this process, whether this complex actually exists. If exists, can we see, can we visualize by some or other mechanism? And if what is the mechanism by which the poison gyres are restored to a functional gyres? So my work is understanding this story. I'll go into this one by one. And so these are my objective, basically to delineate the state and then to understand the structural transition. What are the structural transition involved in facilitating this process? So these are the main part, and I'll try to discuss. First, let's go to the kinetic part. So kinetic part of rejuvenation. So we thought the freight is the best tool available to understand such process, and we went with this. So what is our strategy here? We have CCDA protein, which is antitoxin. We labeled it with acceptor. Our CCDB protein is unlabeled, and our gyrase is labeled with a donor. So hypothesis is that if this complex form, then only we will have a freight energy transfer between donor and acceptor. And if we can capture this freight energy transfer, then we are sure that this is there, and then we can characterize this process in more detail. So to start with, we did a simple experimental studies to ensure that since the fluorescence is sensitive to environment, we wanted to be sure that the binding of CCDB or our experimental condition is not giving us any bias in the process. So we characterize the fluorescence of donor and acceptor in the presence of the various protein, and we realized that there is no change in fluorescence under any condition that we possibly thought to exist. So that means if we see a fluorescence change, it has to be a freight driven. Okay. So now this is the process that we wanted to study. We have a CCDB gyrase complex, we add CCDA to it, and then anticipate that there will be dissociation of functional gyrase and formation of CCDA-CCDB complex. So when we perform this study, we realize, so I'll simplify these multiple lines here. We see that under all the anticipated condition where there shouldn't be any fluorescence change, we see a flat line. There is no fluorescence change. But when we mix these two proteins or complexes together, then we see a fluorescence change which has already occurred, and then it starts restoring to the normal fluorescence. To understand this part, we analyze this process using a stop flow, which is basically a device to study fast kinetics. And here we see a fast drop in fluorescence and then restoration of fluorescence. And how we can explain this behavior? So when CCDA binds to this complex, there is a fast decrease in fluorescence due to freight. And then in a slow process, this CCDA-CCDB complex comes apart. And during this slow process, gradually this fluorescence restores to a normal condition. So we are sure that there is an active complex formation, and that is how the dissociation occurs. So now we know that this occurs, but we wanted to understand this process in better detail and how one can do. So since this is a binding process, which is a second order reaction, and second order reaction is the rate of reaction is dependent on the substrate concentration. So if we initiate this reaction at different initial substrate concentration, we will see a change in its kinetics. And by looking at these such lines and characterizing it with mathematical model, we can figure out exact process. So that is what we did in the next step. So here you can see such rising lines which differ from each other in very little way. So but these differences are significant, and these differences need to be quantitated using mathematical model. So these are the reaction we perform at different concentration that you can see here from 1 to 5 micromolar, and this is what the difference we got. So you can see that the difference in 5-fold change in concentration gives rise to a change in rate which is not even roughly 2-fold. So this small change is because of the fact that the kinetics processes in protein are not simple as in chemistry, because it involves conformation change and it's coupled. And this is the reason why such an experiment allows us to delineate the finer detail. So then in next step, what we did, we hypothesized the possible mechanism, what could occur. So this is what we laid on the paper. Initially we have CCTB, CCD guys complex. Now we are adding CCDA. So there will be a first CCDA binding to this complex forming pentameric complex. Now there are two possibilities. The first thing is that the single CCDA binding is sufficient to facilitate the dissociation, or this is not good enough to facilitate the dissociation. Then it's possible that second CCDA molecule binds here, forming hexameric complex, and then this hexameric complex dissociate to give rise to free gyrase and CCD-CCTB complex. And there's possibility if this step does occur, then there will be dissociation of this pentameric complex to give a free gyrase and then eventually a formation of CCD-CCTB complex. But the question is which pathway does occur and which is actually operating. So to understand it, what we did, we fit this model using computational programming to our data. And what we see is that if we assume that this step does not occur, entire process occurs through this pathway, and then we try to fit the data, what we get is here. This doesn't match to our experimental data. So this seems to be unlikely. So then we assume that this is not the pathway, and then we tried by assuming that this pathway is not operating and this pathway is operating. And then we see that our computational program gives much better resemblance to our experimental data, but still it's not as good as we anticipated. So then we thought it's possible that both pathways are operating, and then we feed the entire model to a data, and what we see is that this entire model fits to our data really well, including all the data. So we came to a conclusion that both the pathways are operational. So the next question anybody will ask is this then your mathematical fitting artifact, or it's your experimental artifact, or it's really there? How can we check this? So to check this, what we did here, like we took an advantage of this dimeric nature. So let's say if we add this CCDA in half the amount, that is half the stoichiometry. Let's say you taking this 2 micromolar and we are taking this 1 micromolar. Then after formation of pentameric complex, this CCD is exhausted. Then this pathway won't be operational. Okay, so before going to the next experiment, I'll summarize here what we see. So these are the kinetic parameters that we obtain. And if we see here the formation of the pentameric complex is much faster than, formation of pentameric complex is much faster than the formation of hexameric complex. That means if we add the CCDA in half the stoichiometric amount, predominantly pentameric complex will form. In those, basically, we are experimentally eliminating the second pathway. And with the second pathway, what we see is the dissociation rate of the pentameric complex. That's much slower than the hexameric pathway. So that means if we use the half the stoichiometric amount of CCDA, we should see a much slower process of the rejuvenation. Okay, so let's see what we see. So we perform this study using half the stoichiometric amount, and the previously a process which used to take a 200 to 300 seconds to complete now takes 1500 seconds to complete. So that means our hypothesis or basically our interpretation is true, that the process does occur by both the pathway. When we have a substoichiometric amount of CCDA, predominantly this pathway is operational. But when we have sufficient amount of CCDA, predominantly this pathway is operational because it's faster. Okay, so this is what the mechanism we concluded, that these are the steps involved in the rejuvenation of DNA gyrase. The second question that we wanted to understand is, this is basically a brief summary I forgot to present. Let's say. So from our study, from the FRED study, we established the kinetic mechanism, and we gave an evidence for an hypothesized mechanism and for the hypothesized transient ternary complex. So we demonstrated that this ternary complex in fact exists. We also demonstrated that single molecule can catalyze this pathway, which was not clear. And the multiple pathway, which was not known, we could delineate here. And then going to the next question, that is, what are the structural transition involved in this facilitating this process? That's another dimension of the problem. So just to visualize what we are trying to understand by asking this question. So when we have this complex as shown here, and when CCDA binds to here, what happens? So there are some structural transition which should occur in CCDA because the DNA gyrase need to dissociate. Without structural transition, it seems to be unlikely. So is this transition cooperative, non-cooperative, and if whatever the nature, what are the transitions that facilitate the dissociation? So simplest thing for us to do is to take the crystal structures and do the comparative study and figure out what's responsible. And what we see here, this is the complex here. The blue color structure here is our gyrase subunit. It's not the full gyrase. And here the yellow and golden color part is our CCDB protein. And here our this green color, that's a CCDA. Okay, and this is a CCDB protein bound to a CCDA and bound to a gyrase. So well, let me clarify this. This complex does not exist. Experimental complex does not exist. This we have generated in computer by aligning two different structures. And again, this is alignment from two different structures for CCDB protein alone. So if we see a CCDB protein from two different complex here, they are very similar. There are hardly any structural difference at one or two places. And wherever we see these differences, they are not key structural differences because they are not involved in either CCD or gyrase binding. So probably they are not relevant. Then the question is how one can understand what are the structural transitions which are important. So for that, what we use is hydrogen deuterium exchange study. So basically we decided to study a dynamics of protein of a CCDB in its free form and in its complex with CCTA and in complex with guidance. So when we study a dynamics, basically we'll see which part of the protein is flexible when it's free, which part gets restricted, loses its flexibility when it binds to either CCD or binds to guidance. And we thought this might give us some answer. So before I go to the data, I'll just briefly explain what is hydrogen deuterium exchange study. So basically, the hydrogens present in protein structure, they are not static. They continuously exchange with the solvent. Okay, and if they're continuously exchanging in the solvent, and let's say we put our protein into D2O instead of H2O, then what will happen that the hydrogen will start getting exchanged with deuterium, and the flexible region of the protein will exchange faster. As you can see at the surface hydrogens, they will exchange much faster. And the deuteriums or hydrogens which are buried in the core, we have to wait for protein to open up, and rigid structure will open up slowly, and they will take time. So this is how we find out which part of the protein is flexible and which part of the protein is rigid. And eventually you look at the entire process and figure out which part is of your interest. Okay, so this is basically we are looking at, and then how one can see that where the hydrogen deuterium exchange is taking place. So typically there are two methods used for this. One is by NMR and one is by mass spectrometry. So here we have used mass spectrometry for multiple reasons. I'll not go into the detail of that, but anyway. So when we perform this study, this is what we see in the raw data. This is the so gray color line, you can see there's the mass distribution we see for a protein at the early stage. And as the deuterium gets exchanged with the hydrogen, its mass decreases, and eventually we see that mass profile shifts to a left hand side or towards the lower mass. Okay, and when we translate this into a two-dimensional data where we see average mass versus the time, we see such a complicated profile. So these profiles are difficult to understand and process. I will not go into the technicality of JAG and make it more complex. So basically I see that you can see that these two lines don't go with this line. What is this line is a free CCDB, and what these two lines are is a CCDB protein when it's bound to a CCDA and bound to gyrase. And you can see that the protein behaves almost in an identical way when it's bound to either gyrase or CCDA. So that was a bit unusual. And then we thought that at intake level, maybe it's apparently behaving similar way. But let's see how different regions are behaving, how we can do it. So basically, before analyzing into mass spectrometer, we chop off the protein using proteasine. So, and after chopping it, we analyze the different fragments of protein. And when we put that into mass spectrometer, we see some difference. As you can see now, these lines are now not overlapping. So we see here some difference, major differences here, major differences here, some differences here as well. Now, getting into each and every differences and rationalizing each and every differences would be little difficult, but I'll summarize here. So if you can see here at the bottom of figure, there are the residues and the structural mark, where the purple color residues, these are residues which are common binding residue to both CCDA and gyrase. And the one in cyan, they bind to CCDA, and one in green, they bind to gyrase. And what we see here that the structural transition, although similar, there is some the story that we can make out. And to further understand what we did, we performed the same study, but now instead of gyrase, we varied the length of CCDA protein. So CCDA, when it comes and bind to gyrase, there is a sequence of binding which is proposed. it seems that the N terminal binds first and followed by the C terminal, and that's what we use. So we use these different sequences here, where the binding binding of different sequence were utilized, and we see a minor differences here. But taken together, what we conclude is that there is an initial binding that takes place on this N-terminal part of CCDB. Okay, and this binding then induces some structural transition which triggers the common binding residue. And once these residues are triggered, then there will be CCDA binding to this residue and then eventually displacing gyrase. So this is something experimental data which tells one story and can be verified. So this is the model that we propose, that that CCDA binds and then induces structural changes which then eventually facilitates the dissociation. So then can we verify this model? So to that, we collaborated with the Professor Srinivasan's group, and they performed this study, complete molecular dynamic simulation study. I'll not go into the data, but what we interpreted here and what we see here is that once we have this complex, okay, and CCDA comes, which is intrinsically disordered protein, then it binds to a region which is far from gyrase. And this binding then induces some changes in the common binding residue. And these changes then eventually facilitate further binding. And this further binding then displaces gyrase from the complex and then forming a CCDA-CCDB complex and the dissociated gyrase. Okay, so this is the story that we have right now. And part of this work we have published in Structure and recently. It's not so recently, but this is, basically we have the kinetic mechanism, how this step occurs, and structural mechanism, that during which step, what are the structural transitions that takes place. With this, I'll thank to everyone who supported this project. So this HX work was mainly done in NCBS in Professor Udnaukar's lab, and a lot of work was done in MBU mass spectrometry facility, was taken care by Dipankar Chatterjee. And this work was mainly driven in Professor Vardarajan's lab, and he provided all the support required for this study. And I thank Inspire for funding and ISC for hosting me. And thank you everyone for your attention. And I'll be happy to have your question. And before that, again, I'll thank the organizing committee for giving me this opportunity. And I thank all the teachers at MSU who trained me and made me capable to get to good such nice work. And thank you everyone. So yeah, questions please.
Yes, students, teachers, anybody?
Apparently, there are no questions. Thank you for an excellent piece of work.
Thank you very much, everyone.
Yeah, all the best to you, and keep coming to the department.
Yeah, he's very near. He's in Ahmedabad. He can come.
Yeah.
So I think I'm frequent visitor to department, but because thanks to Corona, I haven't been there recently. But I'll come very soon for sure.
Yeah.
Welcome, welcome.
Yeah. You can also actually teach certain things, like what you have done by giving a lecture or two.
Sure, definitely.
Yeah. Nice. Introduce the next.
Yeah, I'll introduce the next speaker. Our next speaker is Dr. Shwetank Verma of the same batch as Dr. Nilesh. So he is a student of the 2006 batch. Shwetank did his bachelor's from Pune University, and after his master's in biochemistry, again he also cleared the UGC CSIR NET exam and joined IISc Bangalore for his PhD. He completed his PhD in 2013 under the mentorship of Professor Manjunath on the immunology of Japanese encephalitis virus. During his PhD, he developed an in vitro model for the study of this infection. After his PhD, he joined the lab of Professor Aaron Lukacher at Penn State College of Medicine for his postdoctoral studies. There he developed preclinical mouse models for progressive multifocal leukoencephalopathy and colon cancer. He worked in this lab for 6 years and then shifted to India. Currently, he is a senior research investigator at Biocon Bristol Myers Squibb Research and Developmental Center, Bangalore. He leads clinical discovery and translational research projects on autoimmunity, immuno-oncology targets. He also has supervisory roles, leading a team of scientists. I'm sure with his wonderful training, he will work wonders for research in clinical science. With this few words, I invite Dr. Shwetang for his talk.
Yeah, that's always the problem with the online meetings. We all tend to get into this trap. Thanks, Dr. Pushparobin, for this kind introduction. And I still think about all of you as the names we used to be, like PR ma'am, CRP ma'am, NK sirs. I mean, it just feels odd to say the full name of yours. It's just out of respect. And I mean, it's such a great pleasure and an honor to be talking about my research at my alma mater. I know I haven't been able to come to the department for some time. I came back to India in 2019. Since then, me and my wife, who is also an alumni of 2006 biochemistry batch, Srijata, we have been contemplating about it, but because of all the COVID and other reasons, this hasn't happened. But I promise I'm gonna come to the department very soon. I cannot be thankful enough to all the mentors, all the teachers I had in the department, RB ma'am, SG ma'am, Acharya sir, and I see in attendance we have today NK sir, who was my MSc thesis mentor, and I still remember all my long discussions about science and everything with him in his office. And so everything I am today and I have achieved scientifically is because of what I've learned in the department, and I'm so very thankful for that. And I would love to be in touch. I will encourage all the students to please find me on LinkedIn, get in touch. I would love to help anybody and talk to anyone from the department anytime possible. I agreed when I got the call. Within like few seconds, I was like, yes, ma'am, I will do it. When CRP ma'am called me, so it's just out of all the respect for the department that I have. So from my PhD onwards, I have been working in the field of viruses of the brain, and when I joined my, I'll just share my screen. Just let me know if that's visible.
Yes, it's coming up. Yes.
Sure, thank you. Yeah. So I in PhD I worked on JV, and then when I joined my postdoc, I was working on another brain infecting virus in mice, more as a model system to induce inflammation in the brain and to understand CD8 response. We were trying to understand how the T cell memory establishes in the non-lymphoid organs, like lung. Non-lymphoid organs means lung, skin, brain, and several of those. So this was our model, and today I'm just going to talk about one part of my research where we are trying to understand PD-1's role in neuroinflammation. This was done at Penn State College of Medicine in Hershey, PA, under supervision of Professor Aaron Lukacher. and currently I'm at BBRC Bangalore. Now, we all know about PD-1. We have all heard about it, especially students. You have seen probably empty number of papers being presented in the journal club of the department, how terrible this molecule is, that it makes the immune system to not respond to the challenges of possessed by the tumors, and how it inhibits our system, and how there are several antibodies now being in the market to stop to block this molecule, and hence show some efficacy in the solid tumors. PD-1 actually has been around for a very long time, but it came through the limelight when the anti-PD-1 therapy was used to cure the cancer in President Jim Carter he was president of US, and Keytruda, the drug by Merck, did wonders to the Merck stock options in the market after this news. Well, since then, a lot has changed. I mean, this was back in 2014-15. Since then, a lot have changed, and PD-1 and other checkpoint molecules have got so much of significance, and there almost exponential increase in the number of these indications for which they have been approved by FDA now. This is one of the lists that was updated in last year, and this is still ever growing. So it's been an interesting molecule being discovered in 2000 in 90s, early 90s in Hanjo's lab, and was initially studied extensively in different autoimmune conditions, where people found that this is a molecule which inhibits immune system and prevents autoimmune diseases. Then it was later on when in Chen's lab in Mayo Clinic, where it was discovered in tumor infiltrating T cells, and it also then was further found in various other virus infections, and one of the paper in 2006 from Rafi Ahmed's lab from Emory, which showed that you can block this molecule using a blocking antibody, and yet that restores the CD8 effector function. So that established that the exhaustion as a phenomena is entirely different than energy, which completely decimates the ability of CD8 cells to and inhibits their effector function, and they cannot be rescued. But exhaustion is a reversible process. So this was one of the seminal papers in LCMV model. Now, a lot has been discovered since then for PD-1. Well, this is one, for the benefit of the students, just to show what PD-1 is. It's a molecule that is expressed on several of the immune cells, specifically when we talk about CD8 T cells. It's a molecule that is expressed upon either in acute antigen challenge. That means as soon as T cells or naive T cells get activated, they upregulate the expression of PD-1, and eventually if the antigen is cleared, it gets cleared off. The expression goes down and the memory is established. But if the antigen hangs around for really, really long time, or that what is called as a chronic antigen persistence, what happens, something that happens in chronic infections with viruses, then the expression of PD-1 persists even after the initial burst of activation, and that leads to downregulation of some of the effector functions of CD8 T cells, starting from they first lose their ability to proliferate, and they start losing the expression of IL-2, and slowly as they progress further, depending on the load of antigen and the time period of it, more and more other checkpoint molecules, inhibitory molecules like LAG3, 2B4, and others start getting expressed. Now, this was the story about PD-1. Try to move this away. That's interfering. Yes. Now, PD-1 was initially discovered as a molecule for, pardon me. I'm going to take a pause here. My screen is giving me some trouble. Just give me a minute, please. Yep. So this molecule was was first discovered as an autoimmune in the autoimmune models, and when scientists tried to inhibit the molecule using the blocking antibodies, they found that the mice where the PD-1 signaling was inhibited were succumbed to the infection by the virus. And that led to the an understanding that this is indeed just not an inhibitory molecule, but also a very important component of immune system that stops the immune system to go overboard. Now, patients, we also found that patients treated with therapeutic antibodies sometimes show immune-related adverse effects, which is also the indicator of the fact that PD-1 is a molecule that that is important in maintaining the homeostasis in the immune response. Now, PD-1 expression has also been found and seen in various virus infections, and it has been reported in JC polyomavirus, MCMV, and others. Just to give a brief background on what what CD8 response looks like and the specific cell type of CD8 that I'm going to talk about today, that is memory tissue resident memory CD8 T cell. Once there is an infection, virus levels rise quickly and CD8 response starts to build up. And that gives rise to central memory CD8 response. Now, when this happens in the peripheral tissue or non-lymphoid organs, this peripheral CD8 response goes and infiltrates the peripheral tissue and establishes something significantly different than the central memory CD8 cells, which is called tissue resident memory CD8 T cells. These are CD69 positive and CD103. They can, and they're in some cases they can be CD103 positive or not. Like in this cartoon, which describes the phenotype of tissue resident memory CD8 T cells in lung, they are independent of the infiltration once they are established from the periphery. So this is the cell type that we were interested in. Now, what do we know about the tissue resident memory CD8 T cells and PD-1? Well, it was first reported in human lung tissue, where resected tumor tissues from lung were used for single cell RNA-seq studies, and PD-1 mRNA were detected in CD8 T cells. But since it was a genomic study, they didn't have any surface expression data. Later, around in 2017, two back-to-back papers, one of them being ours, showed that PD-1 is expressed in brain tissue resident memory T cells in chronic infection. The other study was in MCMV model system. The model system we used was mouse polyoma virus. It is a non-enveloped double-stranded DNA virus that establishes a systemic persistent infection in mice. What happens is that, without going in the details of the virus, since it's a model system, I wanted to keep it brief. We inoculated the virus intracranially in the brain, and the virus levels goes up, which leads to activation of T cells in the periphery, say in the spleen in case of these mice, and this seeds in to a brain tissue resident memory T cell population in the brain, and that is established in a persistent phase of infection. As you can see, the virus levels build up initially, and then it is controlled, but it is never completely abolished. The previous graduate student, Elizabeth Frost from our lab, showed that the brain tissue resident memory T cells established in this model had higher TCR affinity, that is T cell receptors from these brain tissue resident memory T cells had higher affinity as compared to the memory CD8 T cells from the spleen. And that led us to ask this question, as in how this, T cells which have the higher affinity against the peptide, viral peptides, is going to be controlled in the brain, because we know that brain is a very sensitive organ with a lot of cell population which cannot be renewed. Hence, we checked whether there will be other inhibitory mechanisms like checkpoint molecules are being expressed on these T cells. So as I explained earlier, what we did was we used a transgenic T cell line, which is against the mouse polyoma virus we are working with, and we injected the virus directly in the brain. At the acute phase of infection, that is early phase of infection of day 9 and day 30, we analyzed the phenotype of the CD8 T cells. As expected, in day 9, the PD-1 expression was high in spleen as well as brain, because, as I mentioned earlier, PD-1 expression goes up as the T cells start getting activated. But to our surprise, at day 30, when the virus levels are almost a log lower in brain and spleen as compared to day 9, the splenic CD8 T cells, memory CD8 T cells, showed no or very low expression of PD-1, while brain resident CD8 T cells express very high level of PD-1. They also express CD69 at day 30 in brain, as you can see here, and around 30% of those express CD103 as well. So this led us to ask the question: is this expression of PD-1 is capable of inhibiting CD8 effector function? And if the ligand for PD-1, that is PD-L1, is expressed on glial, and what is the role of PD-1 in the brain, and how it shapes the immune system? Well, to answer this, what we did was we isolated these CD8 antiviral CD8 T cells from the brain and exposed them to peptide-coated BMDCs, that is bone marrow-derived dendritic cells, in culture, either derived from PD-L1, that is the mice that lacked the ligand for PD-1, PD-L1 knockout mice, or wild type. So these won't have any PD-1 signaling. And as we can see, that both at day 8, that is by the solid round circles, and the hollow squares, the CD8 T cells exposed to the PD-L1 knockout BMDCs express higher level of interferon gamma, while in spleen, this only happened with day 8, but not at day 30. So this showed that at day 30, where the PD-1 expression is higher in the brain, it is going to inhibit the CD8 effector function. So then we asked the question, well, does this happen? This is in vitro. Does this happen in vivo? And for that, we first checked whether the ligand for PD-1 is expressed in the brain, which is PD-L1. And what we found was that out of oligodendrocytes, astrocytes, microglia, and monocytes that we interrogated, astrocytes, microglia, and monocytes expressed good level of PD-L1 on their cell surface. This is a flow diagram with PD-L1 on the x-axis. a shift towards right shows the higher expression of PD-L1. While oligodendrocytes which didn't express any PD-L1 also showed much lower level of viral copy number in them. So this established that yes, indeed, the expression is CD8 T cells express PD-1. There is corresponding PD-L1 being expressed on the glial cells. So the next question was, well, how do we assess whether there is an inhibitory role of this molecule? So for that, we looked at the expression of MHC class 2, which is an interferon gamma responsive gene, and one of the important effector function of CD8 function, right? So what we did was we inoculated the virus into the brain, intracranially into the mice, and at day 8, we looked at MHC class 2 expression on microglia. The thought behind this expression is if there is a higher level of interferon gamma being produced in the absence of PD-1 signaling, that is in PD-L1 knockout mice, we should see a higher expression of MHC class 2 on microglia. And yes, indeed, that is what we found. We found that in PD-L1 knockout mice, there was a higher level of MHC class 2 expression, which said that yes, indeed, in vivo as well, PD-1 is functional and is inhibited function of this. Well, this signal molecule PD-1, this express of resident CD8 T cells, do they have a role in controlling neuroinflammation? So what we did was we took a section of brain from these infected mice at both day 8, that is the early phase of infection, and at day 35, either from mice or from the PD-L1 knockout mice. What we found that I inserted into nanostring genomic analysis, the expression of 245 genes were interrogated, and what we found was 25 genes were upregulated at day 8 in wild type, while almost 50 genes were upregulated at day 8 in PD-L1 knockout mice, that is in the acute phase. While in the latent phase of infection, there was still 27 genes that was upregulated in wild type, but the number went down to just 14 genes. Even the fold changes were much lower. Here, what we are trying to I'm trying to show that the groups where aligned with uninfected mice together. And this happened to the signaling pathways. What we found that yes, indeed, at day 8, there were several of the neuroinflammation and pathways indicating inflammation in the brain were upregulated in day 8 in wild type mice, but all of these pathways were much significantly upregulated in the absence of PD-1 signaling. As you can see, TLR signaling is showing enrichment ratio of barely 0.05, while it's much higher in PD-L1 knockout. What we also found that in the presence of PD-1 signaling, some of this immune response that was upregulated is maintained in a sustained level, but the response is much muted in the PD-L1 knockout, which says that these mice are not being able to sustain a long-term immune response. So we then looked at, well, is there a difference in the memory compartment when we are looking at the expression of the number of antiviral CD8 T cells? DFB LTH359 is the tetramer which is used to stain these antiviral CD8 T cells. And what we found was there was only marginal increase in the number of CD8 T cells, antiviral CD8 T cells in the PD-L1 knockout. Well, there was a slightly higher number of CD103, that is the tissue resident memory CD8 number as well. But this was a very muted response, and we didn't see any difference in the virus levels. So then what we wanted to look at is, well, if there is a reinfection or re-challenge of the antigen, how capable are these cells to counter that? So the mice were first infected with first wave of infection, and we waited for 45 days, after which we did a second round of infection in the brain. Post, we waited for 5 days and then analyzed the number of CD8 T cells, antiviral CD8 T cells in the brain, and also looked at the viral copy number. As you can see, that after re-challenge, there was no difference in the CD8 compartment itself, but what we found that these mice, PD-L1 knockout mice, had much lower control over the virus levels, and the virus levels in the PD-L1 knockout mice were much higher, almost 5-fold higher than the wild-type mice. Well, this says that the CD8 memory T cells, brain-resident CD8 T cells, were not capable of controlling the virus as well upon recall. They also were not very good at controlling the. They were not very good at even recalling the cells from the periphery upon reinfection. As you can see, that the myeloid cells, that is CD11B positive cells in the brain, were significantly lower in the PD-L1 knockout mice. And we looked at some of the inflammatory macrophages using CX3CR1 and MHC class 2 in the brain. And what we found that where in B6, that is wild-type mice, there was almost 22-fold higher infiltration of these cells. Though in PD-L1 knockout mice, this response was much lower. We wanted to understand, is this, if what we have seen here is because the cells that are developing in the absence of PD-1 signaling, are they more exhausted than the B cells? Well, they are functional because they can produce interferon gamma, as we have seen in the earlier slides. Because one of the important papers that was published by John Verey's lab, which showed that just mere absence of PD-1 during the development does not stop the cells, CD8 T cells, from getting exhausted. In fact, these cells get even more exhausted because they express higher levels of EOMs, that is a transcription factor, BLIMP1, and a lower level of TCF1, which is another transcription factor. So as you can, just to keep in mind that if the cells are getting more and more exhausted, they should express higher level of EOMs, higher level of BLIMP1, and lower level of TCF1 transcription factor. And they should also express higher level of other checkpoint molecules like TIM3 and 2B4. So we checked the expression of these molecules on these brain-resident CD8 T cells after day 30 post-infection. But we found that there was no difference in the expression of either checkpoint molecules, neither the expression levels of the 3 transcription factors described by John Verey's lab were different, which said that these cells are functionally quite capable of producing interferon gamma, and they're not exhausted. Well, in conclusion, what I want to bring about is that the antiviral CD8 T cells exhibited higher affinity as compared to the splenic T cells during persistent phase of infection, and PD-1 inhibited effector function of these brain-resident memory T cells both in vitro and in vivo. During the acute phase of infection, expression of PD-1 on brain tissue-resident memory T cells limit neuroinflammation, and the inflammatory signature was much higher could be maintained in the wild-type mice, but when the cells are developing in the absence of PD-1 signaling, this response is much muted, and the T cells are not capable of responding well upon recall. Apart from all these findings, we also found role of PD-1 in the repertoire of the antiviral CD8 T cells, where the splenic cells had much wider clonality. They were there were much diverse clones of CD8 T cells that were available, that were present in the memory compartment in the spleen, but in the brain, the compartment was much oligoclonal. And these are the aspects that lead us to believe, to compel us to understand this molecule even better, because right now in the clinic, the success rate for these checkpoint PD-1, PD-L1 blockade molecules are only 20 to 30%. If we want to bring this even more, we need to probably understand some of these aspects of this molecule. At the end, I'd like to thank my lab, Dr. Lukacher Labs, our collaborator from St. Jude, Ben Youngblood, flow cytometry core, my mentors from Penn State College of Medicine, and all the support we got from Adaptive Biotech and NIH. Thank you. I'm open to any questions.
Questions from anybody?
Thank you, Shwetank, for bringing a very good piece of work and presenting here. If there are no questions, then we move to the next part of the program. And Shwetank, make it convenient to come sometime to the department.
Yes, absolutely. I know I've been trying to, we have been planning to come to Baroda for a very long time, but it hasn't happened.
Make time and come sometime.
I will. I certainly will. Thank you. Yeah. Thanks for the opportunity. It's been a great pleasure. Thank you.
It's our pleasure too.
Yes.
Yeah. So before Professor Pushpa Robin proposes formal vote of thanks, I want to say a few words. First thing is, I want to thank all the speakers who have accepted the invitation, no matter how high and mighty place they belong to. They have all agreed. And very busy people like Ray Cruz could come and present a talk to us. I'm really thankful. And the teaching staff of the department have stood by me in organizing this, and their wonderful cooperation made it possible to organize it without any hitches. Actually, I was in the hospital for about 15 days. I was out of action, but things could happen smoothly because of the cooperation I got. And I want to thank Satakshi, Sandeep, and others who are involved in organizing the program online. I want to thank all the participants because it is meant for them, and they could participate and make a program big success. And I want to thank our Dean, Faculty of Science, for inaugurating the program and also promising financial support for the program. So my big thanks to all the people involved. And I also want to convey my thanks to the VC. He could not come to the program because of syndicate meeting, but he sent his best wishes and for the success of the program. So thank you all. And I request Professor Pushpa Robin to propose vote of thanks. One more thing I forgot. I want to thank ABCD for their cooperation in organizing the lecture series. Thank you.
Yeah, so you have said it all. Professor Atrapa has thanked everybody. She's not missed anybody. I would only like to say that the 3 lecture series that we have seen, Professor C.V. Ramakrishnan lecture series, Professor S.D. Telang lecture series, and Professor L.J. Parekh memorial lecture series have been possible because of funds, donations by our alumni. We are always grateful to our alumni who, even after having left the department, continue to support the academic activities in our department. I'm really very proud of the 3 young alumni who presented today. In just 1.5 decades after they have left the department, they have made a name for themselves as independent scientists. We pray and hope that you continue to grow and continue to do well and contribute to science. Thank you so much. Thank you, everybody.
So we meet sometime once again. The members who right now are not part of the department, and for the rest, tomorrow we have a non-teaching day. And we meet again. Yeah.
Bye.
Congrats, all of you. Nice program.
Yes, thank you, sir.
Bye. Bye.
Okay, bye-bye.